When you're in the auditorium.
Welcome to session. All right. Session 11, Sturgeon. I'm John Kelly, CDFW, my co-chair in the Sturgeon PWT, Alicia Seaschultz from DWR. We'll be running the presentations from over there. We're going to get started with our first speaker, Josh Gruber.
Okay. You know what?
So we have to first.
There we go. Should be pulling it up. It's not showing it up. Here?
Yep.
Okay, good morning, everyone. Hopefully you can hear me. My name is Josh Gruber. I'm a fish biologist from the Red Bluff Fish and Wildlife Office in Red Bluff, California. And I'm gonna present to you guys the data that we collected from 2016 to 2019 on juvenile green sturgeon outmigration. So the North American green sturgeon is a long-lived anadromous fish, which spends most of its time in the Pacific Ocean. It ranges all the way from the Bering Sea in Alaska to Ensenada, Mexico. These fish are divided into two distinct population segments. Today, we're going to focus on the southern DPS, which returns annually to the Sacramento River, primarily the upper reaches, to spawn on an annual basis. And that puts the population at risk of them all being in one place at one time, or at least a large majority of it for that matter. Also, indications since the 1980s of low juvenile recruitment to the Delta has led people to believe that the population itself is in decline, and a majority of their spawning habitat has been blocked off due to the construction of dams within the Central Valley. So in 2006, this species was listed as threatened, and that listing was, again, primarily due to these three factors, as well as there's just a lot of uncertainty at all the life stages, and we knew very, very little about the population as a whole. Since the listing, we've gained some information on various life stages, primarily the adults and the spawning aspect of things, and that spawning is recruiting to larval fish, but we don't know very much about these juvenile fish. Their status has been reaffirmed in 2015 and 2021. And to kind of bring home like the limited or the lack of knowledge on this life stage, I've pulled something out from the 2021 status review, which indicates these juvenile fish move from the upper river down to the Delta between 6 months and 2 years of age. That itself is not anything to be too shocked by, but what I find kind of interesting is that this citation dates back some 60 years. And to think that we know, nothing new in the last 60 years is pretty astonishing to me. So that kind of sets the basis for this study. And here we've identified the research goals was to try to hone in on the spatial and temporal distribution of these age 0 fish in the upper river and figure out when and how they're migrating from that upper river down to the estuary environment, with hopes of getting some information on different travel rates, as well as different patterns of outmigration they may be using. And again, because this is very novel research, we were hoping to get some information as to the survival rate of these fish making that migration. I want to also point out, though, that this was not a robust survival related study, but we're just hoping to see what we could gain from these tagged fish. So in terms of sampling, we used a 16-foot benthic trawl that we deployed off the front of an inboard jet boat. The net was then towed downstream at a speed slightly faster than the river current. And the goal there was to open up the net, but make sure that it maintained contact with the river bottom, because the expectation is these juvenile fish are out foraging and rearing along the river bottom. We then scoop them up and collect them in the net. Our sampling targeted a 50 river kilometer stretch of river from the Red Bluff area down to Corning, California. We chose this area because it was the one we were most familiar with. And also it's kind of smack dab right in the middle of the species spawning grounds. So we were kind of targeting the center of where we expected these fish to be. Our sampling occurred within the first 4 to 6 hours after dark. Or sunset. This is when we expected those fish to be most active, based on the assumption they were hunkering down in the middle part of the day and then come out at night to forage, like most juvenile fish do. We started our sampling in October when we felt these fish were of taggable size, and continued that sampling into the winter months until we either were not catching any fish or our acoustic data indicated these fish had left the upper river. And again, we were primarily targeting these age 0 fish. So in addition to that, targeted sampling effort, we also wanted to take advantage of some additional juveniles that may be caught in a long-term juvenile Chinook monitoring project that we do at the Red Bluff Diversion Dam. The program's been in place for 20 plus years, and we've caught a few handfuls of these juvenile fish. So didn't have major expectations there in terms of getting a lot of fish, but we wanted to take advantage of anything we did collect. Individuals that we did collect over 150 millimeters that were in good health, so to speak, were tagged using a variety of microacoustic JSATS tags. The top 2 are tags produced by ATS. Many of you recognize them as they're utilized in Chinook salmon, but the battery range is, right around 60 to 100 days. And so that was somewhat of a concern to us. So we also wanted to utilize the larger P&L tag, which is quite a bit larger in size, but has almost 3 times the battery life as some of the other smaller tags. We weren't sure exactly what size fish we were going to catch, so we wanted the option to do the bigger fish, bigger tag kind of thing. Overall, keeping our goal of a tag burden less than 2%. And we balanced battery life with detection efficiency by using or selecting a 10-second PRI for these tags. The tags were implanted using a flankless, a flank incision with no sutures. This was a method developed by P&L, and within their study, they found that the incision healed within 20 to 30 days, and there was little to no tag loss utilizing this method. Given the fact that we weren't really experienced fish surgeon, we don't have hundreds of thousands of juvenile Chinook tagging practices underneath our belt, or let alone any sturgeon. We felt that the use of or the lack of sutures would limit our impacts on these fish by inadvertently sewing up their internal organs. So the photos here kind of show you that process we go through, making an incision in the upper left-hand corner. You can see you'd make the incision between those 2 yellow lines between the 3rd and 4th ventral scute. And then at that point, the tag was inserted and leaving that small hole there in the lower right-hand photo. Surgery took about 2 to 4 minutes, and then the juveniles were left to recover within an aerated bucket of water for about 30 minutes to make sure they were good to go before we released them into the basically the sampling location where they were collected. Once released, our fish were tagged, tracked using the extensive JSATS acoustic array that we have within the Central Valley. The receivers are deployed in a gated fashion across the river and able to basically trees or some sort of unmovable object along the riverbank. The receivers are serviced every 100 days to replace the battery and offload the data. Once the data is back on station, we would remove false detections using a 2 PRI detection filter. This filter is a little bit more liberal than, say, what's used for Chinook salmon, but we felt these fish having a more benthic orientation than, say, Chinook salmon. We wanted a bit more liberal filter in order to ensure that we got all of our detections. The river itself was divided up into 3 different reaches. I don't know if you can see that very well, but we have the top part is defined as the upper reach. Essentially, this incorporates the known spawning area of the species, and it extends down to river mile 395, which is the Highway 32 bridge between Chico and Hamilton City. And then the middle and lower reaches were essentially divided in half, but we had to make our lower and upper bounds of those 2 reaches to be a location where we had a receiver for 4 years, and that receiver also had to have a high detection probability so we could ensure that fish were tracked going out and into the next reach of the river. Looking at the data, again, we were trying to figure out some different travel rates. One of the rates we were looking at was the residence time within a specific reach. Basically, the number of days from the very first detection to the number of days from the last detection. Relatively simple. And then expanding upon that, we wanted to figure out the velocity of the fish through any given reaches. So we could compare between those 3 different reaches, essentially taking the distance traveled between the first and the last detection divided by that residence time or time spent within that reach. We teased the data a little bit. One of the things that we did is we didn't include any of the movement data within the first 24 hours. And the purpose of that was to limit any handling effects of collecting the fish, tagging the fish, and releasing the fish. We saw some fish moving right away, but then not moving after that. So we eliminated that data. And we also made a minimum criteria that fish had to pass 3 of our receiver gates in order to be included in our analysis. And then we looked at velocity rates at a much finer scale, basically in between each of the receiver gates. And that's highlighted here with the blue arrows. And that was just to try to figure out what are these fish doing over a shorter distance rather than a big, long reach of river. And that was done in the same way. You'd take the last detection at the upper receiver, calculate the distance, and divide it by the time. These travel rates were then combined by reach and compared with the LMRE package in R to determine whether there was any significant differences between them. Survival was done very basically by dividing the number of fish that were detected here at the River Mile 170, which is the I-80 and Tower bridges that many of you are familiar with, by the number of individuals that were tagged in a given year. Our trawling was pretty successful. We were able to collect 183 individuals. Their sizes ranged from 72 to 322 millimeters. You can see on that map there's some green stars. I found it interesting that nearly 80% of the fish that we collected came from those 5 sample sites. So certain areas were repetitively sampled and provided a number of fish for us. In the end, we tagged 88 individuals ranging from 152 to 322 millimeters in total length. We also added 10 individuals from the screw trap program, and you can see there kind of the breakdown of the tags we used. Again, primarily utilizing those ATS tags. And we were able to keep our tag burden below that targeted amount of 2%. So my little icons here represent the 98 fish that we did tag. 3 of those fish were not detected within the first 24 hours. 3 more were not detected. I'm sorry, let me back up. 3 more were not detected in the array at all, and 3 were not detected within the first 24 hours. And then 18 other individuals were removed because they did not pass 3 receiver gates. So with that, we had a total of 75 individuals that we tracked within this study. Here's a series of box plots to kind of highlight some of the travel rates we looked at. Starting on the left, we have reach residence time. On the y-axis, you have the time spent within a given reach, and then the x-axis breaks down the reaches from upper would be on your left, and then extending to the right being the lower reaches. You know, it's pretty clear with that box plot to see that the upper reach had a much higher residence time, and specifically that was roughly 21 days compared to 2.8 and 0.9 days in the middle and lower reaches. So there was a big difference there. And as you would expect, because residence time is related to reach velocity, we saw a much lower reach velocity in the upper reach compared to the middle and lower reaches. Velocities were 2.0 in the upper and 32 and 38 in the middle and lower reaches. But when you break that down to, you know, the finer movements in between the individual receiver gates, we didn't really see a velocity difference between those 3 reaches. So kind of interesting data there. And, oh, sorry, I forgot to title through basically our significance there on the bottom, which we have discussed. So another way to really look at outmigration is through waterfall plots. So we've done that here. Each of the 4 waterfall plots represents a brood year, so to speak. The upper left being 2016, down there to the lower right being the 2019. On the y-axis, you have river kilometer, with the top being the upper river and the lower being the lower river, and each river reach is separated out through the dashed lines. Individual fish have, you know, their own line, and I'd like to point out that, you know, looking at this graphic, although they're all the same size, the x-axis or the time component is not standardized between individual years. So keep that in mind when you look at these graphics. But what you see is what we just looked at. There's a significant amount of time spent in the upper river, indicated by those horizontal lines versus that vertical lines going through the middle and lower reaches, indicating those fish are moving faster through those sections. You'll also notice that there are some horizontal lines in the middle and lower reaches, indicating fish paused in those sections. And so we basically identified this as two different outmigration patterns. About 2/3 of the fish made a continuous outmigration pattern, meaning once they hit that Highway 32 bridge, they migrated down to Sacramento in a pretty straight shot within 7 days, so to speak. Whereas 1/3 of those individuals migrated down and paused somewhere in the middle and lower reaches, extending that travel time in excess of 7 days. And if you look in the 2017 plot, you can see some of those fish spent, nearly a month within that middle or lower reach. Here's just a real basic graphic on survival. It indicates, essentially we had pretty high survival, ranging overall the study being 65%, but ranging between 35 and 95%. So again, we mentioned some of this before, but these fish are spending a significant amount of time in the upper river, as indicated by those long horizontal lines. But they start that downstream migration in November and continuing into March. So we all as fish biologists, or at least most of us in here as fish biologists, want to ask the question, what is cueing these fish out of the upper river, right? And most fish biologists are going to tell you two things that make juvenile fish move are increases in discharge and turbidity. So I plotted on the top discharge and in the bottom turbidity here over those 4 years of our sampling. And I want to point out in 2019, that pulse flow that you see is kind of, I use the terminology as your knuckles there in the beginning of 2019. There's some erroneous turbidity data that I didn't tease out for this, and I apologize for that. But what I'm going to do now is plot the dates and the times in which all these fish exited the upper river. And I challenge anyone to bring a data set together that shows as much correlation between temperature, or excuse me, between flow and turbidity with juvenile fish outmigrating. And then to further that, we talked about fish that stopped in the middle and lower sections of the river. If you look at the waterfall plots, in addition to the flow and turbidity, what you see is those fish that did pause in the middle reach, their secondary downstream migration was cued on the subsequent flow events. So whether it's coming out of the upper river or moving down through the middle or lower reaches, it is highly correlated to flow. So the other thing to keep in mind is that, what's making these fish pause in the upper river. I've given this presentation a couple of times and people have suggested, maybe size is a component. So we looked at that. You can see there is some differences in the total length between the two groups. The range is about 20 millimeters. And overall, there's a decreased probability of making a step migration as size increases. I can't say that I'm 100% sold on this because, our total length is represented at the time at which the fish are tagged versus when the fish are actually out migrating. And we all know that we had significant, significant residence time in the upper reach. So it may be somewhat misleading. So with that, we'll just review our research goals. Again, these fish are rearing above river mile 395. Hopefully when they do the next status review, they can update that citation to bring it more in date, showing that these fish are all exiting the upper river at least age 0. We identified 2 different outmigration patterns, and it may be driven by size, but it's definitely correlated or starting off or cued through increases in turbidity and flow. And survival is high, definitely compared to the Chinook salmon studies we've seen within the Sacramento River. And I also say that this is an absolute minimum survival because I feel with that long residence time, our tag life is impacting our ability to detect survivable, fish that are actually surviving. So with that, sorry, I raced through the last couple slides, but I ran long. I'll take questions.
All right, thank you, Josh.
We'll take questions from the audience, and anyone listening remotely, just type your questions into the Q&A box and we'll try to get to those too.
Go ahead.
Hi, really cool talk. Do you know those spots where they stop? Do they happen to be holding on a particular habitat? Is there microhabitat in those locations, or do you think it's purely they move with water and when it flows, it flows?
That's a great question, and we don't have the resolution in the receivers in the middle and lower reaches, so we can't determine whether a fish is stopping at a particular spot. We didn't see a high residence time at any of our receiver gates, primarily just between receiver gates. My personal assumption that I have not been able to validate yet is I think it's like people surfing in the ocean. They catch a wave for a little bit, and at some point they fall off the wave and they just hang out until the next wave comes. That's my cartoonish mind working. Any other questions?
Yeah, I've got one from one of our listeners. Erin Greenwood wants to know, is survival correlated with flow?
Did not look at that. I would say the biggest concern we had, or the biggest issue, the drawdown in survival, was in 2017, where we tagged fish and there was a really prolonged period before these fish migrated out of the upper river. So the delay in those winter runoff events prevented us from detecting fish surviving to the delta. And I am just glancing the surface of this research that we did. I've kicked out a technical paper. We're working on a manuscript right now, which is going to really hone in on what those out-migration cues are specifically. So, if you're interested more, make sure you check out all the stuff we're producing beyond just this topic or presentation.
I think we can take one more from the room and then we're going to move on.
Do you think that it's the percent change from the base flow, or is there some triggering level of velocity or something that triggers that, do you think?
So you raise a really good point. We are identifying a specific change in flow rather than a specific CFS volume, because every single fall is different depending on management strategies for that given year. We did not look at an actual river velocity within these particular sites, but there is definitely a percent change cue, and it's, not to let the cat out of the bag, it's about 120%.
All right, I think we need to get moving on. So I'm sorry we couldn't get to some of the questions online. Our next speaker is Marc Beccio from CDFW.
Okay, thanks everybody. My talk today is going to be on the annual recruitment and movement patterns of juvenile southern distinct population green sturgeon in lower Sacramento River and the estuary, basically. I want to thank my co-authors, Nick Bauer, my supervisor, John Kelly over here, and Colby House from Fisheries Branch. Okay, for our acknowledgements, I want to acknowledge our funding. We got a Section 6 grant from National Marine Fisheries Service from July 1st, 2018 through June 1st, 2021, and then IEP picked up the slack after that through the end of this year. And a special thanks to my long-term scientific aide, Jimmy Carlson. He spent north of 250 days with me out in the Delta, and including a couple of days when we really shouldn't have been out there. If you guys have been out in the Delta in the vicinity of Sherman Island, you probably know what I'm talking about. That's why it's a windsurfing hotspot of the world. Okay, introduction. As Josh mentioned, they were federally listed as threatened by the service in 2006. Critical habitat was established in 2009, which includes pretty much the Sacramento River drainage, the Delta Bay, and coastal waters all the way up to Washington. The demographic criteria came out with a recovery plan, and this was actually before we started our survey, but turns out that some of the data we're collecting can kind of be useful to address these two issues. One is a net positive trend in juvenile abundance and subadult abundance, and then a broad distribution of size classes through multiple cohorts that are stable. So like I said, we started this study in 2015. That's when I came onto the department. It was kind of a pilot study at that point. Nobody had done really any dedicated capture and observations of green sturgeon in the Delta since that Radke paper that Josh mentioned. So a little bit of method development went into this before we kind of got dialed in. I want to say you can ever get dialed in, but we made some good progress there. So a method of sampling we're currently using is we're using a 300-foot gill net. We deploy that at the Sherman, in the vicinity of Sherman Lake on the Sacramento River main channel. First couple of years we bounced around. We tried all the way up from Tisdale on the Sacramento River down to this area with very little success, and it was getting really frustrating over time, but I can go into that reasons for that later. Once we capture fish, most of these fish are age 1+, so fish that recruited from the previous spawning year. And based on their size at capture, we also get some larger individuals. We use 69 kilohertz tags of V9, V13, and V16 tags are differing battery lives, much longer than the JSAT. So V9 is basically a year detection worth of battery on a V13, about 5 years, and the V16s on some of these larger fish we put in get 10 years of detection data, so we can track these fish probably after I'm retired. And then we have an array of acoustic receivers that are deployed in the estuary and bay. I want to also mention that when I first started this project, that was a golden age of 69 kilohertz array, where there was like 240 receivers from the upper Sacramento River out to the Golden Gate. 2017, funding from the UC Davis Biotelemetry Lab stopped. We were kind of left in the dark. So all of a sudden the receivers were gone. We're tagging fish, and I had to scramble around, beg, borrow, and steal to get some receivers and deploy them myself. So that was kind of a challenge. Our receiver array location, now this is really dummied down from the original array. This is just the receivers we are maintaining right now. Our sampling site is right there at Sherman Lake, kind of just above the confluence of Sacramento and San Joaquin River. That's where we kind of dialed in and found out where we caught most of our fish. Originally we're fishing a little bit farther upstream, but in the last 5 to 10 years, the amount of, you've heard of probably in previous presentations on submerged aquatic vegetation, emergent. Our bane is drifting aquatic vegetation. So even down in the water column, you've got massive amounts of Brazilian waterweed, coontail, and even water primrose stems just fouling up our nets. And we found out in this area here, a little bit less current and a little bit better, cleaner sampling events. So we kind of dialed in on that. So I'm gonna go into some of our results. Pretty simple, but initially what we're doing is we're just uploading our data into the view program, and basically that we just put all our receivers from one array in there, gives us the locations, fish IDs, and the time of arrival and departure. And also I want to emphasize that these fish aren't continually present in one place. They come and go quite a bit, as you'll see. And then we're doing some more advanced analysis in R. We're going to probably look at a bunch of covariates, and I'll talk about that a little bit later on. But this is just kind of some bare bones analysis just to see how many fish we pick up and where they show up. So here's our results. We tagged in basically 7+ years. We've tagged 197 juvenile. We've captured 197. 190 of those have been tagged. We had 3 mortalities and a couple fish that were too small with the tags we had on hand at the time. 83% of those are like age, we call them age 1+, so basically from the previous recruitment year that made it down to the Delta. As over time, our most productive sampling months are the summer months, but we've been out there, we pretty much fish year round, 1 to 2 days a week. A little bit of caveat on the winter time, sometimes we're busy doing fish rescues or other tasks, but if you looked at the CPUE over time, we're still catching a heck of a lot more fish, and our CPUE probably in the summer months is well over 2 to 3 fish per sampling event. These are just really beautiful fish. These are all age 1 fish that were captured in 2020. Pretty good size. They really put on some bulk and size in the Delta during the time they're rearing there. And this is kind of to highlight the influence of what water year types have on fish. So the caveat here too is a little bit of method development in those first 2 years, but as you recall, 2013 through 15 were kind of a historic drought. I was just scratching my head. It's like, where are these fish? Why aren't we catching them? And it started to make a little sense after a while. So, we had a really good epic wet year in 2017. So 2018, we're starting to catch fish. We didn't really get out there until midsummer to start sampling. So, but our CPUE is close to 1 fish per trip. So it was pretty good. A couple drier years. And then we had another really good wet year, 2019, with some high outflows, and our CPUE is basically over 1 fish per trip, which over the course of the year, that's pretty good. And quite a few fish in the summertime. I think we had one net set where we caught 10 fish, and that was just awesome. So, and I want to emphasize how fast these fish grow. When we start encountering these fish in the Delta in the beginning of the season, they're averaging a 35 centimeter fork length and, 300, 400 grams. They really start bulking up in May through June. I want to also talk about I should have a graph here with water temps, but starting in May, the water temps in the Delta start hitting about 19 and up to 23 degrees. There's no stratification even at that depth. The temperature bottom and top is exactly the same. But these fish are really, really putting on they're really growing fast. So obviously that area of the Delta is really, really good for rearing. Just another picture of these real pretty fish. That guy in the middle there is a white sturgeon. Interestingly enough, we're sampling, we get about 5 times as many juvenile greens as white. So there's a different, species preference in rearing habitat. We just don't pick up that many whites at that location, which I find is really fascinating. We do a little bit of what we're doing. We finally have our acoustic release receiver, our tagging site, but all this data here is from a portable receiver that we deploy while we're sampling. Kind of shows us residence time at our sampling site. Some fish we tag, we don't see them again at the day after they're gone. We pick them up somewhere else. But some of these fish hang around for a long time. I'm thinking some of these fish in 2020, where we're seeing in excess of 600 days, we probably have shed tags or maybe unfortunately fish mortality, because we're still seeing those fish. I doubt they're there still. And we pick up tag detections almost every time. And like I said, these tags are really long lived. So it's a chance we lost some fish out there. I want to go also look at the detections by receiver location. General trend is these fish start to move downstream. It's kind of gradual and it's not like salmon that they decide to really get going down there. They move around a little bit. There's some evidence to show that also some of these fish will move upstream and then south into the San Joaquin drainage. And some of these things are just detected all over the place. As you see, I've got a couple of slides at the end will show what these fish are moving. And the arrival after tagging at these sites is highly variable. But this is just kind of to show you our detections by receiver location. I standardized these. I pulled out all the receiver locations that were there in the golden age of the thing. So these are just sites where we have consistent receiver locations. So you can see low numbers of fish the first few years. 2020 was our banner year. We got a lot of fish, and kind of what happened there, our detections at Benicia and Golden Gate are kind of low. We can't really infer survival from this, but when I looked at the data, it turned out we ran out of V13 tags early on. We just didn't anticipate catching this many fish. There were some V9 tags, which have a year battery life on the shelf, a couple of years old. We used those. We tagged half those fish with V9 tags. So battery life on those was a lot less, and probably some of those tags winked out before they ever made it down to Benicia or Golden Gate, as you'll see from some of our data showing arrival at receiver locations. So this is some of the fish that moved upstream. This is basically mean time from the first detection to tagging site up to Rio Vista Bridge. This is only about 10 kilometers from our sampling site, or 9 or 10 miles, sorry. The farthest upstream of any fish we detected, I think we have a half a dozen fish. DWR had some receivers at Cache Slough Complex. We had 6 fish that went up that far, none farther than that. And they were only there for maybe a couple of days and then started to head back down. So some of these fish, like I mentioned, they move over to the San Joaquin side. These are at, this is detections at Antioch Bridge. Maybe 10% of the fish in any given year will head over to the San Joaquin. And pretty highly variable when they are initially detected there. Montezuma West, interestingly enough, we have receivers at the east entrance of Montezuma Slough and in the middle up at Nurse Slough. We get very few detections there, but I think about a dozen fish we detected in the Montezuma West receiver. And my theory is here, they kind of went down, came up Grizzly Bay and just nosed up into the Montezuma Slough from there, based on not seeing too many fish at the other 2 locations. They're not traveling through Montezuma Slough in any great numbers. Then once again at Benicia, we've got quite a range in initial detection. Actually, we've seen detections as quick as 2 days after tagging, and that's about moving at 15 kilometers a day from our tagging site to Benicia Bridge. And some of these fish, they don't show up until quite a bit later, over 500 days. We pick them up there. So really interesting. They're just moving around and taking their time getting down there. And detections at Golden Gate. My caveat here is we There's no receiver array really at Golden Gate permanent receiver array. NIMS uses acoustic release receivers there to deploy their JSAT receivers. So we have periodic deployment of receivers there 4 to 6 months a year. So we're probably missing a lot of detections of fish that are moving in and out of the gate, but well, at least we're getting some data there. And so a big shout out to the NIMS folks that are using those. That gave us some good supplementary data because we just don't have the resources to put receivers all over the place. So here I'm not going to talk a lot. I'm just going to show you a couple. This is 2 examples of fish, 2 different very life histories of movement in the Delta. This one fish here we have, I'll show you next, is we tagged it in 2016. It's the third or fourth fish we tagged overall. And I don't know if there was a big meth concentration in the Delta at that point in time, but this fish just did some wild stuff. So I'm just going to go through these slides. And the DAT is days after tagging, so you can see what this fish did. Okay, we tagged, there's our tagging site. Same day, up to Rio Vista Bridge, and the 0 to 49 days, that doesn't mean it was consistently detected there, but it was detected nowhere else. So between 0 and 49 days, that fish was basically in that area. Day later, it shows back up at our tagging site, back up to Rio Vista Bridge for a day, back down to our tagging site, back up to Rio Vista Bridge, back down to our tagging site for about 10 days. Then it goes down to the Benicia Bridge for kind of a long period. Then it goes over to the Antioch, to the San Joaquin for a little bit. Back up to Santa Clara Shoal. So it's historically, there's been some green sturgeon captured there. Originally, they thought maybe they originated in the San Joaquin drainage, but I think some of the Sacramento origin fish use that habitat, as this fish is demonstrating. Back to Antioch, down to Benicia. Then we picked it up a few days later, Carquinez. Then it hung out in San Pablo Bay for quite some time. Decided to head back upstream at Carquinez again. Day later at Benicia. Then it's back in San Joaquin. Back up to Santa Clara Shoal, in our tagging site. Rio Vista again. Down to our tagging site. Benicia Bridge. And this is the last time we detected it for about 25 days down at Carquinez. At that point, the tag battery probably died. Okay, now this fish is pretty cool. It's one we tagged in 2018, the end of the year. Jumped up to Rio Vista Bridge for a little bit, back down to our tagging site, Benicia Bridge for a kind of extended period, to the Golden Gate. This fish is probably going in and out of the bay at that point, we're thinking. Back up to the Benicia Bridge, back to the Golden Gate for a pretty extended period. And then it goes down to Monterey Bay. So these are detections at the cement ship. These receivers were deployed for white shark research, but happened to pick up our tag. Up to Año Nuevo for a little bit, back down to the North Monterey Bay, back to Año Nuevo, Point San Pedro, back into the gate, and then back to Benicia Bridge. And that's the end of that fish. We haven't picked it up since. So some of my conclusions here is recruitment of green sturgeon to age 1 class is much greater in wet years. We just get, high outflows. I think the survival is much higher, and the fish show up in the Delta in much higher numbers. They're present year round in the Delta at our sampling site, but we definitely catch more fish this time of year. Growth rate is extremely rapid. These fish really are bulking up from mid-spring through late fall. And they range widely throughout the bay and Delta, as you saw from a couple examples of those fish. We've got a couple more fish that do that. Some do more of a traditional thing where they kind of gradually move downstream. And as you see, timing of ocean entry is highly variable. And then some individuals return to this. I think we have about an instance of 10 fish that were detected at white shark arrays. And this data is from Dylan Stompe, our next speaker. So he sent me those tags, and there was a number of fish that were detected either Point Reyes or Point San Pedro, and then ended up going back into the bay after that. So it's kind of interesting. A few of those fish, if you look at the timing when they went back into the bay, it's the same time as adults are migrating in for spawning runs. So I don't know if they're trying to pick up some learned behavior or just a coincidence. We don't know yet. So as far as future studies, there's a tremendous amount of covariates we could look at to determine why these fish are moving, when they're moving. Are there any patterns? Is it based on water temp, flows, salinity gradient? They seem to be pretty, the way they move around, it seems like they're pretty good at osmoregulating and go back and forth from fresh and salt water. It'd be really cool if we had, there's probably, I don't know, hundreds of VR receivers, 69 kilohertz receivers sitting in warehouses, and not being used right now. It'd be great to have those back out in the Delta and look at more fine scale movements for these fish. We really don't. With this really skeleton array, we don't know what they're doing in between detections of these other things. and also look at some manual tracking. Our ping rate on these tags is about a minute. It's kind of hard to do manual tracking, but if we have continuous pingers, we could, stay out there for 24 hours and watch what these fish do after tagging. It'd be really interesting. So with that, time for some questions.
All right, thank you, Mark. We have time for a question or two, I think.
Well, it's either good or bad.
Any questions online? Okay. Well,
thank you.
Oh, wait,
Okay.
Do you have some fish moving back and forth?
Right.
I can get that that's idle influence or like periods of low flow where the fish is moving back and forth.
Doesn't really seem correlated to that. I mean, I think sometimes it takes them, better part of a day to move, say, from our tagging site to Rio Vista or back down. So we just don't, our receiver, our ray scale is not really tight enough to determine that.
Yeah.
So definitely be kind of cool to look at that, but I'm thinking this maybe it's just, back more like striped bass and salmon, that's all I'll say. So hopefully we can find some patterns on that.
I'm curious whether you've had any recaptures.
We've had one physical recapture, and that fish I think we tagged it in November. We recaptured it in March, about 100 days later. And that fish, the water temperature I looked over that period of time was average 10 degrees C. And that fish grew 1/2 centimeter and 20 grams. So, and that's, that was the coldest part of the year. So I think we had one other recapture. We caught it 2 weeks later. I didn't even bother to take measurements on that. I just pulled it up, go, oh yeah, we caught that guy before.
all right. Thank you, Mark.
Okay, thanks, John.
Our next speaker is Dylan Stompe from CDFW.
Hey, my name's Dylan. I'm going to be talking about sturgeon abundance trends and recent mortality in the San Francisco estuary. So a little bit of background on sturgeon, specifically white sturgeon. They have slow growth. This is from Black Bernardelle 2019. We allow harvest of fish between 40 and 60 inches, which corresponds to 9 to 17 years old. So pretty old fish that are being harvested. Fairly late age of maturity. It's from Chapman's dissertation from Davis in 1989. Potentially needs to be updated, but best information we have. So they're maturing within and outside of our harvest slot. We have infrequent recruitment. This is data from the San Francisco Bay study, our juvenile year class index, and it lines up really well. If you look at that, the line on there is delta outflow, really only recruiting during wet years, like Marc found in his data. And we have 6 to 8 years approximately between good recruitment events. And that's also reflected in our adult data. These are adults that were captured by a guide that I'm working with. So you can see it partitions really nicely into 4 pretty distinct year classes, which again lines up with our high outflow events in the delta. A little history on the fishery. We used to have a commercial fishery for sturgeon, both white and green sturgeon. It was really incidental to the salmon and shad fisheries. This picture is from shad fishery on the Sacramento River in 1916. They weren't worth very much, but they were sold as sort of a cheap fish protein. Caviar had not been discovered as a luxury product yet. So I read an old paper where it was just given away for free. You can see the commercial landings here. It peaked in 1887 at 1.7 million pounds, clearly more than a slow growing late to maturity fish can handle. So it was closed between 1901 and 1915, opened again for a little bit as a test fishery. Was deemed not sustainable, so it was closed all the way up until 1953, both recreationally and commercially. Currently, we do have a recreational fishery for white sturgeon. The green sturgeon fishery was closed in 2007 after it was listed in 2006. So the white sturgeon fishery was opened in 1954. Since 2007, anglers are required to buy report cards. They're required to turn in their report cards. We only get about 30% back. And from that, we know that at least 1,500 to 2,500 fish are removed each year. white sturgeon removed each year. And this is a pretty typical day on a sport boat. This leads to pretty high exploitation rates. This is from Blackburn et al. 2019. They found exploitation rates between 8 and 30%. I'm working on updating this right now. Theoretically, there's no exploitation for green sturgeon, but there probably is some poaching. We also have mortality in the form of ship strikes. So some fish are killed by contact with propellers from ships and tugs and shipping lanes. Poaching, for example, in 2021, a sturgeon poaching ring was busted, or at least 36 sturgeon were removed. And these people target large gravid females, specifically for the caviar trade. So maybe not very many fish, but important fish being removed from the system. Sea lion predation, if you ask any angler, this is the biggest issue for white sturgeon and every other fish that we have. It does happen. This is from South San Francisco Bay. It's unquantified and probably not a huge issue, but it is another source of mortality. Sturgeon are salvaged from the pumps in the South Delta, and presumably some are lost at smaller unscreened diversions within the Delta. And then recently we have harmful algal blooms. This isn't something we've had to deal with before in the past, at least to this degree, but it certainly is now. So we had a big harmful algal bloom in 2022 caused by this thing. It's Heterosigma akashi woe. It causes these red or brown tide events. That's San Pablo Bay. It's not what it normally looks like. It's normally much more green. And it's a marine phytoflagellate that's globally distributed. It has recorded blooms with associated fish kills in Japan, China, New Zealand, South Africa, Chile, Europe, and the Pacific and Gulf coasts of North America. It's thermal and euryhaline, can tolerate temperatures of 5 to 30 degrees Celsius and salinities of 2 to 50 parts per thousand, with maximum growth right around 23 degrees Celsius and 25 to 35 parts per thousand. It has a resting cyst stage, so it can drop out of the water column into the substrate and stay in the substrate for up to a couple of years, letting it go through adverse conditions and waiting for good conditions in order to bloom again. It also produces brevetoxins, which have a neurotoxic effect on fish. At low concentrations, it can have sort of sublethal neurotoxic effects. At high concentrations, it can cause paralysis and death. And it produces hemolytic compounds, reactive oxygen species, and polyunsaturated fatty acids that may directly or through tissue injury cause excessive mucus secretion. These mucus secretions can coat the gills, which can either directly cause mortality, or it can, in a combination with low DO caused by just the physical bloom, combine to cause mortality. It's caused by high macronutrient loading and those ideal salinity and temperature conditions. But kind of interestingly, its toxicity is increased at not ideal conditions. So when it's lower salinity, toxicity is higher, and also when it's nitrogen or iron limited, but not when it's phosphate limited. So getting to the bloom that we actually had in the bay, it was first detected right around Alameda in late July. Started to radiate out into the bay in early August. You can see that red there this is chlorophyll A from Sentinel. It's the primary pigment in Heterosigma. By August 20th, it had radiated throughout San Pablo Bay, Central San Francisco Bay, and the South Bay, with a really high concentration in that sort of Central Bay region, South San Francisco Bay. By August 26th, it was everywhere at high concentrations. So really, really high concentrations from San Pablo Bay down to the South Bay. I started seeing supersaturation of DO in San Pablo Bay around this time. I was out sampling for white sturgeon for our Marc recapture study. It died off starting sort of late August. You can see it's subsiding everywhere, not quite as much in San Pablo. And we had a DO drop system-wide because of this. By September 5th, it was pretty well gone, and DO conditions had returned to normal. So this DO conditions I was talking about, this is at San Mateo Bridge, marked by the black bar on the map. You can see in late August and early September, it gets down below 2 milligrams per liter. which is probably lethal to a lot of fish, at the very least, very, very stressful. San Pablo Bay didn't get quite as bad, marked by the star here. This is the China camp gauge. It did get low, got 3 to 5 milligrams per liter, which is probably stressful, maybe not lethal. While this was going on, we had a really historic heat wave. Everyone was here, I'm sure you remember this. Lasted 1 to 2 weeks, and this increased the water temperatures, which probably encouraged the bloom, but it also increased degradation rates of fish that were killed, which made them hard to survey. We also had severe to exceptional drought throughout pretty much the entire state during this time period, which meant that we had really low outflow conditions to flush out those harmful algae. This delta outflow in August through early September. So it gets down to around 2,000, 2,500 cubic feet per second. Compare that to a wet year, 2017, it was between 6,000 and 13,000 cubic feet per second. So very little fresh water coming through. This all combined to cause a pretty massive fish kill. It was the largest recorded fish kill in San Francisco Bay, and lots of sturgeon were killed. We also lost some other fish species, including straight bass, yellowfin gobies, bat rays, and pretty much anything else that was out there. We collected data on this through a couple different data streams. So we had iNaturalist data, which is a citizen science-based data repository where people can use an app on their cell phone to report fish carcasses. San Francisco Estuary Institute also had a data reporting tool for citizen scientists to report carcasses. The Sturgeon Carcass Mortality Study, which is a group of agency, university, and consulting folks who primarily were collecting data on ship strikes, but pivoted to collecting data on this HAB-induced mortality. And they got reports from citizen scientists, but they also went out and ran their own surveys. And then CDFW shore and boat-based surveys. We had people walking shorelines, and then I was out leading our boat-based survey. So there's some pictures from that. These are somewhat complicated data streams, especially the citizen science, because we had, first of all, multiple reportings. So these are pictures from 2 separate iNaturalist reports. If you look at it closely, you can see that this is actually the same fish. So we had a SciAid go through and manually pull out all of these duplicate photos, which was a lot of effort. It took him several weeks to do that. And he also used GPS points based on their proximity to try and filter that down. But it was a manual process and it took a lot of time. We also had a lot of presence-only data because people were just snapping pictures of carcasses that they were finding. We're not sure which areas were actually surveyed. So like this shoreline here, we have a high density of sturgeon. It's unclear if the other side just didn't have any survey because of access issues, or potentially people just don't walk over there. So this complicates expansion of our counts. Some people scramble their GPS points on iNaturalist, so we end up with carcasses in the Berkeley Hills, which clearly are not real. We reached out to a lot of people to get these unscrambled, but some people did not reply. So this adds to our count, but it makes it hard to detect duplicates because then we don't have the proximity of the GPS. And then finally, we have unknown detection efficiencies. These sturgeon sink pretty quickly. We're not sure how many of them actually make it to the shoreline. So we're not sure what percentage of the mortality we're actually surveying. Like I said, I was running the open water survey. So we started off, we were just running our normal trammel net survey, and we started seeing carcasses showing up in the bay. Here's one that actually floated into our net. We very quickly stopped trammel netting and started just doing a carcass survey. We were trying to hit every carcass we could get when we started, which resulted in some haphazard coverage. And we were pretty quickly overwhelmed by numbers. From each fish, we collected samples, including length. We got sex when possible, got genetic samples, and collected a fin ray for aging, and then also to mark the fish as counted. And after a week, we instituted a random polygon sampling using a design-based method where we gridded the entirety of San Pablo Bay and selected 20 random polygons. And the idea was we were going to go out and we were going to census each polygon and then use that as an expansion to get a number of carcasses throughout the bay. We looked out and the weather was unseasonably beautiful there. It does not look this good on San Pablo Bay much of the fall. Unfortunately, there were no carcasses left floating by September 6th. So we surveyed 20 polygons, we didn't see any, but now we have this method for next time. So from all of these disparate data sources, we ended up with 864 sturgeon total. This is our minimum count. It's almost certainly higher than this. Of those, 195 were confirmed as white sturgeon and 17 confirmed as green sturgeon. See how those shake out in late August and early September, within a week or two. So a lot of mortality within a fairly short period of time. These were also very large fish. So from our sturgeon that were unidentified, most likely white sturgeon, maximum fork length of 238 centimeters, median of 142, white sturgeon, max of 195, median 140, and green sturgeon 166, and median of 107. So at least for the white sturgeon, representing a lot of adults and slot size harvestable fish. These fish were ending up all over the place. So throughout San Pablo Bay, Central San Francisco Bay, South San Francisco Bay, and even out into the Pacific Ocean. The fact that we were picking up fish out in the ocean indicates that there was some toxic effect. It wasn't just because of a decrease in dissolved oxygen. And those were all green sturgeon for the most part. They were picked up out here, and I guess some unidentified ones. This is really concerning given our pattern of decline. Obviously, green sturgeon are listed, so we're concerned about their population. White sturgeon are not doing so hot also. So these are our abundance estimates of legal size, the slot size fish between 1979 and 2021. We estimate around 33,000 fish in the legal slot currently. And of the fish that were killed, a lot of them were slot size or larger. So either fish that are already experiencing high mortality from the fishery, or fish that are outside of that slot and are representing our spawning population. So the implications of this, because we're losing slot size and oversized fish, this is probably a population scale impact from this HAB event, especially considering our probable gross undercount of total carcasses. So we think that there's many, many more fish that were killed than what we actually counted. This adds to an already high mortality rate. So that fishery is still ongoing. We still have all those other sources of mortality that I talked about. This just adds to that. We may have hit a distinct population contingent, which Kirsten's going to talk about this next. But there's a group of fish that are distributed further downstream, and we may have disproportionately killed those fish with this HAB. And we may have lost some of our spawning potential because they spawn, or at least recruit more effectively during wet years. We've got a really wet year right now. We may have fewer big spawning fish to go up and take advantage of that. So in the future, we might see a depressed signal from this wet year cohort. And we're preparing for future events. We don't think this is the last time this is going to happen. Clearly, we're in cycles of big drought like we haven't experienced before. So we're pretty certain that we're going to see big HAB events like this in the future. So we're trying to develop SOPs for open water and shoreline surveys, and also have a better way to use citizen science data, maybe so that it gets collected in a more useful way. So with that, I will take questions.
All right, thank you, Dylan, for delivering some tough news.
Yeah.
Do you have any questions?
Hi, Dylan. Great talk. Have you looked at any of the genetics from the samples you've taken?
So I have not looked at them. We gave them to Andrea Schreier at UC Davis. I think she was going to use them for her sex determination project that she's working on, but I have not looked at any of those personally.
So the the driver of the harmful algal bloom that creates cysts that'll stay in the environment.
Mhm.
Do you know of any other countries where this has occurred or other locations, whether once that standing stock takes off, then it starts happening a lot more frequently? And what does this huge bloom mean from a population standpoint of that algae?
Yeah, so obviously we have cysts distributed throughout all of San Pablo and San Francisco Bay now. I don't know that anyone's looked at recurrence of it, is, I guess, in that specific way. It does happen fairly frequently in places like Japan. So it's, I would anticipate that having a standing stock of cysts will result in more frequent blooms, especially of that species.
Yeah, I'm curious about the future use of citizen science to help you out on this. Do you have any concerns or reflections? You mentioned right at the end that you're preparing to do this again when it happens again. What are the things that are on your mind to make that a little bit more trackable survey?
Yeah, so ideally having some sort of metric of what shoreline was actually surveyed. So we know which areas are zero counts and not just blanks. So that would be good. I'm not sure if we can actually do that in the iNaturalist framework, but that's something we're thinking about. The fact that they take pictures is great. Establishing a project early for people to report to so that we don't have that GPS scrambling, I think would be a good move. Somebody else started the project that we ended up using, and they added us as admins later on. But I think if we start it next time, when we first detect this thing in the bay, then we can have a better handle on it.
I think we can do one more.
Hey, Dylan, thanks so much for that. The lack of smaller juvenile fish, is that because they're less likely to be detected, or they have higher, like low dissolved oxygen tolerance?
So we're not sure about that. Could be that there were fewer juvenile fish in the vicinity of the HAB. Could be that they just don't float. We have no idea if they were impacted by it or not.
All right, thanks, Dylan. Our final speaker of the sturgeon session is Kirsten Sellheim from Kramer Fish Sciences.
Hi, everybody. Thanks for coming. My name is Kirsten, and I'm going to talk to you today about some studies that my collaborators and I have been working on using FinRay geochemistry to reconstruct migration histories in white sturgeon. And I want to say thank you to John and Alicia for ordering our talks in this way, because Dylan provided a really nice background. And I'm also going to talk a little bit about the algal bloom. So I can just get right into the meat of my talk without having to go through that same information. So thank you. So yeah, just acknowledging my collaborators real quick. Malta and Levi and Jamie have been really great to work with. They know a ton about geochemistry, and all the lasering was theirs. And Joe and Jim really provided, that's their brainchild. They provided kind of the impetus for this work to begin with about a decade ago. So I just want to acknowledge them. Tribal land acknowledgement. So all of the sturgeon that were collected for the study were collected in the bay. So in traditional Ohlone and Miwok lands. And then where we stand today is traditional Nisenan lands. So just acknowledging that. All of the funding for this project was Fish and Wildlife Service funding over the past decade. We also had a lot of collaboration with the UCs, CDFW provided some samples, and then NOAA Fisheries as well. So very briefly, sturgeons are in decline worldwide. The reason being is us, humans. And sturgeon are particularly vulnerable because they're really long-lived. They migrate all over the place, so they kind of are exposed to the full range, the spatial range of stressors in the Central Valley in particular. And we really have limited information on their full life history because of these things, because they're really long-lived. They move all over the place. So how can fin rays help? So fin rays are really cool tools. They operate much like tree rings. So you can actually see the rings in this image. So you basically take a pectoral fin ray off of a sturgeon and you can slice it cross section and you can see these age rings in the cross section of the fin ray. You can also measure growth, and then using geochemistry tools, you can reconstruct their movements in space and time, both among watersheds in the Central Valley and then also across salinity gradients. So most everyone has heard of otoliths. They're used a lot for Chinook salmon, reconstructing rearing duration and river of origin. So fin rays are similar in many ways, but they also are distinct. So similarly, they grow throughout the fish's life. We can do the age and growth reconstructions. We can do a lot of different chemical analyses on them, including kind of assessing diet and migration history and all of those things. They do have a different chemical structure. The fin rays and so the otoliths have a really similar morphology across otoliths, whereas fin rays have this kind of complex, sometimes they can have secondary rays as shown in that middle picture on the slide. You can see that little lobe, which makes it a little bit trickier to analyze. And otoliths are used a lot. They're a really well-established tool, whereas fin rays are pretty experimental. Our study was kind of the first of its kind for looking at migration history, so a little bit less known about them. And importantly for green and white sturgeon, fin rays can be collected non-lethally. So otoliths, as you can see from this really gross picture on the left, they're not going to survive that, right? Like when you take the otolith, the fish is dead. Whereas with fin rays, there's been some lab studies that have shown like very little behavioral impacts to cutting the pectoral fin ray from the fish, and no mortality. So there is a strontium isotope mapping, the variation in the 87-86 ratio across the Central Valley. And that's used extensively for Chinook salmon, looking at river of origin. I'm mainly going to talk about salinity gradients though in this talk. So Jim Hobbs and his lab developed this relationship between strontium isotope ratios and salinity. And so it gets a little bit more variable as salinity goes up, but up to about six parts per million, you can really detect pretty clearly where these fish are in the salinity gradient. So up to, kind of Sassoon Bay-ish, we really have a pretty good sense of where the fish was based on the strontium isotope ratios. So outline for my talk, I'm briefly going to go over some of the validation studies that we conducted to really get a good sense of, what the fin rays can do for us in terms of temporal resolution, how early we can start to look at life histories, and then go into kind of the fun part, which is the application. So we have wild white sturgeon that we're looking at reconstructing histories for, and then some of the applications of the results of our study on the algal bloom impacts. So we first conducted this 12-week study that was kind of a proof of concept looking at white sturgeon at Caba. So Dennis Cockrell, big thanks for helping us with the study. It was really complicated. We were using Pewter Creek water that was pumped directly from the creek, as well as American River water, which we trucked in on a weekly basis. And it has a distinct chemistry signature. That's why we used it. But the setup of the tanks was very complicated. So we essentially had treatments where we kept fish in the same water type for 12 weeks. And then we had treatments where we would move them for different periods of time. And so I'm going to show you a lot of these plots. So I'll just orient you real quick. You can kind of think of the x-axis as time. So it's from the core of the fin ray, or in this case, from the start of the experiment to the end of the experiment on the x-axis. And then the strontium ratio is on the y-axis. So in this case, it's the two different water sources with the two distinct strontium signatures. So the one on the top was a fish that was just left in the same water for 12 weeks. And then on the bottom was a fish that was moved. And then we used a predictive model to determine whether we could sort of using the strontium data predict which treatment that fish was in. And so we got really high predictive power from that study. And we also learned a lot about kind of how much temporal resolution we can get from the fin rays. So we found that, about after 22 days of exposure, we're getting this really, distinct signal that reflects the strontium signature of the water. So there is a little delay there, but it's pretty strong. And we also controlled for feed source because we weren't sure how much feed source was going to affect the strontium isotope signature. So we really found that the feed source had really a negligible effect on strontium. So this is Jamie Sweeney's master's thesis. I'm looking at the timing of calcification. So she was using lab fish to look at kind of when fin rays started to calcify, when they started to take on the information that is useful to us for reconstructing their history. And she used a clear and stain method of different ages of fish. So those are the images on the top there. And the one on the far left is showing a 20-day-old fish. There's the red is kind of indicative of there being bone there. And so you can see the 20-day-old fish really didn't have any bony structure. And then as you get to the one on the right, which is 47 days old, you can see that it's really well established. So on average, calcification begins about 20 days post-hatch. And then by 72 days post-hatch, 95% of the fish had kind of full-on calcification. And Jamie also looked at some kind of interesting other elements that we might be interested in exploring a little bit more, such as lead, selenium, rubidium, these things that are associated with kind of environmental exposure or toxins. So this is kind of future studies, but it was interesting to explore sort of how those different elements came into the fin ray over time. All right, so now the fun part, the white sturgeon. So we had subsetted 112 fin rays that we chose for this study to reconstruct the migratory histories of the individual fish. They were collected from 2012 to 2016, and it was a combination of CDFW trawl surveys, Fish and Wildlife study on the San Joaquin River, and then we also visited fishing derbies, sturgeon derbies in the Suisun Bay. So you'll see from this, so the little circles are the number of fish captured per method. So you can see there's kind of a gap there in the South Bay that is frustrating, but we kind of took the fin rays where we could get them. So it's a little bit skewed towards low salinity condition in terms of the sample size. But we had 112 fish of varying ages from 4 to age 30. So these are the plots that came from the strontium isotope reconstruction. So again, time on the x-axis, those vertical dotted lines, or the white lines, are age. So age 1, 2, 3, 4, and so on. And then the vertical is labeled by salinity. So the lower section is pure freshwater, that thin green bar. And then as you get into the darker blues, you're getting into high salinity, which is the darkest blue and kind of bay. You can think of it as the bay, San Pablo, South Bay. And then the very top is the ocean. So this is a fish that spent it really moved directly into this kind of low to medium salinity area, which is kind of Rio Vista, Suisun, Eastern Suisun Bay, for the first really 7 years of its life. And then it kind of moved out to the bay for several years. And then there was some kind of migration back into those medium salinity waters. And these actually were all collected in the San Joaquin River. So kind of interesting. They're making these sort of short migrations that appears into freshwater, which you can see on this plot. So that if you look at the far right, you can see that little freshwater signature is, the fish was captured in the San Joaquin River. So it's sort of been there for just a little bit of time. But yeah, this fish appeared to really go out to high salinity bay right away. We didn't see any evidence of low salinity freshwater residence for this one. It came back after its first year into those low salinity waters and then moved back out again. And then this fish was super interesting. It never really went out into like Suisun Bay, like it really kind of hung out in this low salinity condition for 12 years of its life before we caught it in the San Joaquin River. So a lot going on, right? A lot of different life histories. And this is kind of a mosaic depicting that, right? Like each of the vertical bars are a fish. And, you can see that they're doing all sorts of things. So kind of making sense of this data was a real challenge because there's a really huge variety in what individual fish are doing. One of the things we looked at just to kind of try to parse out the data was splitting it by age. So the bars on the left are age 1 fish all the way up to age 18, which the total number of fish are fewer there because a lot of our fish were 13 or 14 years old. But we didn't see as distinct of a pattern as we thought. We thought we were going to see a lot more kind of low salinity freshwater in those young fish. But really, any age we were looking at, they were doing any number of things. They were really sort of scattered through, all ages of fish were scattered throughout the salinity gradient. So that was pretty interesting, interesting result. And then kind of pivoting over to this harmful algal bloom. So Dylan did a really nice job of kind of setting the stage for this. This red tide happened, and actually all those images kind of gave me a little PTSD. This is really sad situation, but yeah, the red tide really caused this really massive sturgeon die-off. Lots of dead sturgeon on the beaches of the Bay Area, really low DO levels. And so we were trying to use the Finray microchemistry data that we collected to try to better kind of understand what happened and what the impacts might have been. And so, yeah, so we're really talking about that kind of medium high salinity area of the Bay and then coming into Sassoon there. So I aggregated the data. So we had all those kind of sturgeon years of salinity residence in that big mosaic that I showed you. We try to aggregate that data into, sturgeon years. So this is just showing freshwater to high salinity, what proportion of the years were spent in those environments. So, this is a little bit of a coarse way to look at impact, but 13% of the fish in our study were, or 13% of the years, the sturgeon years in our study were fish in that high salinity condition that is like the Bay. 61% of years were spent in medium salinity. So that's sort of, it's Carquinez Strait. So it's like, maybe they were in the Bay, maybe they were sort of in that safety zone where we didn't see as much mortality from that event. But a pretty high proportion were sort of in that danger zone where we were seeing the algal bloom occur. And then we also, with that, this is data from our microchemistry study. We reconstructed the migratory history. We used the migratory reconstructions to do a cluster analysis to try to determine whether there were these particular migration patterns or particular fish that exhibited these kind of types, these migration types. And so the cluster analysis identified four different migratory types. So you can see there's a lot of spaghetti around there. Those gray lines are individual fish. So there's a lot of variation even within these clusters, but these came out as significantly different from one another. We have this first cluster that's kind of a pure freshwater, low salinity. These fish really didn't appear to go into medium or high salinity waters at all. Cluster 2 sort of remains in that low salinity with a slight tendency towards higher salinity habitats. And then cluster 3 seemed to wait a little bit longer before moving into high salinity. Cluster 4 really went straight to the Bay. These are fish that really were in the Bay their entire life through the first 10 years of their life. So juvenile sturgeon. So just kind of coarsely looking at that. So the yellow bars are the area where the harmful algal bloom occurred roughly in salinity space. So we really, from this, we can kind of see that those two, not surprisingly, the two life history strategies that were in higher or medium salinity were really more likely to be affected by this algal bloom. And then the other thing we can do with our microchemistry data that is sort of applicable to this this harmful algal bloom situation is we created a regression on the total length of the fish that we used for the microchemistry study. And we had the ages for those fish from the fin rays, right? So we created this regression, it's pretty tight. And just applying that regression to the lengths of the fish that Dylan showed earlier, it was a lot of big fish. The average age of the fish is estimated as 17 years based on that regression. And it was anywhere from 2 to 34 years of age. So some really old fish. I mean, 2.6 meters, that's a big fish. So it's Yeah, it's a real bummer. And much of this data was collected by CDFW, but also that mortality study that Dylan mentioned. And I've been a part of that for the last 3 or 4 years as it's been occurring with a lot of different resource agencies and universities. And so that was a really kind of interesting use of community science. Like we got a lot of emails about fish that were dying from the algal bloom. So that really allowed us to mobilize pretty quickly and collect a lot of fin rays, a lot of genetic data from fish that we might not have seen otherwise. So that was, yeah, that was kind of how I got segued into the algal bloom, but it was a good way to collect samples pretty quickly and get some information about the algal bloom impacts. So just to summarize the fin ray portion of the presentation. It appears that we can get the temporal resolution from the fin rays of about 3 weeks. So, we might miss sort of short-term migratory movements, rapid migrations, but we can detect dominant habitat use of fish. We might miss the natal origins. We're not really getting calcification until about 3 weeks. So we might miss that very early period with the fin ray microchemistry reconstructions. But yeah, applying this methodology to wild white sturgeon, we really saw this extensive use of all the salinity habitat throughout the bay and freshwater systems of Central California. And a lot of variation, right? The individuals were doing really different things from each other. But we did observe these kind of migratory types that are characterized by how much saltwater versus freshwater time is spent. At least for juvenile sturgeon, we cut it off at 10 years. And then the harmful algal bloom impacts piece. So the higher salinity migratory types were really more likely to have been impacted. Combining the medium and high salinity sturgeon years, up to 74% of the population could have been exposed to that. And just a reminder that we didn't actually sample in the South Bay for the microchemistry study. So there may be this kind of, we didn't sample evenly. We might've been a little biased toward more freshwater fish. And then, the carcasses rolled, the average age was 17 years, up to 34 years of age for those fish. So this method has a lot of potential. There's a lot of next questions. I'm really interested in the contaminant exposure question. I think it would be really interesting to sort of overlay the strontium isoscapes you can get at where and when sturgeon are exposed to different contaminants. I think that's a rich area of exploration for this, as well as kind of overlaying growth with environmental conditions. So is growth higher in lower water years? How do various environmental conditions influence growth and habitat use? We also have a lot of green sturgeon fin rays. So I think looking at those and looking at how their migratory reconstructions differ from white sturgeon. We have a lot of little sturgeon, and I think that would be pretty interesting because that's a piece that we don't have a good handle on. There could be some fin ray resorption in that core part of the fin ray, and we just want to understand that a little bit better, see if we're seeing a little bit more freshwater signature in maybe younger fish. And then lastly, we collected a lot of fin rays from the mortality event. So my crew, the citizen science crew, that project, CDFW, we all mobilized as quickly as we could to try to get as much information as we could during that kind of 2 weeks when things were really crazy. So yeah, just kind of determining whether there were particular migratory types that that died during that event. I kind of set forth that hypothesis that those high salinity variants were probably more likely affected. So testing that with the actual fish that died in the algal bloom would be really good. So yeah, this is just a little more information about that community science project that I'm working on with a bunch of collaborators. And I have stickers that look like that. If anyone wants one, come find me. Yeah, with that, I will say thank you and take any questions.
So thank you, Kirsten. That was a really cool analysis. We run a little late, so if you want to ask questions, please seek her out. We're going to have a break now until 10:10. The next session is session 12, Diversity of Fishes in the San Francisco Estuary. Thanks for joining us this morning. See you soon.
What time was I super late?
The next session, if everybody could take their seats, please. All right, my name's Adam Naniga. I'm the program lead for the Delta Juvenile Fish Monitoring Program run by the U.S. Fish and Wildlife Service. I'm also a co-chair on the Resident Fishes Project work team with Nicole Kwan of DWR, and she couldn't be here, unfortunately. So there's a lot of different definitions of resident fishes. The way it's sort of evolved over time to mean all the fish that we work with that aren't steelhead, or I'm sorry, salmon, sturgeon, and Delta smelt. So we need to give all the other fish some love and attention. And with that, today I've organized this session so that we have two presenters who are presenting mostly on sampling techniques and two that are more individual species oriented. And with that, we'll go ahead and get started with Eric Gruber of the Fish and Wildlife Service.
Hi, thanks, Adam. Hi, good morning. My name is Eric Gruber, and I'm a supervisory fish biologist for the U.S. Fish and Wildlife Services Lodi office. Today, I'm going to talk to you about the Lodi Fish and Wildlife Office's beach seine and boat electrofishing fish monitoring surveys of Delta littoral habitats. Both surveys are part of the Delta Juvenile Fish Monitoring Program, or DJFMP. Before starting, I'd like to thank my collaborator, Dylan Stompe, who is an environmental scientist in CDFW Sport Fish Program for the Bay Delta region. First, some background. About 10 years ago, an IEP scientific advisory group was commissioned to provide a comprehensive programmatic technical review of the DJFMP. The purpose of the review was for the review panel to determine if the DJFMP was 1, meeting its goals and objectives, 2, robust enough to answer future questions, and 3, providing information that could be integrated to facilitate the protection, restoration, and management of naturally produced salmonids and other native species in the Central Valley. In June 2013, the Lodi office published a background report prepared for review by the IEP Scientific Advisory Group. In it, they addressed limitations of the DJFMP's beach seine program. Quote, beach seine sites were originally selected by the DJFMP based on access and to maximize efficiency when sampling juvenile Chinook salmon. As a result, beach seines are only deployed within sites containing unobstructed littoral habitats, including boat ramps, mud banks, and sandy beaches. Because the majority of the shoreline in the Delta and Lower Sacramento and San Joaquin rivers is dominated by alternative habitats like riprap and aquatic vegetation, beach seine catch data may not accurately reflect the abundance or distribution of all non-salmonid populations. Therefore, it may be advantageous for the DJFMP to reinitiate boat electrofishing within the Delta to supplement beach seine monitoring to better assess the status and trends of fishes occupying littoral habitats within the Delta, end quote. In the past, the CDFW Delta Resident Shoreline Fish Monitoring Project conducted boat electrofishing surveys intermittently from the 1980s to early 2000s. Also, UC Davis conducted boat electrofishing surveys from 2008 to 2010. So we asked this question today. How well do the DJFMP electrofishing surveys supplement beach seine surveys to comprehensively monitor fishes occupying Delta littoral habitats? Today, I will be presenting beach seine and e-fish data and comparing 1, relative and standardized catch differences between the two surveys, 2, survey selectivity differences for Delta species, and 3, size differences. First, some background about the beach seine monitoring program. It has been in operation since 1976. Weekly beach seining occurs during daylight hours at fixed sites throughout the Lower Sacramento and San Joaquin rivers, Delta, and San Francisco Bay. A 15.2 meter by 1.3 meter beach seine net with a 3 millimeter Delta square mesh, a 1.2 meter central bag, and float and lead lines attached to 1.8 meter tall poles is used. In general, the Seine is deployed along the shoreline by two crew members within unobstructed habitats. Obstacles are defined as structures that could compromise safety or gear efficiency, including steep banks or holes, fast water current, submerged aquatic vegetation, or large woody debris. All fishes captured are identified to the species level and enumerated. All or a subset of fishes are measured for length. The electrofishing program has operated since November 2018. A generalized random tessellation stratified spatially balanced sampling design is used. This is a method of sampling that reduces biases associated with sampling at fixed sites. It involves the division of a larger geographic area into smaller subregions. Once divided, each region is randomly sampled using a probability sampling method. Sampling is conducted during daylight hours within approximately 160 to 240 meter long by 10 meter wide transects along accessible shoreline habitats. Transects are divided into 6 independent sampling segments to allow for spatially dependent occupancy and detection probability estimation. The sampling is conducted within 6 main delta regions: the Cache Slough Complex, North Delta, Central Delta, South Central Delta, South Delta, and West Delta. Each region is subdivided into 3 subregions, yielding 18 subregions in total. Subregions are sampled monthly on a year-round basis, except the West Delta, which is only sampled during high outflows when salinities are low enough for electrofishing. In addition, up to 3 regions are selected on a rotating basis for a second round of sampling to improve estimates of spatial variability and occupancy models, resulting in up to 24 transects completed each month. Here's a picture of the DJFMP's electrofishing boat named Zeus. It is an 18-foot Smith Root heavy-duty series equipped with an apex control unit providing up to 80 peak amps of DC power. E-fishing, it says the computer's about to restart. Oh.
It won't. It won't let you smooth.
Okay.
There. Is that okay? At least you can see it.
Okay, thank you. Okay, excuse me. E-fishing uses direct current electricity flowing between electrodes, one of which is an anode. When the proper amount of current is applied to the water, fish are drawn toward the anode through involuntary muscle movements. Upon reaching the anode, fish are stunned. Generally, salinities greater than 8 parts per thousand are too saline for e-fishing with our system because the ions in the water diminish the electric field. 2 experienced netters and 1 boat operator comprise the field crew. Here's information about the monthly sampling schedules from November 2018 to July 2022 for the beach seine and e-fishing programs. A green cell indicates that sampling occurred that month, and a white cell indicates that no sampling occurred. Normally, beach seine sampling occurs year-round. However, sampling was disrupted in April and May 2020 due to the pandemic. Originally, e-fishing did not occur from June to September. Also, e-fish sampling was disrupted from April 2020 to June 2021 because of the pandemic. Multiple years of comparative data for both programs are available from November to March in water years 2019, 2020, and 2022 in the North and South Delta regions. I will present data today in 2 different ways, either all available data from November 2018 to July 2022 for all regions, or from November to March in water years 2019, 2020, and 2022 in the North and South Delta regions only. A total of 66 fixed seine sites and 1,971 randomly selected e-fish locations were sampled from November 2018 to July 2022 for all regions. Note that e-fish sampling is more complete within the Delta, but that beach seine sampling is conducted in areas where e-fishing cannot occur, such as the saltier San Francisco Bay and areas of the Sacramento and San Joaquin River main stems that are sometimes too shallow for a boat. A subset of 15 fixed seine sites and 652 randomly selected e-fish locations were sampled from November to March in the North and South Delta regions during the 3 water years. Now on to the results for relative and standardized catch differences, including regional differences. Here are relative catch data for all sites sampled from November 2018 to July 2022. A total of 77 species were captured by seine surveys. Strong species dominance is apparent. Mississippi silverside, Sacramento sucker, and red shiner comprise 75.7% of the total catch. Mississippi silverside, shown at the top, comprised over half of the total Seine catch. Only Sacramento sucker is native. A total of 39 species were captured by e-fish surveys. Strong species dominance is also apparent, but the species differ. Bluegill, largemouth bass, and red ear sunfish comprise 77.5% of the total catch. All are non-native and in the Centrarchidae family. Bluegill, shown at the bottom, comprised over 30% of the total e-fish catch. Note that species richness differences are largely due to the fact that 28 species were uniquely captured in the San Francisco Bay Seine sites. Again, e-fishing generally does not occur in salinities greater than 8 parts per thousand, which precludes e-fish sampling in the bay. Here are standardized catch data from November to March for the North and South Delta during the 3 water years. Same data on the left are standardized according to water volume sampled. E-fish data on the right are standardized according to the amount of time sampled. As expected, Mississippi silverside dominated the Seine catch in both regions. However, abundance in terms of CPUE was approximately 5 times greater in the South Delta. Chinook salmon, western mosquito fish, bluefin killifish, and bluegill represent the 5 next most abundant Seine sampled species in the North Delta. Red shiner, threadfin shad, bluegill, western mosquito fish, and golden shiner represent the 5 next most abundant Seine sampled species in the South Delta. As expected, centrarchids dominate the e-fish catch in both regions. Red ear sunfish, smallmouth bass, largemouth bass, and bluegill represent the 4 most abundant e-fish sample species in the North Delta. Bluegill, red ear sunfish, largemouth bass, and warmouth represent 4 out of the top 6 e-fish sample species in the South Delta. Non-centrarchid fishes abundant in the North Delta, according to e-fish data, include Mississippi silverside and golden shiner, whereas those abundant in the South Delta include common carp and golden shiner. Next, I will describe the results of survey selectivity. Here is a rather busy table, so I will take a little time to walk you through it. Shown here are species survey ranking data for 36 Delta species. Habitat associations are also indicated for each species. Data appearing in the far left column are species survey ranking data for the DJFMP e-fish program. That is followed by ranking data for species captured by the 14 long-term surveys plus the State Water Project South Delta salvage operation investigated in Stompe et al.'s 2020 San Francisco Estuary and Watershed Sciences paper. The formula used to calculate species survey ranking is presented in Stompe et al.'s paper. A value of 1, color-coded in dark green, corresponds to the species that has been caught in the highest cumulative numbers and frequency for a given survey. A value of 0, color-coded in white, corresponds to any species that was not caught over the life of a given survey. Data from November to March for water years 2019, 2020, and 2022 for the North and South Delta were used to calculate species survey rankings for DJFMP's e-fish program. All other data are from Stompe et al.'s 2020 paper. If you squint your eyes, you can see that the e-fishing program appears to sample fishes in higher cumulative numbers and frequencies than most other surveys. This may be due, at least in part, to the randomized sampling design and the ability to sample in unobstructed habitats. When species survey rankings are averaged according to survey type and habitat associations, we see that e-fishing appears to be far better suited for sampling submerged aquatic vegetation associated fishes than beach seines, midwater or Kodiak trawls, and otter trawls. We also see it as most suited for sampling fringe-associated fishes. Not surprisingly, midwater or Kodiak trawls are best suited for sampling pelagic fishes, and otter trawls are best suited for sampling benthic fishes. These findings are similar to the findings using past CDFW and UC Davis e-fish data. In their 2021 paper, McKenzie and Maharja note, quote, "We used fish catch data from past boat electrofishing studies, a long-term beach seine survey, and a couple of long-running trawl surveys to compare the relative probability of detecting various fishes across these sampling gears. Overall, we found that boat electrofishing led to notable improvements in the detection rates for many native and non-native fishes." Now on to the results for fish size differences for beach seine and e-fish sampled fishes. Here's a bean plot showing length distributions for all species captured by beach seine on the left and e-fishing on the right from November to March for the North and South Delta during the 3 water years. Fork lengths are measured for fishes with forked or heart-shaped caudal fins, and total lengths are measured for fishes with square or rounded caudal fins. As you can see, the median length for e-fish sampled fishes is over twice as large as seine sampled fishes. Also, unlike beach seines, e-fishing catches fishes in much larger size classes. Indeed, 22.8% of all e-fish captured fishes were larger than 150 millimeters, whereas only 0.4% of all fishes captured by beach seine were larger than that length. Here are cumulative length frequency distributions for Chinook salmon, a species of concern, and one of its predators, largemouth bass, captured by seine and e-fishing from November to March in the North and South Delta regions during the 3 water years. Similar to the results presented for all species combined, e-fishing samples larger salmon and bass overall. It is worth noting that adult salmon are encountered during e-fishing surveys, but are not netted and measured for length due to sampling permit restrictions. Note that largemouth bass become piscivorous at approximately 60 to 140 millimeters. Also note that since 2000, Central Valley hatcheries stopped releasing fry-sized fall-run Chinook salmon, so all fall-run fish less than 55 millimeter fork length are natural origin fish. All races of Chinook salmon are included in the cumulative length frequency distribution plot presented here, but 95% of salmon captured by seine and 88% of salmon captured by e-fishing are fall-run fish according to length of bait criteria. Before concluding, it is worth noting here that DJFMP's e-fishing program samples in less studied Delta regions. On the left is a figure copied from Stompe 2020 SKUs paper. It is a heat map of sampling intensity according to number of stations across the 14 surveys mentioned earlier. The color scale ranges from faint blue for a low number of stations and dark red for a high number of stations. So on the right is a similar heat map for all 1,971 e-fish sampling locations sampled from November 2018 to July 2022. As you can see, DJFMP's e-fish survey samples in less studied interior Delta regions, especially the Cache Slough complex to the north and the South Central and South Delta. So the main take-home point is that DJFMP's electrofishing survey complements seine surveys and represents an important component of the Littoral Delta habitat monitoring program. E-fishing helps complete the IEP monitoring puzzle by sampling undersampled fishes, like centrarchids, and less studied habitats such as those dominated by submerged aquatic vegetation. Sampling is also conducted throughout the Cache Slough complex in South Central and South Delta regions, which to date have received less attention than other regions. Stratified random sampling e-fish design also helps reduce biases associated with fixed station sampling. On the other hand, DJFMP's beach seine monitoring program provides a long-term data set that is essential to detect changes in fish status and trends over decadal timescales. The survey is particularly suited for sampling salmonids, especially wild origin fish. Also, beach seine sampling is low-tech, so it is relatively inexpensive and not prone to technical difficulties that could otherwise lead to sampling gaps. Furthermore, unlike e-fishing, seine sampling is not limited by salinity or depth, which increases the overall sampling area for the littoral monitoring program. A major goal of the DJFMP's e-fish monitoring program is to develop a statistically robust approach to determine population sizes rather than just standardized catches and abundance indices. Next steps include developing occupancy models for correlated e-fish surveys with autologistic regression and following that up with an N-mixture approach that fits the e-fish sampling scheme. This work is currently underway and led by Noble Hendricks with support from Russell Perry, Adam Pope, Brian Mathias, Brian Maharja, and Ryan McKenzie. Before wrapping up, I would like to thank the U.S. Bureau of Reclamation and California Department of Water Resources, which fund the DJFMP program. I'd also like to thank the IP for their 2013 review of DJFMP's Littoral Habitat Monitoring Program and for recommending that electrofishing sampling be reinitiated. I'd also like to thank IEP for organizing this meeting and allowing me to speak to you today. Lastly, I'd like to thank past and present Lodi Fish and Wildlife Office staff, including Jeff McLean, Lori Smith, Ryan McKenzie, and Brian Maharja, who developed the electrofishing program. I'd also like to thank Adam Naniga and Jacob Stagg, who lead the DJFMP in our e-fish program. Last but not least, I'd especially like to thank all the field technicians and small craft operators who arduously continued to gather field data throughout all months of the year. So I don't think there's time for questions. So thank you for your attention. And I'd be happy to answer questions on the lobby. Thank you, Eric.
All right, thank you, Eric. Our next presentation is from Emily Hubbard of DWR. Okay, sorry, you have to deal with this thing here.
He's snoozing.
It won't let us for some reason. Sorry.
Okay, hi, I'm Emily Hubbard, and this presentation is about hitch in the YOLO bypass and trends with the YOLO bypass fish monitoring program that I looked into as a scientific aid for DWR's aquatic ecology unit. Okay, so first I'm going to start with some background on the YOLO bypass fish monitoring program, or YBFMP. And then a little bit background on hitch, which are not a species we often talk about, and is our species of interest for this presentation. I'm going to go over some interesting fish catch trends and related water quality trends, and then I'll finish up with some main takeaways from this data. Okay, so the YOLO bypass fish monitoring program has been operated by California Department of Water Resources since 1998. The bypass, shaded in yellow, on the map, is the largest remnant floodplain for the Sacramento River and was structurally implemented at the beginning of the 1900s. The bypass floods or inundates when the Sacramento River stage height exceeds the 32-foot weir at the north end of the bypass. And during the rest of the time when flooding is not occurring, the bypass is home to private duck clubs, livestock grazing, YOLO bypass fish wildlife preserve, or YOLO bypass wildlife preserve, and local agriculture. So these factors, plus the fact that the southern end of the bypass connects with the Sacramento-San Joaquin Delta, means that water is present in the tow drain, which I outlined in red here, all the time, regardless of whether it is a flood or a drought year. YBFMP monitors fish in the tow drain using 3 different gear types. The first 2 circled in red here are a fyke trap, the top one, and a rotary skew trap, the bottom one. The fyke trap is deployed from October through June and is designed to catch incoming adult migratory fish or adult resident fish in the system. The rotary skew trap, on the other hand, is deployed from January through June and is designed to catch outgoing juvenile fish or small resident fish. Lastly, YBFMP has a current set of 8 standard beach seine sites that kind of extend along the full length of the tow drain, circled that in orange here. And there's also a few additional beach seine sites that are sampled when flooding occurs, and so extends, the full length of that yellow shaded area. The beach seine sites are used to monitor resident fish and are conducted every other week for the entire year. All 3 of these trapping methods can and have caught hitch, the species that I'm talking about today. Hitch, or Lavinia exlicata, are part of the Cyprinidae family, and they're native to the Sacramento, San Joaquin, Clear Lake, Russian River, and Pajaro Salinas drainages. They're scattered in abundance throughout and are typically found in slow-moving stretches of river, clear, low-gradient streams, warm, low-elevation lakes or sloughs, but also clear, cool, sandy bottom streams, such as areas in Poudre Creek. Juveniles are commonly found where aquatic or emergent vegetation occurs, and adults can be found in more open waters, but similarly with areas of coverage. They're not aggressive swimmers and are easily stopped from upstream movements by small dams or other structures. Interestingly, they're known to have the highest temperature tolerances among the native fishes of the Central Valley. There are 3 subspecies, but obviously today I'm just reporting on Central Valley or Sacramento hitch. Hitch have a small upturned mouth and are omnivorous open water feeders. They can feed on anything from filamentous algae to aquatic insects and terrestrial insects. We have quite a few different species of minnows that we catch in the YOLO bypass, and at small sizes, hitch can be easily confused with other minnows. Here is This is taken directly from U.S. Fish and Wildlife's ID guide for juvenile cyprinids, just to show you how similar they can all look. But everyone doing fish monitoring for the Aquatic Ecology Unit is trained on fish ID. And so just to show here for hitch, the long anal fin, higher anal fin ray count separates it from most of the other cyprinids, and then it's slightly longer dorsal fin ray count separates it from golden shiner subsequently. At larger sizes, notable features in addition to the fin ray counts are their moderately large scales, the small head with an upward pointing mouth, the lack of a sharp keel on the belly, and their narrow caudal peduncle. Okay, so let's get to the reason why I am talking about hitch today. YBFMP noticed a large increase in hitch counts in the last couple of years, and we were wondering, is this significant? Is there any trends that we could attribute to this? Why is this happening? And this plot shows count data, so just straight counts, separated out between our beach seine sites and our pike trapping method. Historically, while we do catch them, we don't consistently catch that many, and it's very, very low numbers. Between 2000 and 2022, we've collected a total of 551 hitch across all of our different gear types, which to put that in perspective, we've caught like that many of silver sides in a week from beach seining. So ultimately, this is low numbers, but we're talking about this because 71% of that total count happened in the last 4 years. That's pretty significant, in my opinion, and is worth looking into. 68% of all the hitch we've ever caught were from beach seining methods, while 23% being from the pike trap. I'm not going to be showing any data from our rotary screw trap because we've only caught less than 10% of all hitch count from that method. Okay. So this next plot shows that same data, but represented in catch per unit effort, seen on the y-axis. The effort data for beach seining is from seine volume, and the effort data for the pike trap are based on hours of operation. The x-axis shows water years from water year 2000 to water year 2022, which will be same for every plot that I show. The top plot is for beach seining, and the bottom is for the pike trap. Historically, we've had spikes in, albeit small spikes, in catch of hitch primarily or entirely in the months of June or July. But the last few years, however, we're catching hitch in large quantities in October, November, December, in addition to those catches in June and July. So why? Why are we catching more hitch all of a sudden? Wish that I was a statistic savant, but I'm not, still learning. And while I looked at a lot of different variables, I didn't find any significant ones that attributed to a model regarding why we're catching more hitch more recently. To be fair, this is a pretty newly emerging trend, and we still have overall pretty low values as far as CPUE and catch. So, and we've caught them before flooding events, after flooding events, and warmer temperatures and colder temperatures. Is kind of a mix of variables there. So nothing too clear. So instead, I want to show you guys just some different trends and things that are happening simultaneously to this higher hitch count that I thought were interesting, both with fish catch and with water quality. So first, I'm going to start off with some species trends. I initially was looking to see if any other fish species had a similar increasing catch in the last couple of years or decreasing catch in the last couple of years. This plot shows 4 different species, including hitch, which is on the top, that we catch in the Yola bypass. All of these species can be typically found with hitch. And they're not, floodplain spawners like Sacramento splittail, which are notably linked with wet years or flooding years. The graphs on the left are from beach seining, and the graphs on the right are from the pike trap. And the colors match up with the 4 different species of fish. The y-axis is again in catch per unit effort. And I want to mention that they're not the same scale for every single plot. The point here is not to compare between different fish CPUEs. The point is to look at, kind of a long-term trend comparison. Okay, so to start, on the bottom, we've got golden shiner. Golden shiner are a minnow which is non-native to the Central Valley, and they can be found primarily in warm, shallow ponds, lakes, and sloughs, and are similarly associated with beds of aquatic vegetation. Looking at the beach Seine data, it appears that the golden shiner were more prevalent pre-2010 or are back somewhat consistently since 2015. No obvious spike similar to what we were seeing with hitch is present with the golden shiner. The trend doesn't match hitch, but you can see with the pike trap that we are all of a sudden catching them when we weren't previously, although it's a little bit more longer scale since like 2014, but we did have a big spike in 2020. Next up, we have the Sacramento sucker. They're part of the Catastomidae family and are native to the Central Valley and can be found typically with other native minnows, usually more like Sacramento pike minnow, but with minnows in general. The bigger difference obviously being that they have a different diet than Sacramento hitch because they're feeding along the bottom. But they can be similarly found with, in slow moving waters among emergent vegetation as juveniles. In the beach Seine, we seem to catch, somewhat consistent amounts, including that 2-year large spike. But, no like large spike happening in the last couple of years. If we're looking at the pike trap, there appears to be like a 7 to 9 year cycle of catch increase to decrease, which honestly is pretty interesting in and of itself. Kind of wish I could look into that, but they're not our species of interest today. But the point I want to show here is that they're not seeing any increase or decrease effect on this trend in the last couple of years. It's staying pretty consistent over time. Okay, and then lastly, we have Sacramento blackfish, a native minnow found commonly alongside hitch and have even been known to hybridize with hitch. They're very similar appearance, but have much smaller scales. Historically, we've found them somewhat consistently for catch in both beach Seine and pike trap. But what I want to draw your attention to is the last couple of years where Sacramento blackfish catch in both deer types has completely dropped to 0, which coincides with this increase in hitch count. So odd, don't know what's going on there, but definitely something worth looking into, especially since we pretty consistently caught them in the past. Okay, so this next comparison, this on the left is the same plot that we were just looking at with hitch and Sacramento blackfish, just on the same plot. I'm just gonna show what's going on there. And then I wanna just tie it in to some other fish that we've got in the yellow bypass. So historically, we catch a lot of mosquito fish. They're one of our main fish that we catch in the beach Seine. And recently, in the last couple of years, we noticed killifish in 2017-ish, slowly. And in the last 2 years, 2021, 2022, we had a huge boom in killifish, coinciding with a drop off in the amount of mosquito fish that we're catching. Not exact same trend that we saw with hitch and Sacramento blackfish, but I'm bringing it up to show that we could be having some species shifts happening in the yellow bypass. And so both with, our native fish and with our non-native fish. Okay, so I'm gonna stop talking about fish for a second and move on to water quality. Let's see, so whenever we do fish collection out in the yellow bypass, we also take water quality measurements using a handheld YSI. These are all the parameters that we look at. And I looked at all of these to see if there were any trends happening in the last couple of years, changes in our conditions that maybe could be related to our increase in hitch count. I didn't see anything significant, but I did want to talk about turbidity and secchi depth, which are kind of inversely related. And they have some interesting trends that I thought would be fun for everyone to see. Okay, so long-term trends with discrete turbidity. This is from 2012 to 2022, because that's when we started taking turbidity measurements. And you can kind of see that we've had a pretty slow, gradual decrease in turbidity, both in our beach seine sites and at our fyke trap site. And it's gotten less variable too over time. Okay, and then as one would expect, discrete Secchi depths have been the opposite. So it's slowly increasing Secchi depth over time, both at our beach seine sites and at our fyke trap sites. Then this doesn't show, we've had kind of an increasing occurrence of times at beach seines where we couldn't even collect Secchi depth because it's so clear you see the bottom. And that's something we're gonna look into, but I don't have that information here on this plot, but definitely something we've noticed more recently. I wanted to show these because anecdotally, we're also getting just way, way more vegetation in our system. I obviously don't have any qualitative data for you or quantitative data for you. We over the last 10 years have noticed an increase in a duration of time that we have vegetation. And so we've started collecting essentially presence absence of vegetative data. Obviously, everybody's heard the vegetation talks that we had yesterday. And it's a problem in the Delta overall, and definitely a problem that we're experiencing in the Yolo bypass. And so anecdotally, this could be related to why we're having lower and lower turbidity over time as well, because we're having prolonged times where we have more and more vegetation growing as well. Okay, so I threw a bunch of trends at you. Why does this matter? Why do we care about hitch? We've definitely had a lot of talks over the last couple of days about how global warming is changing our delta. We're having some pretty widespread effects that have been affecting the delta and are going to continue to affect the delta. Things like changes in temperature and increasing vegetation. And we know that there's rapid decline of California native inland fishes. And that's something that we want to pay attention to. And so I've noted that Yolo bypass might be experiencing some fish species shifts, and hitch could be equipped to be dealing with these changing conditions that we're having in the bypass. They could even have found an area that they can do well in. And so, you know, years of drought, increasing vegetation, decreasing turbidity, these are things that hitch seem to do okay with. And they could be hanging out in the bypass, and it's always good to note some positive effects that could be happening from all these changes. I'd really love to look into food availability in our bypass and see if any changes in zooplankton or drift invertebrate quantities are maybe related to why we have an increase in hitch the last couple of years, but not something that I've yet looked at, but it would be interesting. I'd also love to know what is going on with hitch in the rest of the delta. And I don't know if anybody else went to the poster sessions, but Claudia from US Fish and Wildlife has done some of that. So very interesting, definitely look into that. And then I'm really, really curious what happens after this year's flooding event. We had a lot of overtopping in January and currently, and I'd like to know if they leave. So Joaquin, I want to know how many hitch we have in June. So yeah, and I'd like to thank all of Aquatic Ecology, my BFMP field staff, and IAP. And I think we are out of time. So if you have any questions, I'd love to hear them later, I guess. Yeah.
Thank you. Up next is Scott Blankenship with Kramer.
Morning, everybody. Thanks for attending this session. Thanks for your attention. I'm going to be talking about fish community data from a restoration site using DNA metabarcoding. This is a diverse crowd, so I've tried to keep this pretty high level. So nobody should be challenged from a technical standpoint today. The community data was produced by Katie Karpenko and Cheryl Dean from our group at Kramer Fish Sciences. And advance. There we go. Starting with some acknowledgements. While I'm going to be talking about just some community data from one particular site in the Delta, this project is a fairly extensive project with a lot of folks working on it, including there's a sort of a sister project that this is incorporated with. Because of that, there is not anybody at Kramer that doesn't have their hands in this from an analytics side or a fieldwork side. For funding, the data I'm going to be showing today was funded by California Department of Water Resources, the Multi-Benefits Group. As I understand, the Multi-Benefits Group is particularly focused on flood related things, but the projects that they do have aquatic species benefits, hence the multi-benefit name. But I have a theme here about the importance of place, just to give a higher level context for some of the community stuff. For those that do or are familiar with genetics work, location or place tends to explain the primary aspects of genetic variance. To illustrate that, this is a photo of Pacific Northwest and beyond. Right down the middle of that is a ridge, the Cascade Crest. Shown here are about 12,000 rainbow trout. The primary axis, that x-axis there, is coastal subspecies of rainbow trout and inland subspecies of rainbow trout. And that gap down the middle is the Cascade Crest. So it's explaining most of the genetic variance by where these fish are located. And the purple group on the top is from fish further north. Also, a species being present at a location tends to correlate with abundance. That might sound obvious. There was the N mixture model talked about in an earlier talk. But if you grid out an area, you're more likely to see, you're more likely to detect fish in an area or target species in an area. They're more likely to be abundant there. So you can set up your sampling schemes in an informed way to get at more aspects of what's going on with that species, be it habitat or abundance or whatever. Also, place is important for biodiversity. So the more species that are present at a spot, the more biodiverse that spot is, right? There's lots of biodiversity is a thing. Agencies are concerned about this. NGOs, science groups are concerned about this. And the surveying which species are where is important. This actual chart is from a metabarcoding approach. So the fish community information sort of under the hood is information like this. There is a need for molecular tools. There's a couple of drivers for that. As I see it, one is the rarity of native species. Another one is the rate of change that habitats are going through. On the rarity side as well, it's difficult to get access and permits to study protected species. So that's sort of some friction there. And the rate of change where you just have to have more observations than often there is funding for. So you need some adaptations and efficiencies for the gear that you can use there. Sticking with rare species, sticking with a rare species example, an obvious rare species example is Delta smelt. Everybody's aware that the population has declined over time. They're difficult to sample. It's challenging for any gear to pick them up. Reasons for that. One obvious reason is habitat. So there's lots of information on this, but essentially the habitats that native species have evolved around don't exist anymore. So you're right. A lot of emergent marsh, native species reared and lived in doesn't exist anymore. The habitat itself is fairly inhospitable now, and species are declining as a result of that. Hence, the effort to rehabilitate and establish new habitat. It's challenging because those species are rare to get access to sampling those habitats, and they're difficult to detect in those habitats, and the habitats themselves are difficult to deploy a lot of different gear. You know, in mud, marshy areas, it's difficult to drag a net through there. So having some different options is useful. This is just an example of some, not metabarcoding, but it is environmental DNA data. So this is a heat map of quantity of DNA detected relative between two species. Not super important, but it's just a way to show this. My primary point is I've been thinking about this as a network analysis of late, where you might have a lot of information over time about which species are present, but you may not know what's driving those correlations in why those species are there or why those species are not there. But you can over time get some inference about that. So a real world example of that might be, you know, if you're searching for a coffee shop around here, you'll get a histogram of why that shop is busy and why it isn't busy. Google may not know, and they probably do, but they may not know why that place is busy at 2 o'clock in the afternoon, but you can get some insights just from observational and frequency data. A little more specifically about what we're doing, monitor some of these habitat. I'm going to be talking about the Dutch Slough restoration site. There's a lot going on out there. I'm going to be talking about a very small bit of it. And there's some other projects that are associated with this from the multi-benefit group that kind of overlap, but I won't be talking about those today. This is a top-down shot of a small bit of the Dutch Slough restoration site. Right down in there in the middle is one of the crafts we're using to sample this. That's not a small boat, so this is a very big property. I think it's I don't know this the acreage, but it's a fairly big property, fairly expensive to set up. It's been a decade or more in the planning. So what are some of the things we're looking at as the group that's going in there surveying fish communities? Primarily because this was not exposed to the bay and then had levees breached. Primarily, the groups out there are looking for how this habitat is going to evolve over time. We're looking at fish. There's other folks looking at carbon sequestration, geochemistry. There's a lot of species-specific groups that are out there as well, terrestrial and aquatic. We want to know what species are present, fish diversity, native fish use, those sorts of things. And this habitat's really difficult to survey, right? It's tidal, it's reticulated, it's difficult to get some sort of traditional gears in there like nets. A brief pit stop into regulations. You could have an entire symposium, obviously, on regulations. Maybe there should be, but just in the highest level, barest sense, how fish presence may interact with regulations in this sense. For the multi-benefits groups, they do things like repair levees, set them back, create beneficial habitat off of that. The criteria by which the performance of those projects is judged is that there's no net loss of habitat. That's super vague. I don't really know what that means, and they're often not monitored. But if a species is present, at least you have an indication that it is habitat and the habitat hasn't been lost. So it does tick a box there for areas that aren't typically monitored that need some sort of performance metric attached to them. Another is habitat restoration. So just in general, in North America, about 10% of habitat restoration projects have performance measures attached to them. Of those, fish response often isn't in there. They're more physical factors. So having an indication of whether the target species is there at least gives you some indication about the habitat's value. It's not super quantitative about species response, but it's something. And again, permitting, it's difficult to set up sampling designs that are optimum for a lot of protected species. You often can only go out there when you're allowed to go out there. At least with some of the molecular tools they're benign, they don't need permitting, so you can set up your surveys to be optimal around those of when those species might be there, which is handy. On to some observations. This is just to cue me in that this to give a real quick primer on metabarcoding. So there's different ways to analyze for DNA in samples. This particular fish community work is a metabarcoding approach. So there's ways to target species. This doesn't target species. This targets fragments that are generalized for taxonomic groups. You go in there and sequence all those fragments that you get out of your samples, and then you have a big list of what's present. So you can sample a lot of different material, water, soil, what have you, extract the organics out of that, target whatever taxonomic groups you're going for. In this case, we're using fish-specific primers. They do pick up reptiles and amphibians, not ideal for reptiles and amphibians or birds. They do pick up mammals fairly well. There's different ways to dial those in for different taxonomic groups. We're targeting fish, so that's our focus. The information I'm showing you today, it's pretty straightforward. You're looking at what species are present at a point. It could be 2 points relative to each other. It could be different points over time, that sort of thing. But it's a composition of what organisms are there. Nothing too serious going to be presented. Dutch slough, top-down view on Dutch slough. Some high-level information about how the survey itself is done. The targets are where the environmental samples are taken. In 2021, there were 2 surveys per month, May to October, 1 survey per month, November and December. That switched in 2022 to be 2 surveys per month. This year, we've since gone back to do 1 survey per month during the winter. I'll tell you why that is. We were picking up native species in the winter, which typically aren't sampled because of permit restrictions. So we altered the survey a little bit to not impact continuity, but we wanted to get some information about that time window to inform future sampling designs. Species counts on the y-axis, date on the x-axis. The different colors are the different parcels. So there's partial to the west, partial to the east, and then there's a deep kind of lake area in the middle called the burrow. The dashed lines down there are when the levees for those parcels were breached so that the property was exposed to bay influence. On the left is native fish, on the right is non-native fish. For native fish, before the levee was breached, there was, on the order of 1 or 2 native species present on the properties, kind of in general. After the breach, that sort of bumped up to on the order of 5 or 6 native species. So native fish species use increased after the property was exposed to the bay. On the non-native fish side, kind of depended, but before the breach happened, there was a bit of a range there, but you're in the, what is that, 5 to 10 species range for non-native fish present on site before the breach. After that, it bumped up, running in the range of 15 to 20 species or so. Obviously, this is a compendium of all the species here. We know what species these are. We can go in and look at each one of these if we wanted to. So a little bit more high resolution information digging down into particular species. This is looking at native fish. You can look at native fish, you can look at non-native fish, whatever. This is looking at native fish at a particular place. Before the breach, there was sculpin present. After the breach, there was sculpin present. There's always sculpin present. They seem to not care. Native fish showed up in 2021, Chinook and Thule perch. The control site's not, I wouldn't necessarily call it a control site for DNA. It's a control site for the video surveys we're doing because it's not the same place. Not a real rigorous control for DNA because there's some flow through potentials, but there is the nearest control site shown just for reference. A year post breach, a few more species there. There's hitch showed up. Chinook are seen there. Pike minnow are seen there. All right, so you can look at native fish presence. You can look at rare species detection. So this one's a little tricky depending on which side of the coin you're looking from. If a species isn't in a habitat all that often, then maybe that habitat's not important for that species. That's an interpretation. If a species are rare or you really want to know where these species are, you do see these species in and around these places, just not very often. So some of the rare species that we saw on site, green sturgeon, longfin smelt, and Sacramento blackfish, didn't detect them very much, but we did detect them. You can get seasonal patterns. Here's some just 5 examples. There's a lot more you can pull out for Chinook. You see them in the wintertime. The green highlighted areas were when the breaches happened for the properties. But you do see them, but there's a seasonal trend and when you see them and when you would expect to see them there. We were picking them up recently there. Hitch. Not present on the Gilbert property for whatever reason, the first year, but showed up sort of commonly second year. Something's going on there. Split tail in the control, sort of outside the area that first year. The second year, it's there part of the time. We haven't looked into any, there's plenty of environmental data and habitat data that's going along with this. We haven't dug into any of that yet. Sculpin are there all the time. Three spine spickleback weren't there at all that first year. We're there the second year. Who knows what's going on with that first year. Thule perch in the control, in the vicinity. Thule perch are pretty common native fish around. They moved into the property that second year. So to close, place is of fundamental importance in biology for a variety of different reasons. DNA metabar coding is an efficient means to determine fish biodiversity. Observe species-specific trends should inform future hypothesis testing, Dutch Slough Living Laboratory. Thank you. Maybe one question? Yeah, one question. Yeah. Oh, wait for the mic, sorry. Halt.
Oh, okay. Thank you very much for the information. Regarding detection of species, the detections can identify any DNA of a certain species within a water column. So detection of the presence means that the DNA is there.
Yeah.
But how do you know that the species hasn't been dead for like a week or so and is actively using that actual area?
Yeah, if there's carcasses or dead fish there liberating the DNA, you're going to detect it. DNA in the water column tends to not stick around that long if the organism is absent. But if the organism's there, you're going to keep detecting it. So that's a frequency of observation or a weight of observation kind of thing, because you detect that. If birds are flying around and they're dropping carcasses into that lake area, yeah, you're probably going to detect stuff. Yeah, exactly.
Great, thanks.
That's all the time we have.
Thank you.
Questions. Thank you, Scott. Our final presentation of this session is from Matthew Young of USGS.
See, cool. Hi, good morning, still morning. Thank you for coming. Thank you for coming to this meeting. Thank you, Adam, for organizing. I'm gonna zoom out a lot. So that it was great to see Scott's like zoomed out picture of the Pacific Northwest, because that's where we're going. So I'm gonna be talking about variability in coastal habitat used by longfin smelt, or the non-resident distribution of this anadromous fish. So just sort of like a little bit of a segue here in today's session. I don't need to introduce this species to most of you, I imagine. Longfin smelt are one of our native osmerids. They live up to a couple years. They have a pretty fascinating life history. And they're sort of obligate anadromous. So this is the life cycle figure lifted from Joe Merz's paper, but they'll go ahead and be born in fresh water, head eventually out to estuaries in the ocean, and then come back to spawn. And most germane to this talk is their range. So this is like a very fascinating species in that they can be found from South Central California all the way up to Southwest Alaska. So pretty fantastically diverse, capable. They're really interesting species. We think about this species mostly within the realm of the San Francisco Bay, right? That's what we talk about, that's what we think about, but that's just a tiny fraction of their overall range, and it's at the southern extent of their distribution. There's tons of work. I don't need to tell you about it. You know it, most, like you guys have done it, but there's a lot of work showing longfin smelt trends, status, ecology here. There's very little understanding of longfin smelt once you leave the confines of the San Francisco Bay, let alone out in the Pacific Ocean. So the purpose of this talk is to sort of fill in one of those missing gaps in our understanding of their life history, specifically what's going on in the ocean. There was a paper that came out in, I believe it was 2017 by Rebecca Garwood that synthesized a lot of marine observations of longfin smelt off the California coast. But what we want to do is see what we can learn about their distribution range-wide and their overall habitat use. So we're going to talk about three things today. The Pacific distribution of this species, so just an update to our understanding of their overall range. And then zoom back into California and get a sense for what coastal habitat for longfin smelt looks like, and then how that has varied over time. This has been a data compilation exercise. There was no new data. This is the perfect COVID project. Starting in April 2020, I started spamming every fish biologist I could find, literally between Los Angeles and Anchorage. So it was like, I had to keep a spreadsheet just of emails I sent to people because I forgot who people were. Data came from a whole bunch of different sources. I'm not going to list them all, but you can see some of the logos here, just a whole bunch of different places. And this is what our compiled data set sort of generally looks like. So again, this data represents every point in this data is an individual observation. Were longfin smelt present at this place at this time? We had roughly 900 unique observations of 50,000 or more individuals, and split up across the entire Northwest Pacific. Diving into how we sort of looked at this, one of the limitations of these data are that it is presence only. So all we know is that a longfin smelt was present. We don't know if it wasn't there because sometimes you don't look. That's just what we do. So the best sort of way we've realized to handle this is using a maximum entropy approach. That statistical analysis handles these presence only data sets pretty well. We can talk about, if you want to learn, we want to talk about that, we can talk about that. But I'm not really going to now. In looking at the Pacific distribution, we looked at summer only. For some reason, people in Alaska have better things to do in January than look for osmerids. It's cold and dark and they're not doing it. We identified 4 continuous covariates that we thought were important in predicting the distribution of these fish. There were 2 that we sort of thought of as geographic. One is just bathymetry, so depth in the ocean, and the other is distance from an estuary. We know that this fish is an obligate estuary user, and small, so presumably that's a predictor. And then we looked at broad oceanographic variables, including sea surface temperature and sea surface chlorophyll concentrations obtained from satellite data. Based on the maximum entropy models, so we were able to get sort of continuous curves looking at the relative importance of these different parameters in predicting fish distribution or fish presence. We'll start just orienting you. This x-axis is the parameter of interest. So in the top left is left? Yeah, left is distance from an estuary in kilometers. Top right is bathymetry. You can see that these fish generally are close to estuaries and shallow. There's some covariation there. We can talk about that, whatever. We also see pretty clear threshold effects for temperature. This is partially expected given the seasonal constraints of our model, but generally speaking, we saw fish with a clear preference, if you will. That's a loaded term, but generally speaking, roughly between 10 and 15 degrees C is their sweet spot. And there was some influence of sea surface productivity, as measured by chlorophyll, but that was not necessarily the most useful indicator. I like pretty maps, so we're gonna talk about pretty maps for a second. I wanna orient you for a minute. So these model outputs aren't in probability of occurrence. It's not like there's an 80% probability this fish was there. Rather, it's in a probability ratio. So if you have a probability ratio of 1, that means longfin smelt are just as likely to be here as they are anywhere else. Like a coin flip. Are longfin smelt present? I don't know. If the higher you get, the more likely they are to be present. And we don't have a percentage on that number. I kind of prefer this metric because it doesn't give us a false sense of security, right? This is a pretty qualitative answer here that we're getting. This is what this output looks like for a few select locations within the Pacific Ocean or along the sea. So Cook Inlet in the top, the Skeena River and Haida Gwaii there in the middle, and then the California coast. The light gray represents our model domain. So we actually modeled whether longfin smelt are present in that light gray area. And anywhere where you see colors ranging from blue to the warmer reds, that's probability ratio of 1 or greater. So probably good longfin smelt habitat. We did this for multiple seasons. And the way we approached this for assessing just updating their range is anywhere within this model footprint that ever had a probability ratio greater than 1, we called plausible longfin smelt habitat. And then we add all that together, and that's what this red, that's what these red indicators are on this map. Prior to this analysis, this inset was our best understanding of longfin smelt range. This is the sort of automatic species distribution model spat out by Fishbase. Many of you might be familiar with that. But it's pretty coarse, and this sort of refined our understanding pretty considerably. Much more coastal, and actually potentially further south than expected. The Pacific Ocean is huge, it's variable, it's different. Not all parts are equal, and we already know that these different subbasins within the Pacific Ocean are associated with longfin smelt population distribution. So other work has documented that the sort of Alaska current population is different than the California current. So it gave us some handy justification to focus on what's going on for the distribution of longfin smelt within the California current. Same analysis, same covariates. This is just sort of a table indicating when these covariates were important predictors of longfin smelt habitat suitability. All I want to do here is point out the consistent importance of depth and sea surface temperature. They are a shallow water fish that doesn't like it very warm. Not super surprising, but we can use this to help constrain our understanding of what habitat of the ocean might be. Again, this is just sort of those same species distribution maps plotted out just for the California current. You'll notice that there are some pretty consistent hotspots, if you will, located with on the San Francisco, just outside of the San Francisco Bay, Humboldt Bay, and Puget Sound. No surprises there either. But what I want to do is start talking about how variability in climate, variability in conditions has influenced the availability of suitable habitat for longfin smelt in the ocean. So if you've been paying attention, it's been crazy out there for the last couple of years, right? Like things have been bonkers. And that's sort of outside the realm of our understanding. We don't have a good sense of what that means for habitat for these fishes. So here I just want to give a representative sample of what that's looked like. So these species distribution models that we built are sort of based on a mean temperature from a 30-year time period, from 1982 to 2012. But in recent years, we've seen years that are much colder than average. 2008 is one example, and years that are much, much warmer than average, at least off the California and Oregon coasts. So that's 2015 here. So we're going to talk about what the distribution of longfin smelt off the California coast. We'll start with just south of Cape Blanco, so California and Southern Oregon. Based on these average baseline conditions, 1982 to 2012, roughly 14% of our model domain is suitable, right? Again, we defined suitability earlier as just habitat that is plausibly utilized by longfin smelt. In a cold year, 2008, we see moderate range expansion or habitat expansion, 17%. Two parts of this. One, it expands a little bit. Two, it's kind of hard to see, but these colors are much warmer so that there's more habitat and it's better habitat in cold years. And then in warm water years, we see substantial contractions in what is plausibly suitable habitat for longfin smelt in the ocean. So from say 14 to 17% of the model domain down to 5 to 6% of the model domain. This is all relative to that baseline. So here you see that South California current plot. Is the mouse showing? No, anyway, bottom half of this. You can see that 2008 period where there's a moderate expansion in longfin smelt habitat, that 2015 period where it contracts considerably. And what we did is actually generate predicted habitat for the entirety of that 20-year window from 2002 up to 2021. And you can see it comes in waves. There are periods where habitat suitability for longfin smelt expands, periods when it contracts, particularly 2014 and 2015 when we had this warm water blob show up that played havoc with all sorts of things. This is what we see at the southern extent of their range, Cape Blanco South. If you look at sort of the entirety of the California current from Vancouver Island to the Mexican border, you can see that we still see some variability, but it's muted. This isn't necessarily a surprise. When you look at the southern, we would expect a species constrained by temperature to see the strongest impacts at the southern extent of the range, which would be us. I'll finish a little bit with sort of what our next steps are and how we're going to be applying this information. But in summary, we have dramatically refined our understanding of what longfin smelt habitat in the Pacific might be and constrained that considerably. It's likely that there's a lot fewer longfin smelt along the Aleutians than previously thought. And there's the potential for more marine habitat use further south than previously thought. There's a lot of variability in habitat and availability of habitat just within the California current, and we have a decent understanding of what that is and how that changes seasonally. And then in addition to these seasonal changes, we've started figuring out how these interannual periods can affect habitat suitability for longfin smelt off of the California coast. The big question is like, what does this matter, right? Like the whole point of this was to fill in a missing piece of that life cycle diagram, right? And so what we're wanting to do now is work in this information we have for predicted longfin smelt habitat suitability in the ocean to help me explain some of the existing variability in our understanding of San Francisco estuary populations. We know that outflow matters. We know that all sorts of other pieces matter, but we also know that there's variance there that isn't explained by other pieces. And we're looking to explore how that, how this marine habitat information could fill in some of those gaps. Another missing piece here is that I mentioned that this is a presence-only data set. We only know when longfin smelt are present. We don't know when they're absent. And this is because most marine surveys, they catch a smelt and they call it osmerid. And so there's a lot of information that could be gleaned by building out our understanding of species identifications in a lot of these marine surveys. This isn't just true for longfin smelt. There's a lot of missing information for many of the other species, surf smelt, night smelt, particularly Eulachon. There's just a whole bunch of information that could be gathered just by sort of addressing these difficult field identification questions. This work was funded by the state water contractors. I think I neglected at the beginning to thank my co-authors. So Fred Fire at USGS and Steve Lindley and Dave Huff with NOAA. We've been working together on this for several years, so thank you to them. There a whole lot of people deserve thanks for helping pull data together. This is, I'm sure I forgot somebody, I'm sorry, but it's been a huge project. Many thanks to all. The database of these marine observations is actually publicly available. If you can't get this link in time, I will gladly send it to you. It is a work in progress. I've already received additional observations from the last few years that I'm looking to add to it. And then there's a manuscript associated with this that is currently in review. And I'd love to talk about that if you're interested too. I'm done. I'll go away.
We do have time for a few questions, if anybody has questions.
Bruce, oh, no. You're the, you do it, yeah.
Awesome talk. I'm glad that you're going to look at the influence of ocean conditions on abundance indices of adults. I'm wondering if you're also planning on looking at maybe conditions that would affect recruitment and trying to see if that is influencing, if that needs to modify your model. So if there's a year with less recruitment, you would expect to see fewer smelt overall, and therefore your detection probability might be lower in some of the places that would otherwise be good. And then also seeing if there's a connection there in terms of the population and yeah.
Yeah, it's a really good question. I think the resolution of our species distribution model is not going to allow necessarily for building the life cycle component into it in that way. But that's something Levi Lewis and I have talked a lot about, is finding ways to relate marine conditions to possible recruitment success, and then also just condition of fish when they return, and sort of how that might play into it as well.
Hi. So you got fewest responses from your emails from Oregon, and my feeling from talking to people is that that would be based on the fact that they don't catch them up there, rather than they're nasty people.
There were no nasty people.
Do you have any reason? So, but your habitat, you show habitat off the Oregon coast, at least as much as off Northern California. Do you have any thoughts, insights? Are there fish we're missing, or are they not there for some reason, or do those nice people just never go out and look?
Yeah, that's a really good question. So Bruce was asking about potential gaps off the coast of Oregon. And, you know, there was seemingly no reason why there should be a gap. So there's the NOAA's surveys for juvenile rockfish and juvenile salmonids encompasses the northern coast. They didn't catch any. Many of the commercial bycatch and observer data that I worked with, one of those observations did come from Oregon, but there weren't that many. And then in talking with folks, they weren't catching very many Eulachon either relative to Washington coast and California coast. And I have no idea why, whether it's the sampling bias thing or whether there's some undefined element of habitat that we don't quite understand. Speaking of gaps in our study, we also didn't see very many longfin smelt in the basically from the Fraser River to the Skeena River. So that area of the British Columbia coast dominated by glacial archipelago and fjords. And that's another aspect of this is, to what extent does this coastal habitat, the shape of the actual shape of the habitat and that sort of things actually play into things? I don't know, but I think there's a lot of room to learn more. I don't know, that's not a satisfying answer.
No, it's okay. It's been 20 years how much they connected to us. And nobody steps up from Oregon. Oh, well, we take them all the time.
Well, I should add that there was a recent paper, which many of you might have seen, by Colin Brennan and colleagues looking at estuary observations of larval longfin smelt from here to the Oregon border. It stopped at the Oregon border. And folks who've been doing larval fish work in Oregon did not have data, did not see longfin. Some of that might be an ID thing, osmerids versus individual species. I don't know. No idea.
Unfortunately, we got to move on. Thank you, Matt.
What happened to the, oh, there we go.
Oh, look at you.
Introduce our session.
Yeah. I think, look at that. The computer's about to restart, but yeah, look at that. Just don't touch, don't touch, don't do it. I don't.
Can you move that down? So can you move that down?
Okay.
Earlier? Somebody brought that up.
Is that okay? How's that? The software center. Hey.
Sneak on.
No,
here.
will not.
Yes, I am. Okay, you guys are up first.
Is there no break? No.
Yeah.
Erin Ferguson is up first.
Oh,
She'll be presenting trends in Central Valley Omicus fishery at
all. I don't want to hear about that.
You already heard it.
I know.
What's that? Oh, John had some questions.
Oh.
That's nice stuff.
Don't ask me. It's
should I let people like move a little, or
I think everyone thought we had a break. It's okay. Let's do it while there's no people in here.
we don't have a break, and we're going to cut it.
all right, all right.
So
Is it okay? All right,
yeah.
all right. Hi, everyone. We're going to get started with the next session, session 13, which is the Steelhead session. My name is Robin Bilski. I am one of the PWT co-chairs and co-chairs for this session, along with Mike Beeks. And I think we're just going to get rolling with our first speaker, who's from California Department of Fish and Wildlife, Erin Ferguson. She works in Fisheries branch as a senior ES, and I'm just going to hand the mic to her. Or not hand.
Thanks, Robin. Can you hear me okay? All right. Morning, my name is Erin Ferguson. Like Robin said, I work in Fisheries branch. I work on the Central Valley Angler Survey, as well as serve as the Central Valley Steelhead Coordinator. So data that I present today was collected through our Central Valley Angler Survey project from past years' versions, as well as the current iteration. Sorry, it wants to restart. All right. So in my presentation today, I'll be giving a brief background on Central Valley Omicus before moving on to discuss aspects of the fishery, including protective measures, how we monitor it, and then discuss trends that we see in fisheries metric collected by our survey. So steelhead and rainbow trout are the same species, Oncorhynchus micus, but they vary in their life history strategies. So rainbow trout are the resident life form. They spend the majority or their entire life in fresh water, where steelhead are the anadromous form, where they are born in freshwater, migrate to the ocean where they feed and grow and return to freshwater to spawn. In the Central Valley, we primarily have winter steelhead, meaning that the adults return to freshwater in the fall and winter and then spawn within months of returning. Juvenile steelhead as well as rainbow trout will rear in freshwater year-round before steelhead migrate back out to the ocean, typically in late winter through spring. So in California's Central Valley, historical Omicus habitats span from the upper tributaries of the Sacramento River in the north to the Kern River Basin in the south. And that distribution is notated by the black lines in this map. Dam construction has restricted current Omicus populations to approximately 20% of their historical spawning habitat. And so Omicus in the Central Valley DPS, which is the brown shading here on this figure, are federally listed as threatened under the Endangered Species Act with federal protections given to all Omicus in anadromous waters, including hatchery origin and natural origin fish, with the exception of steelhead produced at Nimbus Fish Hatchery. And that's due to historical out of basin egg transfers from North Coast populations. So the mission of the Department of Fish and Wildlife, in short, is to manage California's natural resources for their ecological values and their use and enjoyment by the public. And so this does mean that when it's possible, we allow angling opportunities on fish species, including Omicus. So, oh no. All right. So additionally, subsection 6 of the Fish and Game Commission's anadromous rainbow trout policy provides guidance to the department on how we should manage the fishery. And so this section of the policy states that angling opportunities shall be encouraged when angling will not harm present or future generations of the population. And additionally, there may be protective regulations that are required to provide a fishery. And so in order to provide a fishery for Omicus in anadromous waters, we do have protective regulations that are in place. In the central, or in California, we have a Marc selective fishery for Omicus. And so that means that only hatchery origin steelhead may be taken in the fishery, so harvested. So those fish are identified by a missing adipose fin clip, as in this photo. And unlike Chinook salmon, 100% of hatchery origin steelhead are missing their adipose fin. Additionally, some rivers have seasonal closures or gear restrictions such as barbless hooks. We also have bag limits to manage harvest. And these regulations can vary by water body and even within water body. And that's to provide, additional protections to all the life stages of Omicus that may be present. Also, anglers are required to report their effort in harvest targeting of Omicus. So the department primarily monitors the sport fishery in two ways. One is through our report card program, and the second one is through our angler surveys. So California anglers targeting steelhead in anadromous and coastal waters are required to report their effort and catch targeting Omicus by location. And then for the purposes of fishing regulations and reporting, we have a size delineation at 16 inches total length, which distinguishes between a steelhead, which is 16 and larger, and a rainbow trout, which is less than 16. But regardless of the size of the Omicus, the adipose fin must be missing in order to be harvested. Data that we collect from the report card are not used to estimate population. They're just a management tool. And then revenue generated from card sales, fund projects that can provide angler benefits through restoration, monitoring, and education. All right, so the second monitoring tool we have are angler surveys. Our project is called the Central Valley Angler Survey. This project has been operating at various temporal scales since 1991, with the current iteration of the project starting back up again in 2006. The objective of our project is to collect the data that we need in order to estimate angling effort, harvest, catch, CPUE of the major anadromous and resident sport fishes in the rivers that we monitor. Additionally, we collect biological data on the catch. And so for Omicus, this includes length, weight, sex, origin as determined by that adipose fin clip. We collect scale samples and occasionally we'll collect otoliths. So using expanded data collected through our survey, we can look at trends in angling metrics such as effort, catch, CPUE, harvest, and see how that varies spatially and over time. We also can, using the scales and otoliths, we can look at life history, gain life history information from them, such as anadromy, age structure. So for these last three, we collect this information. I wasn't able to gather all of that in time for today, but it is something that we can look at using data that we collect. All right, so this is a general overview of the Omicus in the sea bass. That's a abbreviation for our project. Monitored streams between 1991 to 2016. The Omicus fishery is consistently the third most popular fishery in the Central Valley, and that's measured by angling effort. And so that puts them behind Chinook salmon, which is number 2, and striped bass, which is number 1. Anglers spend an average of 268,000 hours targeting Omicus every year. And this fishery has a relatively good catch per unit effort at 0.8 fish per hour. However, retention rate is very low in this fishery. It's on average less than 10%. So continuing on with general trends overview, angling effort, which is on the left, has not significantly changed over our survey period. Although the slope of the regression is slightly declining. Catch, however, has significantly increased over time. And so when I say catch, that's the number of fish harvested and released added together. And so just some observations on why this may happen is that anglers are getting really good at catching fish. You know, over time, methods have improved. Information sharing through social media has really changed the game in all of the fisheries that we monitor. But we don't collect that kind of information. This is just what we think is happening. All right, so this is a heat map of catch per unit effort. And so you'll see that this also varies spatially and over time. And so we have the rivers that we monitor on the left-hand side progressing from downstream to upstream and then month across the top. And then the various shades of blue represent CPUE, where the lightest shades are low CPUE and black is the highest. And so I don't think that if you're familiar with the fisheries here or even with the systems and their populations, this map doesn't really surprise you at all. Rivers that are really known to have steelhead, there's a high degree of anadromy, such as the American River and even the Feather River, sees highest CPUE when the adults are in the system, so late winter through the spring, where other rivers like the Upper Sacramento River that is known for a trout fishery sees the highest CPUE through the spring and then the summer. And then other rivers that have kind of a mix of both just sees a really broad distribution of CPUE. So now I just wanted to share some individual river trends for some of those metrics that we measure. And so the effort, catch, and retention are based on simple linear regressions. A circle with a line through it means that metric is not changing. An arrow pointing up is increasing, arrow down decreasing. Significant metrics have the stars next to them. And then I also included average retention rate and CPUE just for more information. So just to highlight a few trends, like I said in the past slide, effort is not significantly changing anywhere that we are monitoring, although you will see that catch is increasing in the upper Sacramento, Feather, and Yuba rivers. And in some of those, they're significantly increasing. And I think that it's increasing in these particular areas because of the popularity of the trout fisheries there. More people are going there. You'll also see that retention rate really varies widely across the landscape, with highest retention rates in the American and Feather rivers. And then CPUE is just all over the place as well. All right, and so these pie charts represent the size composition of the catch. Catch again, just meaning the number of fish contacted, and then the size differentiation here is based on that 16-inch delineation. So red are steelhead, and then blue are rainbow trout. I left origin out of this because some of our sampling took place before the implementation of the clipping program in 1997. I just wanted to have as long a period of record as I could for this analysis. And so, like I was talking about before with the composition of what the fishery is made up of, you know, when you have primarily steelhead or high degree of anadromy, you see larger fish in the catch. I will note that the American River does have the influence of the genetics from the North Coast populations, which have larger fish in general. And so those fish are always bigger than our Central Valley strain that we see in the other rivers. But the upper Sacramento, Yuba, and McCollum are known for having trout. And so the fish that we see there in general are smaller. Sorry, my slides don't always advance. All right. So in summary, the trend analysis presented today included angler-derived data from the Sacramento, American, Yuba, McCollum, and Feather rivers for years 1991 through 2016. Angler effort targeting Omicus has not significantly changed over time. However, anglers are catching more steelhead and trout, but harvest is not increasing. It's actually decreasing. And so just in my experience talking to anglers, some reasons why we see a high degree of released fish in this fishery, you know, there's a lot of reasons. So one is the size of the fish, they're yearlings, they're smolts, they're small. They're a natural origin fish that can't be taken. You know, somebody doesn't have a report card. It's a bycatch species. They were fishing for something else and this is what they caught. But the main reason is that anglers are practicing catch and release fishing in this fishery. It's a really huge component of this. I think that it happens because anglers are pretty well educated. They know that this species is not going to die necessarily after it spawns. It's really hard biting. It's beautiful, you know, and it's just an overall really fun experience that they want to relive again. So they release these fish. All right, so the Central Valley Angler Survey has much more data that we're wanting to explore, including our large collection of scale samples. We'd also like to look at the trends in contact with natural origin versus hatchery origin, Omicus that I couldn't get to today. We also think it'd be interesting to validate report card data where possible, where there might be location overlap, and see if trends that they're observing matches what our survey sees. Additionally, we have long-term goals for this project. We'd like to do analyses like this for all of the sport fish that we monitor. And then when possible, we'd like to contribute our data to other projects that are out there if you find it useful. So with that, I want to give a big thank you to the Central Valley Angler Survey team, which includes both permanent staff and a whole village of scientific aides. And of course, a big thanks to anglers and to SFRA for keeping this project going. And with that, I'd be happy to take questions. Perfect.
All right, if there aren't any questions, they can keep going here. All right, up next we have Brad Cavallo with Crane for Sciences.
Thanks, Mike. So this presentation is going to talk about some results from a study that began in 2021, kind of as a pilot year, it's kind of a partial year. And then in 2022, we've completed like a full year of the study. So a lot of the results you're going to hear about are from 2022. I realized we had to turn these slides in really early by my, standard of usually doing slides at the last minute. And I realized that I did not include a map of the area. So I need to orient you a little bit. The Stanislaus River, everybody knows the Tuolumne, I think, because everyone knows Yosemite, right? So the Tuolumne is just to the south of the Stanislaus. And the portion of the Stanislaus we're talking about is below Goodwin Dam. That's the upstream barrier to migratory fish, and down about 20 miles-ish to about Oakdale. That's where there's spawning and rearing habitat for juvenile salmonids. So that's where we've been doing our work. And thankfully, Erin already covered a lot of this, so I'm going to try and move through it quickly. ESA listed fish. There's this issue of Omicus and the fact that we have both anadromous and resident fish that are contributing to the population. We don't have a lot of information about abundance or about how those two populations are mingling. And in general, they're already an ESA listed fish, but we need more information so that we can, assess their viability and hopefully take management actions that will contribute to their recovery. So Chinook salmon and steelhead are quite different. Chinook salmon come back from the ocean always, and they spawn and then they die. And when you see juveniles from Chinook, you can pretty safely assume they're from the fish that spawned recently, or if you can make an inference about which kind of cohort or bird year they're coming from based on their size. But not the case for steelhead. Again, we have anadromous fish coming back from the ocean. We have fish that are living in the river. They both spawn. They don't always spawn. The juveniles they produce may or may not emigrate. And they don't emigrate in a predictable schedule the way Chinook do. So you could have, age 2 fish deciding to go out as smolts. So quite different. Little conceptual model of the life cycle. The green arrows are sort of major transitions, like in life stages or aging, the fish getting bigger. And the dashed or the kind of black dotted lines are sort of behavioral life history transitions that are really important for us to understand the species and be able to manage them more effectively. And really, in a nutshell, the purpose of this investigation we're doing on the Stanislaus is to try to estimate the frequency or the probabilities of these transitions occurring, at least where those transitions are relevant and where we can get data to inform that. So what are some of the more specific questions that we have? Well, we want to know how many there are, how many adults. Certainly, we want to know when they're maturing and spawning. We want to know how many anadromous fish there are and how they're contributing. And when they're spawning in the river, we want to know how successful they are. For juveniles, for the migrants, we want to know at what size and age they're heading out of the system. We want to know how many there are. And really importantly, we want to know under the conditions under which that's happening. What time of year, what kind of flows, what kind of weather, all that kind of stuff. So on the Stanislaus, we've deployed a bunch of different methods to collect data that all kind of gets tied together. And I'll say a little bit more about how we're tying it together. I'm not going to go into a lot of details on sampling design and all that kind of stuff. But so for adults and juveniles, we're collecting fish by a variety of methods. We are using electrofishing. Anglers have been a big part of it. Professionals and volunteers, seining. And there is a weir at the downstream end of the Stanislaus River that's operated by Fish Bio. And Michael's going to be presenting on that after my talk. And that's definitely contributing to this study. And if you few important ways. So on those fish, whenever we collect adults or juveniles, we're collecting scales, we're putting pit tags in any fish that's larger than 65 millimeters, and we're collecting tissue samples for genetics. And then we're aging those fish, those scales that we sample, and so we can estimate growth. We have conducted egg incubation survival experiments in the river to see how well eggs can survive in artificially constructed redds in an incubation chamber like you see here. And then for the juvenile outmigrant component, obviously we're pit tagging all the fish that we can upstream. So we have a dual array at the exit, the downstream end. And there's also a juvenile monitoring, like a capture station, one that's operated by us and one that's operated by U.S. Fish and Wildlife Service, I think, at Caswell. So one of the things that really ties all the data together that we're collecting is close kin mark recapture. And I don't know if everybody here is familiar with it, but I think most biologists recognize that tagging of animals is an incredibly powerful tool that we like to use. It allows you to do marker capture and estimate things like abundance and survival and document migratory behavior. And frankly, we don't do as much of it as we'd like to because of the physical difficulty of getting tags into fish and the fact that the tags don't always last. The really cool thing is that all fish have a tag. They're born with it, a genetic tag. And this genetic tag cannot be shed. It doesn't have a battery that conks out. And most importantly, the tag is heritable. So when a fish spawns, it passes its tag on to its progeny. So the genetic tagging concept is the basis of the CKMR that we're using for a lot of the analyses you're going to see here. In this figure, you could spend a lot of time looking at this. I'll just kind of hit the highlights. The big fish at the bottom are the adults. The smaller fish at the top are the juveniles. Where you see dark colored fish, those are fish that you sampled. And where you see dark lines is where you detected a parent offspring pair. But there's a lot of other connections that are reflected by the dashed lines that you may not have seen in your sampling event. Other sorts of sibling relationships that provide additional information that can also potentially be incorporated. So by collecting this data, you're getting information that sort of crosses generations that allows you to do marker capture. So genetic tags, I mean, we've known that every animal has a genetic tag for a long time, right? And this concept has been used in a hatchery context where it was called parental-based tagging, mainly for salmonids, as far as I know. But the application of this concept to wild populations is a little bit newer. And I've thrown up a few different papers here that I found really helpful. One of them is about Arctic grayling. One's about bluefin tuna, and one's about brook trout. And they, provide some really good examples of how this works in kind of testing assumptions and all that kind of good stuff. I think this methodology is really promising and probably has applications to other species that we've been hearing about here at the conference. Math. I'm not going to talk about math a lot. If you're familiar with mark recapture, you may recognize this equation at the top as a kind of a Lincoln-Peterson estimate, a very simple methodology for estimating abundance. And it's really the same thing. We have a sample of juveniles that we have, and we have a sample of adults. That's what's in the numerator there. And then in the denominator is the parent offspring pairs. That's like your recovery rate, the rate at which you recover the tags that you put out. So you can estimate the population with that simple data. Of course, in reality, when you have an age-structured population, larger fish have different fecundity, they have different propensities to spawn in different years. So you need, in some circumstances, and we do need this ultimately for the Stanislaus Omicus study, you need a multinomial model so you can incorporate all those kinds of different transition probabilities because they're not the same across sex and age. Okay, results. Should just add that, so these are the results primarily from 2022. We're continuing the study through 2025, kind of adding a few things, improving some things we think we need to improve on. So first result to share is on egg to fry survival. This is based on 3 different sites in the Stanislaus River. There's 4 reds, 4 artificially constructed reds with the little egg chambers put in there. And the average survival is about 55%, but as you can see, it was very highly variable within sites and even between the replicates. We think that there was fine sediment was a factor that was influencing this, and there was you can see in the graph here, there was a flow pulse that occurred during this period, and we think that may have moved some fine sediments around and led to some deposition that affected egg survival. That's something we're taking a closer look at in this year. Age and growth. We've detected 6 age classes of fish. The young of the year, when we do our sampling in the spring, are small enough, they're easy to distinguish between age 1 fish and older. So that's a good thing. The young of the year are growing very fast. And we're not seeing a lot of differences in growth rates among, or size at age among the males and females. We did see 7 fish that exhibited growth that appeared to be consistent with anadromy. And all of those were fish that were age 3 or older. Abundance. So in 2022, we collected tissue samples from 315 young of the year, age 0 fish, and 158 samples from age 1 and older fish. And from that, we detected 82 parent offspring pairs. And that leads to a spawner abundance of just shy of 1,000 individuals. So this includes residents and anadromous fish. One of the key assumptions for this is that the samples that we collected are representative of the population. And I think in large part they are, but there are definitely a few areas that we were not able to effectively sample. So one of the things that we're doing this year is we're using a raft-based electrofishing to more effectively get at some of those spots. I should add too that this abundance estimate is based on the simpler version. We have not yet plugged in the sex and age specific probabilities into the estimation, because we're just now getting enough data to be able to do that. Okay, the little ones are spawning. Age 1 fish, males and females, are spawning, and this has been confirmed by both genetics and by checks on scales. Among all age 3 fish that were sampled, about 10% were anadromous. But as we're seeing with as much spawning activity of the age 1 fish, which are quite abundant, the anadromous fish are kind of a small proportion of the reproduction that's going on in the Stanislaus River. At least that's what we've seen in 2022. Imagine it might be very different from year to year. We're seeing heteroparity, fish spawning more than once. 13% of the fish of age 2 or older had evidence of a previous spawning event, including 1 anadromous individual. So another thing you can do with the genetic data is just analyzing the juveniles that you sampled, is you can make an estimate of how many adults contributed to the juveniles that you observed. And when we did that, you come up with 261 adults for production in 2022. That's probably a minimum estimate just because of the more juveniles you sample, that number will start to trickle up, but there were at least that many that contributed in 2022. In this graph that you see here, the blue bars are success. That's the number of adults that produced juveniles successfully, and the green bars are adults that didn't produce offspring that we detected. And what you see is that the highest success, both in terms of percentage and numbers, is among the younger, the age 1 fish, which is pretty surprising. We did have an age 5 male who was assigned to 10 juveniles, which is pretty amazing. There are a lot of hatchery fish in the system, and Michael will talk about that from the weir. We only got a sample from one of them last year. It was a female, and she did spawn successfully, and we detected 5 of her offspring. Juvenile migration. So we have in total from 2021 and 2022, 265 fish that have been pit tagged. The graph that you see here, the green, so basically length frequency. The green is the sizes of the fish that we've pit tagged, and the blue bars are the fish that have been observed over 20 years at Caswell, outmigration trap. So basically, we're tagging the right sizes of fish, we think, and we're tagging quite a few of them. We have not seen any of these tag fish fully immigrating from the Stanislaus to date. There was one fish that kind of hit our first upstream antenna, but didn't go all the way through. But I'm happy to report that in this year, just this year so far, we have captured 2 Omicus, one in our trap and one at the Caswell trap. So that's encouraging. That's definitely something we didn't see last year, and we don't know about the pit tag data yet. Dropping my notes. So just a little bit more about CKMR and how it helps us. This is just kind of a plot of so 2021 and 2022 are the real data that we collected, kind of stratified by age. And then for 2023 and beyond, this is kind of like, what we hope to sample or what we might sample. What I really want to convey is that by this methodology that links generations, ties things together in a way, so that, an age 1 fish that we may encounter this year may be from a new spawning pair that we did not sample last year. So our spawning abundance estimates and other metrics change over time as more and more information comes in with this approach, which is super useful. So kind of wrapping up here, we were able to estimate the abundance of the spawning population in the Stanislaus River. There are definitely anadromous fish present. So far seem like a small component in terms of their juvenile production. The fish are spawning, the Omicus are spawning in age 1, which is really surprising. Hatchery Omicus are present and they're spawning successfully. And we are so far not seeing a lot of juvenile outmigrants. And hopefully that's something that will change this year with all this crazy weather we're having. So I want to thank Reclamation for funding this study, and some of our other partners. The San Joaquin National Wildlife Refuge has been kind enough to host our trapping and pit tag arrays. Calfish and Wildlife has been really helpful doing red surveys, providing scales and tissues from steelhead or Omicus they encounter. Some of our professional anglers, Devin Olson, Fish Bio for operating the weir, and of course the many biologists and staff at Kramer Fish Sciences that have worked really hard to pull this study together. Thank you so much.
Thanks, Brad. We have 4 minutes for questions, Robin.
Yeah, we do have one. How do you determine the spawner living at?
By scales.
Any other questions for Brad?
So if they're reproducing by age 1, technically this year you could start seeing 3rd generation. So I was wondering, does this method, how many generations can you track using this technique?
Well, we can track as many as there are, of age classes. And so far the oldest fish we think we've seen is age 5. And I'm not sure what the upper end of that is, but as long as the fish are living that long and the study continues, we will continue to get more and more generations that are included in the sample.
Yeah, great presentation. It's good to see all this data put together this way. What I'm curious about the assignment probabilities that you might have, from one generation to the next. So as you go multiple generations down, kind of as Jeremy was mentioning, how likely are you to be able to assign, to a great grandparent or whatever?
Yeah, so we aren't doing grandparentage analysis at all. This is all like parents and offspring and siblings. Those are things we can, especially with the aging, that's where that connects, because then we can distinguish between a parent offspring pair and sibling pairs. You can do a heck of a lot with that. The grandparentage thing is a little dodgier, kind of an emerging area. We may be able to do something with it at some point, but right now we're not counting on it. thanks. I'm happy to talk more offline.
Awesome. Thanks, Brad.
Yep.
All right. So up next, we have Michael Helmer at the Fish Bio. Is going to continue some of his presentations on some of the life cycle monitoring in STAMP.
Wait, how do I get it up? How do I get a new presentation?
Sorry. That was my one job. All right.
Good, excellent, thank you. Is it afternoon? It is afternoon officially. Good afternoon, everyone. And yeah, thanks for being here at this lunch hour for this session. As introduced by Mike and Brad here, I'll be presenting on monitoring steelhead in the Stanislaus River, this effort we've been talking about, and just give a recap on the methods that we're using, particular to the adult steelhead monitoring. Some of the challenges that we've encountered over the years, what we've learned from them, and also some of the insights that we've been able to gather over the past few years. And again, the overarching goal of this monitoring effort is to provide accurate estimation of adult escapement, adult steelhead escapement, to improve our understanding of the current population status of Omycus and steelhead in the watershed. Evaluate their migration characteristics and facilitate these juvenile production estimates. So a little bit about methods to set the stage. I know Brad described this, but this is the Stanislaus River flowing roughly 100 kilometers downstream of the barrier to anadromy, downstream of Goodwin Dam. With the Stanislaus River, we're located about halfway down before the confluence with the San Joaquin River. So just above river kilometer 50, well downstream of the known spawning reproductive rearing reaches on the Stanislaus River. So for the methods, the predominant method here is weir monitoring. Sounds simple enough, right? Build a fence across the river, intercept the fish, funnel them down into a trap box and sample them as they come through. That might be fairly easy to do when the weir looks and the river looks like this. This is a picture from last December at about 200 CFS. You'll note on river right, that trap box, that's where the fight goes into the trap box. That's where fish are trapped. So they're current flowing from top to bottom here. So fish are coming from the top, hit the weir, look for a passageway. The only way through is through the fight and into the trap box. And there they can be trapped, netted, handled, and the physical samples collected. On river left here, you'll notice there's a passage panel for downstream migrants or kelts. So that would be, we haven't been able to trap any kelts. I think kelts are extremely rare in that river. So, but if they do come down, that's the only way to pass over the weir in a downstream direction, and we'd be able to trap them there. So as I mentioned, the intention is to intercept migrating adults at the resistance board weir downstream of the spawning ground. So all the adults that are expected to contribute to the spawning population will have to migrate past that. There's a passive detection there. So there's a vacuum river watcher integrated in that system, an additional camera system in case of vacuum outages and to verify species ID, presence of adipose fin, et cetera. And there's pit tag detection systems integrated in the passage chute of the weir as well. As I mentioned, the intention is to capture and physically sample the migrants in that trap box when conditions and permits allow. In the past or during year one, the winter 2021, 2022, we were able to operate the weir continuously. I'll share more about that in a little bit. So pretty much when discharge is less than about 1,500 CFS or so. However, trapping was a little bit more limited due to the location of the trap box. We were able to trap successfully at just above 500 CFS or so. And the other limitation, and I'll mention a little bit about that too, is we can only trap outside the peak salmon migration period, and it'll become fairly obvious here why that is. So a little bit about the challenges, three main ones, and perhaps the main one is flow. So we need suitable flows for continuous weir operation and trapping. I showed the picture earlier of the Stanislaus River at about 200 CFS. So it's easy peasy when the flows are like this. When they look like that, high and brown, this is about 4,500 CFS, picture taken in January of this year. The weir completely submerged. All the electronics monitoring equipment that you would see on the left side of the image here had to be moved out of harm's way. So that makes it fairly difficult to trap. We have made some improvements to our trapping approach that allows us to trap at higher flows this year, but certainly not at flows as high as this one. The other challenge that I mentioned is the Chinook salmon migration. Chinook are clearly much more numerous than steelhead in this system, and are fairly prohibitive of trapping during the peak migration period. So we are limited to, at least we try to limit ourselves to a maximum number of about 30 Chinook at any given time in that trap box, which typically results in us starting to trap after November 1st. That being said, the maximum number of Chinook we've held in the trap box during a single trap check, and mind you, during peak migration, these trap checks occur pretty much 24 hours a day on a 1 to 2 to 3 hour interval. We had up to 53 Chinook in the trap. That was in early November of that first year of 21, with another close to 40 fish trapped later that day, and I think over 150 over that 24-hour period. So those are measured and sexed, and adipose fin clip or adipose fin status is determined before they're released. So there's no tissue or any other physical samples collected from those fish. And then to the last main challenge, which is actually the inherent steelhead behavior itself. It turns out that steelhead are much more difficult to trap in that trap box than Chinook are. We have observed, based on the Vackie and the attached camera system, multiple times that steelhead will move through the passage chute, go into the trap box, fall back out, move through the river watcher, through the passage chute again, move back up. So we usually have to do a bit of sorting to determine the actual net upstream passages. And happy to announce that retrofitting the actual fyke trap, or the entry into the trap box with these trap fingers, is a concept that we borrowed from the commercial trap fishing world, has drastically improved the trapping success. So far this monitoring season, so the 2022 and 23 winter, out of the 5 fish that we have seen moving through the Vackie and the camera, while the trap was actually operating, we've been able to trap all 5 of them. So some of the insights from past monitoring. So this weir actually has been in operation since 2003, but with the monitoring effort mostly geared towards Chinook salmon. And as Brad summarized, Chinook monitoring and steelhead monitoring have some important differences, migration timing being one of those as well. However, what we have been able to get passage data for a fairly large number of steelhead or Omicus in general over the course of those 20 years, with the very broad insights that upstream migration is, as expected, greatest from October through February, but in the Stanislaus relatively rare overall. And the other insight that kind of, with some variation over the course of the year, but the other insight that kind of holds true is that about 50% of individuals overall are of hatchery origin. So if we look at this graph, and mind you, the disclaimer here is that this is not standardized monitoring effort throughout the year, because the monitoring was focused mostly on Chinook salmon in years past. The weir and associated equipment was typically operated from early October, sometimes through December, sometimes it went all the way through early mid-June. But this is just a summary of all the observation or all the Omicus observations that we have collected over those years, with the naturally produced, as indicated by the adipose fin status fish pictured in blue here. Those with an absent adipose fin of hatchery origin with the red dots, and then there's some of unknown origin where the picture, the imagery just wasn't clear enough to decisively determine which one was which. So this includes also this regulatory or nominal cutoff of 406 millimeters. So this graph includes just over 300 fish that were smaller than that steelhead classification cutoff of 16 inches, and close to 200 fish that were larger than this. And you see a pretty abrupt stop to the fish detections here at 200 millimeters, and that is attributable just to the sensitivity of the Beccio River washer that doesn't reliably pick up fish that are smaller than that. So from that first full year of monitoring, since the Bureau started spearheading this monitoring effort, when we actually had continuous monitoring from September all the way through May, in 21, we started monitoring in early November, which is typically right after the floating public goes through after Labor Day weekend, and then stops or continues just until before Memorial Day, when we see, again, increased recreational use of the river and associated risk to the public when they have to go over the weir and get hung up on the weir. So we try to take the weir out right before that happens, and also because just based on 20 years of monitoring experience and monitoring data is that steelhead passage before or after that time is extremely unlikely. So in 2021, 2022, we had 27 total net upstream passages that meet that size category of 406 millimeters, with about 17 of exactly 17 of those being ad-clipped or of hatchery origin, and then 23 net upstream passages that were smaller than that 406 millimeter size cutoff. So that particular year was fairly easy. We pretty much have a steelhead escapement census, which was 27 for the season. And over the course of the season, this is kind of how it fell out. With the first detection, I believe, was September 25th. And some detections kind of throughout the year. The last one of steelhead size was observed on 2/11, so early February. And again, the symbols represent that different size classification, with the circles being those smaller fish that seem to be much more likely to be detected during the spring period. And the orange colored symbols are those fish that we were actually able to trap, capture, and handle in the trap box. Maybe to back up briefly, so when fish are trapped in the trap box, they are measured fork length, total length, they're weighed, their adipose fin clip is determined. We take a tissue sample, we take a scale sample, they get a Floyd tag, so they could potentially be rediscovered during carcass survey. We scan them for the presence of a pit tag. If they don't have one, they get a pit tag. That whole process takes a couple minutes, and the biological samples that we collect during that effort are then passed along for the kinship analysis to the folks at Kramer. So a couple insights from the current water year. I probably don't have to tell anybody in this room that it's been a little crazy when it comes to flows. We've had intermittent monitoring starting the middle of September and ongoing, expected through late May, with the weir currently being out, or the monitoring equipment being out due to high flows. We do expect a planned decrease in flow here early next week. I think this flow is supposed to come down for a couple days. That's when the weir is going to get back up and running and trap operations are going to resume. So far, we've had 6 detections this year, only upstream detections, and only larger fish, or fish that meet that 406 millimeter size cutoff. The earliest one was in late October, 10/22, when we were not trapping yet. And then the latest one so far was in early March. I already mentioned no small fish yet this year. And then, yeah, just a couple of general insights from last year to this year. The trapping operations and at high flows are, in theory, much improved over the year before. We were able to trap in the first year at flows over just over 500 CFS, as I mentioned. This year, we were able to move the trap box associated equipment closer to the bank, put in a walkway that staff could access the trap during higher flows. We were able to use or install a crowder to corral fish into deeper water. And can now operate the trap at flows to about 1,500 CFS, and that is the point when the trap box becomes completely submerged. So that's, what is it, just over 5 feet tall. I already mentioned we modified the trap entries with the installing the flexible pike, or those trap fingers, to reduce that fallback of Omicus, and that seems to have helped greatly. And kind of auxiliary to all this, and we sent a crew out to the McCallamy River hatchery to help us refine the length coefficient for the steelhead measurements. So most of the measurements that you saw in that plot come from, it's an extrapolation or an expansion factor. So the Vacuum River Watcher, the infrared scanner, basically measures fish depth and then uses a user-defined coefficient to extrapolate to the total length. Since we had a fairly limited number of individuals to work with in the past to define that coefficient, we wanted to refine that, and had a crew go out and measure over 150 steelhead at the McCallamy River hatchery. And it turns out that length coefficient, based on the larger sample number, is a little bit larger than we were previously aware of, than the coefficient we were previously using. So that being said, we will be using the updated coefficient to update past numbers as well. So a lot of those individual fish that were hovering right around that estimated 406 millimeter cutoff will probably officially make it into a steelhead classification, changing those summaries slightly. And with that, I want to acknowledge first and foremost the Bureau for spearheading and funding this larger effort, and then the collaborators at CDFW, Oregon State, and Kramer, who are all involved in various aspects of this broader monitoring effort. And with that, if there's still time, I'll try to answer any questions if there are any. We have a couple minutes if we have a question. Going once.
All right. Thanks, Michael.
All right, thank you.
All right, before I introduce our final speaker for the session, I did want to note that Steve Culberson wanted to give a couple quick announcements before everyone heads off to lunch. So if you guys wouldn't mind just sitting tight after this next talk, that'd be great. So last speaker for the session is Lauren Diaz, a PhD student at Oregon State University, and she's going to be talking about some exciting new modeling on steelhead habitat use and life history expression. So you, Lauren.
Thank you, Mike. So I'm going to be talking to you all about a meta-analysis I did on the ontogenetic niche shifts of Omicus. So in the sense of defining my jargon, ontogenetic just means like changes associated with growth and development. So habitat use for different age classes, and trying to find correlations between those habitat uses and different geographic and climate variables. Also, I know I'm the last person before lunch, so thank you for sticking with me. I'm also quite hungry. If I forget words, that's why. So I'm going to be talking about niche breadth, which basically refers to the range of a species niche. So generalist species have broader niche breadths than specialist species, and that can be dependent on that can be relative to other species, so and different spatial scales. So a species may be a generalist at broad spatial scales, but then need more like specific habitat requirements at those finer scales. And it's also as we also believe that this relationship is related to geographic range. So species with broad geographic ranges are going to have broader niche breadths. That makes sense. And so there's also these concepts of niche separation and niche overlap. So species that the tails of their niche breadth don't really overlap as much are obviously going to not be sharing resources. There's going to be little competition between those species. And when there's regions of high niche overlap, that's where we're going to see competition. So the concepts of niche are also usually when you're comparing different species, but not as often they're applied to within a species when you're talking about different age classes. So within species, you're probably going to be seeing more niche separation where there's either growth in orders of magnitude, like for this hellbender that's going to be experiencing really big changes in growth throughout its life cycle and using vastly different habitats, or for species that exhibit metamorphosis. So for example, a lot of aquatic insects, an adult mayfly is not going to be using the same habitats as its larval counterparts. But then in species that exhibit more gradual changes throughout the life cycle, you're going to be getting more niche overlap, so more intraspecific competition. So Omicus are a really good example of a species that exhibits both these gradual changes and also that are more age structured, and then these drastic changes that are more stage structured. I will say this is probably not the best example of like gradual change. I've heard this is quite the chonky rainbow trout. So this is probably not the typical size. So you've heard a lot about steelhead today already, but just to reiterate some of these concepts. So they have two main alternate life history forms. There's the freshwater resident form and then the anadromous form. And so these two life history forms are believed to exist to maximize resilience to environmental change, as different forms can maximize fitness in different conditions. So between steelhead, the migratory form, and the freshwater stages, that's going to be an example of niche separation. There's not really going to be much overlapping habitat between those. But then within the freshwater stages, you're probably going to get more niche overlap and more intraspecific competition. So why does this matter for this context? So steelhead are declining pretty much across the board. That's no secret here in the Central Valley. And rainbow trout are still ubiquitous. So it's kind of an interesting management problem that we aren't really sure what to do with. So anthropogenic changes might be incentivizing residency. So different ocean conditions or low survival through the delta, or more higher productivity in freshwater might be incentivizing residency. So it's not really worth it for a fish to invest the energy and potential mortality in migrating to the ocean. So we might be seeing a higher proportion of these populations remaining in freshwater as residents, and rainbow trout becoming the primary adult life history form. And so more rainbow trout than historically in these freshwater populations might increase or exacerbate intraspecific competition and density dependence, and may lead to slower juvenile growth, basically creating like a bottleneck in the life history distribution of these populations, since maybe less juveniles would be able to grow to meet those size thresholds necessary to actually smolt and migrate to the ocean. Alternatively, this may be creating cycles in life history expression, where smaller juveniles create smaller, less dominant adults. Future cohorts can then grow to these smolting thresholds and so on and so forth. So we just basically don't really know how these shifts are going to affect population dynamics. And so here in the Central Valley, steelhead are a threatened distinct population segment. So there's an interest in understanding how anthropogenic changes interact with maybe like individual characteristics like genetic and sex, to understand how those shifts will alter population dynamics and potentially population resiliency. So my goal is to develop an individual-based model that can hopefully get at answering that through simulations. And so this is kind of my conceptual model of how the system probably works. It's believed that Omicus, that growth is basically what makes them turn into a rainbow trout or a steelhead, growth in early life. And so these are all the different factors that could be influencing that growth and then influencing how their fitness would be different for these different life history trajectories. So I'm going to start to tackle this by tackling, I've been one thing behind, I think, this whole time because I'm looking at the wrong side. Okay, so I'm going to start by tackling this habitat use aspect of how are different age classes using habitat and how is that going to affect, how is that going to affect their growth? So I did this using a meta-analysis. So I basically synthesized the available literature on Omicus across their global range. So that's including their introduced range as well. And I had two main objectives, which were to relate their habitat use to geographic and environmental factors, and then to identify regions of niche overlap that may be contributing to interspecific competition. So I did this, I started by searching the first 15 pages of Google Scholar and Web of Science for papers that were relevant to this topic. So I originally downloaded papers that were original research results. So I did not include like reviews or book chapters that targeted Omicus specifically. So not necessarily Salmonids or Ongryncus as a whole. They were spatially and temporally explicit, which basically just means they listed when and where the study was, which you'd be surprised that not all papers did that. They were field studies, so I didn't include any laboratory or hatchery experiments. They addressed age-specific habitat use. So the three ages I focused on were young of the year, juveniles, and adults. So a paper didn't have to include all three as long as they mentioned which one specifically they were talking about. And adults in this context refers to resident adults, or some of the papers talk about summer steelhead, like their holding habitat. So that would also count as that. And then finally, the paper needed to be quantitative. So it needed to give me an exact number of what kind of habitat they were using, not just like high velocity, low velocity. So after I downloaded those papers, I went through and searched them a little more thoroughly and extracted use metrics for total depth and water velocity. I also did this for substrate and focal elevation and distance to cover, but for the purposes of this talk, I'm just going to focus on these two. They're also the most important and the ones with the most data available. And then I also included any information present in those papers about obviously age, location, so basin characteristics like slope or drainage area, study characteristics like season, or the evolutionary history of the location. So that would mean, are there congeners? Are they landlocked, whether it be natural or through an artificial barrier? Yeah. So my first question again was, what drives ontogenetic variation in depth and velocity use? So first I wanted to quantify how the variation in all these studies related to identify what portion of that variation was due to just like the study or the ecoregion. And then I did a second level of fitting models within that, where I fit models related to age, climate, geomorphology, and biotic interactions. And then my second question was, where is interspecific competition likely to be the strongest? So I fit standardized mean differences to all of the data, which is basically a meta-analytical term that will tell you how different two groups are, including the amount of variation between those two groups. And so I did these pairwise comparisons between each age class. So young of year versus juvenile, young of year versus adult, juvenile versus adult for both velocity and depth. And so of those, of all the papers, all the 177 papers I downloaded, I could only use 36, which was not very much at all, a lot less than I expected given how commonly studied rainbow trout are and their global range. But there's a lot of reasons for that I don't have time to go into in this talk. But even within those 36 papers, we still managed to get a pretty good amount of diversity. So those 36 papers ranged across 53 years, 4 countries, and 5 different ecoregions. So for depth, we were able to identify study and ecoregion level variation. So 42% of the variation in all of the results had to do just with study, and then 46% with ecoregion, and then the rest of it could be accounted for by our covariates. And then for velocity, 99% of the variation was just study specific that could not be accounted for by any of our covariates or even by ecoregion. And so by our covariates, that means like precipitation, temperature, for like basin characteristics, like slope, drainage area. And so what this tells us is that there's a big part of the story that we're not really seeing. There's probably more specific site level variation having to do with, say, like biotic communities or different flow or management regimes. So yeah, there's a lot that we're not seeing here. So our top model for total depth included the effects of age, mean annual precipitation. And so to see what that looks like. So here in this graph on the x-axis, we have mean annual precipitation and the y-axis is predicted total depth. And then we have the 3 different age classes, adult, juvenile, and young of year. And then the shaded areas are their confidence intervals. And so what we see is that although there's a slight quadratic effect and a plateau, for the most part, as mean annual precipitation increases, depth use increases. Kind of makes sense. We can't really see the exact mechanisms as to why these relationships exist with this high level data, but probably just means that there's more water available in the system. And then as you can see, adults use deeper water than juveniles, which use deeper water than young of year. So this is representative of a Mediterranean California ecoregion, which obviously that's where we are today. But I did mention that there was a lot of variation associated with ecoregion for depth. So when we look at that here, you can see that temperate mixed forest and northwestern forested mountains used below average, fish in those systems used below average depths. And then North American desert and marine west coast forests were about average. There are these 2 lines kind of on top of each other. But what's interesting is that in Mediterranean California ecoregions, fish were using way deeper water than average. And so this is interesting. I don't live in this system. I don't do field work. So if anybody has any idea why this would be, I assumed it might have been a like temperature drought kind of thing. But then when we look at North American desert, that's not the case. So I'm curious to see what other people think about this. And then for water velocity, our top model included the effects of age, although a pretty weak effect, and then the negative effects of mean annual precipitation and summer mean high air temperature. So for mean annual precipitation, so this is the same graph as before, just for velocity instead of depth. We see that there's not really much differentiation between the ages at all. The confidence intervals are all kind of on top of each other. But as precipitation increased, fish were using slower habitats. So again, we can't really say much about the mechanisms from this high level data, but I would guess that it maybe has to do with more of the like floodplain area being inundated in areas with high precipitation, which are usually slower velocities. And then for mean summer high air temperature, we see pretty much the same relationship. So in warmer climates, fish are using slower water. So this can maybe be because of energetic constraints. Fish don't really want to waste energy in maybe these climates that are close to their thermal maximum. It could also maybe be due to them selecting thermal refugia in like deeper pools. But there interestingly wasn't any correlation between depth use and velocity use. So yeah, again, can't really tell why this relationship is what it is based on this high level data. All right, so my second objective, niche overlap. So before I totally get into it, I just want to go over what we're looking at in this graph. So each panel represents an age class pair. And then with standardized mean differences, the direction of the effect is going to be about like the first relative to the second. So in this first panel, young of the year are using shallower water than juveniles. So that is kind of like just standardized for, if I were to switch the way that I compared them, that would be a positive effect. It's also a significant effect if it's greater than 0.5 in either direction, and the confidence intervals don't overlap 0. So when we're looking at the significant effects here, we can see that they're all for depth. So this is basically just telling us in a different way what our regression results told us, which is that the age classes are using significantly different depths from each other. And that the smaller fish are using significantly smaller depths than the adult fish in each of these comparisons. And then again, for velocity, this is basically telling us the same thing, well, the opposite, which is basically that there isn't really any niche differentiation, and there's high niche overlap between the age classes. So anyone who works in these systems will probably tell me that this is very like obvious data, and to a certain degree, yes, like these are things we mostly know, but it takes putting them together to see the big picture of what we know and what we don't know. So what do we know? We know that fish are partitioning habitat based on depth, and we know that all ages use about the same optimal flow velocities. And what we don't know is kind of what is the causality of this. Like, is it due to availability? Is it due to actual preference? Or is it due to biotic interactions? So unfortunately, when a lot of this data is collected, it is mostly habitat use data, and we don't really have a lot of habitat availability data. So it's hard to say, like, if fish are actually using a habitat that they prefer, or if they're just seen there, maybe a bigger fish actually use their preferred habitat, and then the surveyor sees them there and is like, oh, this fish likes this habitat. Also, these things don't exist in isolation, like depth and velocity are going to be interacting with substrate, with each other, with a variety of different things, obviously, that we could not pick up in our analysis. So there's a lot more to be studied here, and a lot more finer scale variation that would be cool to put into this meta-analysis. But I think there's still some takeaways that we can get from this that are useful for informing management. So basically what I get from this is that habitat heterogeneity is key. So it's important if you're restoring habitat to make sure that there's like ample space for juveniles to grow away from adult encroachment. So yeah, so that basically makes it so that there's multiple depths available at these like shared velocity levels that they all share. And a really great way to do this would be maybe ensuring access to floodplain habitats. That's really good for juvenile rearing and that adults don't really use. So yeah, again, something we know, but it doesn't hurt to add evidence to the pile. From this, we can derive habitat suitability indices that we can adapt to different ages, different ecoregions, different watersheds, and so we are going to put those in three kind of broad definitions. So there's the entire range of used habitat, which is like, okay, what habitat is suitable for the species? There's the median, which is kind of the 25th to 75th percent quantile of used habitat, which is like what is better than just like wherever they are. And then the narrow, which is kind of 10% plus or minus around the mean, which would be kind of their preferred habitat. So we can know what that is at different age classes and incorporate all that geographic variability into that to get really fine scale estimates of what habitat is good or what is not good for the species. So from that habitat suitability, we can then use that to develop rule sets for how these different fish interact and apply that to 2D hydraulic models. Specifically for the Stanislaus River, which you guys just heard so much about. That's going to be our model system. And then hopefully incorporate those rule sets into an individual based model to figure out how these different interactions go along with habitat availability in different flow levels and how we can expect these interactions to maybe affect how fish are growing and how we can expect different life history trajectories to come from that. So just to reiterate a little bit, so our model system is the Stanislaus River. It is directly managed by Bureau of Reclamation, which is our funder. So it's a really good system to later apply all of this research onto. It doesn't have a hatchery on it, so that kind of simplifies population dynamics a little bit, and it has a depressed steelhead population. So we are getting information from the life cycle monitoring program that you guys heard from Kramer and Fish Bio and Cal Fish and Wildlife, and putting that all together into this individual-based model that can then derive a metapopulation model. And so I say metapopulation model because we do have really high stray rates from other hatcheries. I suspect it's from the McCollum, but we don't really know specifically where. That's going to be like a later thing I hope to incorporate. And we can use these models to then make management decisions using decision support models and figure out maybe how these different management actions influence life history diversity and population resiliency. So that is it for me. If you have any questions, feel free to email me. Do we have any time? Okay. Thank you.
I have to push when I talk, okay. It doesn't work? Oh, it does work, okay. Hi, welcome to the afternoon sessions. So this one is about monitoring and management of Central Valley spring Chinook salmon. I'm Flora. Really happy to see you all here, and thank you for coming back from after lunch. We're going to have some nice talks. And the first one, I want to introduce Eric Holmes from UC Davis.
Thank you, Flora. Thanks to everyone for coming back after lunch. Flora is a co-author on this as well, along with Miranda Bell-Tilcock and Karsten Jeffries and Rachel Johnson. Today we'll be talking about juvenile spring run Chinook salmon rearing and growth patterns in the Butte Sink and Sutter Bypass. I'll begin with a quick introduction about the historical decline of spring run, and then I'll dive into juvenile life history strategies as a background, the ones we see in Butte Creek. And then we'll talk about the research that we're doing measuring the habitat conditions in the Butte Creek, in the lower Butte Creek watershed, including the Butte Sink and Sutter Bypass. And then we'll get into a little bit of the application to wild fish. So a little recap on the historical distribution of this population of spring run. The Central Valley population used to have access to high elevation Sierra Nevada habitats. They would take advantage of snow melt to climb high up in the watersheds where they could find cool water habitat to over summer, and they had plentiful spawning grounds. They also had their juveniles also benefited from an estimated 4 million acres of seasonal wetland habitat in the Central Valley. But as we all know, the construction of dams, impassable dams, cut off a lot of that spawning habitat and access to cool water, which really constricted that distribution. And there was also loss of that important rearing grounds on the valley floor. And this led to a precipitous decline in this species from which the escapement numbers were estimated hundreds of thousands and now are just in mere thousands. But it's not doom and gloom everywhere. Butte Creek has shown a resurgence over the last couple of decades. And this can be This is due in large part to conservation efforts in the 90s, which were very impactful, including like the Western Canal siphon, which was put in 1995, and other passage improvements for juveniles and adults in the lower watershed. And also the stewardship from CDFW, people like Tracy McReynolds and Clint Carmen, and countless people up there who provide great stewardship of this population. But its relative performance compared to other nearby watersheds has a lot of people interested in what's going on in this watershed and how is this population doing so well when others are still struggling. So to investigate this, we're going to talk a little bit about the patterns of juveniles in the watershed and how they're growing over time. So this is data from the rotary screw trap, which has been managed for over 2 decades by CDFW and Chico. And this, the file that I saw had over 1.7 million entries in it. So there's a lot of work being done here. And it's really nice to have a vision of what's happening up there. And this is right at the valley floor, and it's catching those juveniles as they're coming out of the headwaters and into the lower portion of the watershed. And if you see it the way I do, and actually I view it a lot similarly to the way Jeremy does, I'm going to use his terminology here. This is Jeremy Notch, who describes the runners and walkers. So you have the runners, which are coming and exiting the headwaters soon after emergence. They're coming out, below 40 millimeters in length, fork length, very small fish. And then you have your late ones, your walkers. They tend to hang out in the upper watershed for longer. They spend a little more time and they grow to smolt size and they emigrate then, a little later in the season. There's also yearlings, which are fairly rare and to my knowledge are not well understood. And I'm not going to cover them anymore in this talk. I'm really focused on the fry and par, these runners, and the opportunities they have to grow in the lower watershed, which includes the Butte Sink on the northern part of this yellow square or rectangle, and the Sutter Bypass, which is the northernmost flood bypass in the Central Valley Water Project. So we identified 4 primary habitat types in this region. This includes river channel, canal channel, wetland, and agriculture. And the wetland agriculture, these are off-channel sites. These are your floodplain sites. And so we interspersed sampling sites out here. So we had 45 unique site and year combinations. This was a 3-year study. So in the river channel, we had 5 sites that we repeated over the course of 3 years. And at these sites, we measured water quality. We looked at zooplankton trends using zooplankton tow nets. We also did a cage experiment where we had individually pit tagged fish, and we looked at their growth rates over the course of about a 6-week deployment in each year. And then we also caught wild fish opportunistically in these habitats using seines and pike nets. And we're trying to look at their growth rates and kind of match that up with the cage data, make sure everything's looking good. Before I can dive into the results, though, I need to remind us all of the hydrologic context here. How wet were these years, and how does that affect the results that you're going to see? So as you can remember, 2019 was a very good water year. There was a lot of weir overtoppings and a lot of flushing events. And conversely, 2020 and 2021 were dry and critically dry. This was like the heart of the drought. 2021 was the lowest I've ever seen it out there. It was ugly. And so this affects the results that you're going to see because we were under full flood during the experiment in 2019 versus very low flow conditions in 2020, 2021. And so the food web is very responsive to this hydrology, these flooding events. And so the top image here shows the bypass when it's fully flooded. It's turbid, it's fast, it's cold. It's kind of like a wide, shallow river. Whereas the bottom one, the flood event has passed and you're seeing a longer duration inundation. You're seeing higher residence time, allowing that food web to really take off out there. And you can see this in patterns of dissolved oxygen. That's the green squiggly line there. More diurnal variation is kind of showing that productivity taking off versus during the food web reset, you're not having much photosynthesis respiration happening during the day. You're also seeing a dilution of the zooplankton getting flushed out of the system. Those red bars indicate zooplankton amounts. And of course, the blue was the hydrology. All right, now we can dive into some of the results here. So if you've been following along with floodplain productivity research over the past couple decades, including work done in the Cosumnes River and in the Yolo Bypass, you're not going to be surprised that the floodplain had much higher zooplankton densities than did the river. So very similar to past results. So we affectionately refer to these cups as the zoop soup. This is when it gets very productive out there, and the fish can really take advantage of that. So you will notice though that the top one has slightly lower zooplankton than in the dry years, so comparatively. And so because these things are orders of magnitude different in densities, I'm going to go ahead and shift this to a log scale, just so we can kind of get this thing off the bottom of the plot and we can view temporal trends in this. So when we look on the graph on the right, I know it's busy, but I just want you to look at 2019 and see how hydrology is affecting your zooplankton densities and how it's sort of homogenizing the system. And this is a trend you're going to see later as we look at fish growth versus in years where you have long residence. And these are wetlands that are being managed through irrigation canals and managed for waterfowl habitat, but also providing fish access and fish habitat. And how are the salmon in the cages? What are they eating out there? So this is also a busy plot. So I'm going to give you a minute to sort of acquaint yourself with the colors. So the red color here refers to amphipods. The purple is cladocerans, and this a lot of Daphnia pulex and the larger bodied ones are the ones we find in the guts. Copepods, primarily Acanthocyclops. And we have ostracods as well as insects, and this includes your dipterans like your chironomid midges. So I'll focus on the off-channel ones first, and we note that their diets are mostly composed of amphipods, cladocerans, and copepods. And conversely, the river channel is much more dominated by your chironomids, your aquatic insects. And then when we look at the effect of hydrology, we see that the floodplain has more of a river-dominated diet in the wet year. Not too surprising. You may also have noticed that I didn't talk about canal channels. They're a little bit weirder. They have much more variance in their diets. And we'll get into that. There's a spatial component, which we'll get into in a minute. And we look at the fish growth patterns. We notice that off-channel sites were the highest in all years, and their maximum growth rates reached up to 1 millimeter per day, versus your river channel sites on the other end of the spectrum, which had slow and consistent growth, which was sort of surprising given differences in hydrology. They were always consistent, always about the same. And here's the canal again, showing the extremes. There's a lot of variation here again in the canal channel habitats. And you'll notice that this homogeneous system, this theme of system homogenization during flood events is happening here, is shown here in the fish growth rates, where we see this compressed range of values. All right, so what's going on in these bypass canals? This has been a source of a lot of variation. And so I've taken the growth data and I've plotted it vertically. So the x-axis is now looking at your growth rate. And you see that this spike in the northern part, and this number 39.2, this is your latitude, so it corresponds to the map. And so these points with the arrows pointing to them are in close proximity to the wetlands of the Butte Sink. And this is really the driver of that. These connected wetlands are really driving these faster growth patterns by providing the food subsidy. And so you see it very clearly when I switch this from fork length growth rate to median large cladoceran density. You see the same spike there coming out of the wetland. However, you see this longitudinal depletion as it moves downstream. The apple is still relatively intact. It has higher zooplankton density in total individuals. However, you're missing a lot of those high value prey items, which to me signifies that there's a lot of competition for those high value prey items and a top-down trophic effect. And we look at the correlation between this median large cladocera density and your fork length growth rate. You have a very strong relationship here. All right, so what does this mean for the wild fish? If you have access to these different habitats, what's that going to do for your growth rate trajectory and your development timetable? So we estimate from our cage data that it takes about 47 days, plus or minus 9, to get from 45 millimeters to 75 millimeters, to get up into that early smolting range. Conversely, And this is a bit of an extrapolation because we only ran for 6 weeks. And so we weren't able to see the full range of growth in the river, but that could take up to 4 months. And that's time that can be pretty important. But that's just cage data, right? So what are the wild fish doing? When I plot fish that we catch in the lower watershed, you see that they follow this wetland track pretty well. And I showed you a minute ago that they don't necessarily have to be in the wetland to get a wetland growth rate. They can actually benefit from them from that positive flow coming out of the wetlands into the canal channels. And when I refer back to Jeremy's work here, personal communication with Jeremy, he shows through his years of doing acoustic tagging that the migration survival is very tightly correlated to the hydrologic conditions, how much flow, the temperature that's on the landscape. And so this migratory window in the spring can stay open in a year like this where we have a mountain of snow up there. But in other years, this window can slam shut as early as April. And what you're left with are these runners, which got in there and had the opportunity to grow quickly and potentially get out of the system before it deteriorates. And that at least is the way we think about the system. But it needs testing, right? And so the next steps of our projects are to look at the returning adults. These are the winners. These are the ones that survived and came back. And we're looking at archival tissues, looking at the otoliths to see, look at their growth rates and when they, their strontium profiles to see when they left the system. And so here we show that around 80% of adults grow to smolt size before leaving the Butte Creek watershed. And complementary data to that is in the eye lenses, where you see the integration of off-channel food resources by this depleted sulfur signature. And about 98% of the adults collected in 2021 showed integration of this off-channel derived food resource. And we need to get more years, more returning classes to look at the patterns, dry versus wet, and see how this tests our hypotheses. So what does this mean for other Central Valley Chinook populations? Well, they need a place for the runners to grow. And so this means enhancing like more floodplain restoration, continuing with the weir notching, kind of lowering the threshold of activation, and then potentially flow pulses to activate floodplain habitat. And in years where you don't have enough water to get the floodplain wet, there's potential to bring floodplain food resources to the fish. And with that, I would like to thank our partners. This includes agency partners and funders, U.S. Fish and Wildlife Service, and as well as our private landowners, letting us come out there and sample their fish, or the fish that are on their property, and the tireless work by our hardworking lab. So thanks, everyone.
Any question on Zoom? Any question in the audience?
Hi, thanks. Great presentation. What is it about those? Is it the Claudocera that they're targeting in on? Like, do we have any idea of like the caloric content of those versus like all the other stuff?
There's a body of literature out on that. I think actually one of our later speakers, Russ Perry, would probably know quite a bit about this. A lot of stuff I've read is, has been done in like the Columbia River or farther north, where they're dealing with larger size fish, and they actually show the Daphnia pulex and these cladocerans to have a fairly low relative caloric value. But I think for this size class that we're looking at, the factors are the Daphnia pulex, the one I'm going to focus in on. They're pretty ubiquitously distributed through the water column in space and time. Where you look at some of the other food items like chironomids are more patchy, in space and time out in the system. And so it's kind of this it's good metric, because when they're there, they eat them and they must process them well. It seems to be when you see a lot of them, you get a lot of fish growth. And so to answer your question, there's a lot of research out there, but it actually doesn't point to them being higher in quality. I think it's just their abundance and their easiness as a prey item lends itself to the growth. Thank you.
All right, thanks. So our next speaker is Brett Harvey from DWR. He's going to talk to us about the JP.
Good afternoon, everybody. Hope everyone's waking up after their food coma, that rush hour after eating lunch. Like Flora said, I'm Brett Harvey, and I'm in charge of a fairly large multi-agency effort to develop an approach for estimating a spring run juvenile production estimate for the Sacramento River Valley. I'm just curious, how many people here know what I mean when I say juvenile production estimate, or I think they have an idea? So a lot of people, that's good. Well, just in case, I want to emphasize that a juvenile production estimate is that what we're talking about is a forecast. So it's being able to forecast how many spring run juvenile salmon are going to come out in any given year, hopefully before they come out. And the reason why we're doing this is, well, because we're required to. But besides that, they're listed fish. We don't really have a great method for figuring out just how many are coming out each year. There's also a performance measure in the current preferred alternative, if anyone knows what that means. But we're also, we have a number of life cycle models, and there's other life cycle model in development for spring run salmon, and this effort's also going to fuel those efforts. So we had a big workshop that a lot of folks that are listening to this talk right now probably attended, and culminating in the production of a science plan that was approved in February of 2021. And now there's a lot of people working really hard to implement that plan. And I won't go through the list, but this is just the agencies, and at each agency there's scores of people in some cases. At the heart of the plan there's 5 key elements that we outlined. There's monitoring of spring run, obviously, to collect data. We've started a run identification program, genetics. I'll talk a little bit more about all these actually in the talk. But there's a run identification program. There's so we can tell which of those fish caught in the monitoring are actually spring run. There's a pretty comprehensive data management plan that we're implementing and a data management system. And then there's modeling. And then finally, there's a structured decision-making process that most agencies are involved with. And just to sort of step into that structured decision-making program a little bit, we've defined various approaches that could be taken to estimating, to producing a spring run juvenile production estimate each year. First of all, there's one that's sort of like the winter run JPE, which is where you just come up with one big number of how many spring run you expect to come out of the Sacramento River each year, and enter the delta where that star is on the map at the point of delta entry. But we've also envisioned that it may be useful for developing minimization, operation impact minimization measures, if we were able to break that pie up of the JPE into the contribution from the different tributaries that are producing spring run. Similarly, it would be potentially really useful to understand the contribution of yearlings versus sub-yearlings or young of the year. And finally, and I don't know if we'll be able to do this, but actually trying to come up with some sort of a prediction of the timing of when we expect not only the full cohort of spring run to enter, but also the various contributions would probably be fairly useful in development of protective measures, but also just for developing other conservation actions. And in essence, like the juvenile production, the approach we're taking is to develop a stock recruitment model where we look at the number of juveniles that come out historically. We look at the number of different various life stages, whether it's passage of adults in or adults holding or post-spawn carcass surveys. There's various life stages you could use in one of these models. And then you plug a bunch of potential environmental covariates into that, or biological covariates like water temperature or flow or pre-spawn mortality. But gathering all that data requires a lot of effort out in the field. And so from that monitoring component of the program, we bolstered a lot of the monitoring going in what we essentially we're calling representative streams. We were thinking we would take a subset of spring run streams and really focus on those. But as it turned out, the only thing everyone could agree on was basically all of the spring run tributaries that we think have, that aren't just satellite populations or dependent populations. And in particular, I just want to point out, we implemented a new screw trap location in the lower Feather River that's kind of a complement to the one on Sacramento on the Knight's Landing. And we also have a similar, another new screw trap array that's down near the point of Delta entry. And these are experimental trapping locations. So from the run ID side, we've been sending out genetic testing or sampling kits to all of the screw trap locations in the Sacramento River Valley. And we're using those to develop probabilistic length at date models. I think many people are probably aware of the length at date model. I'll just actually show this. So a length at date model, for those who don't know, is essentially where you catch a fish on a certain day, it's a certain length, and then you determine which of those black lines it's between, and then you say it's either spring run or fall run. But typically the true, or what may be true progeny of spring run or some other run may not fall within their designated line. So we're developing models that for every screw trap location that will give a probability for the for which run that a particular fish might belong to, and then we'll scale that up to come up with stock estimates for the various runs at these screw traps. And then we just started this, but we're hoping we can then back look at the historical data to help, because right now it's kind of difficult to come up with a spring run estimate based on historical data and length of date. And just a quick peek at the, I'll just throw a few quick peeks of data in this talk. Just to get an idea of what's going on with spring run genetics, this is from the first year of results, and the green of each pie are the length at date designations that matched the genetic designations. And Jeff's going to talk a lot more. I don't know how much Jeff you're going to talk about this stuff, but this is about it for this slide here. But it's just to give you an idea that the gray area are the ones that did not match up. And so we're trying to shrink that pie. So we're hoping that our probabilistic models have a lot more green and a lot less gray. But more importantly, we want to be able to propagate that uncertainty through to the juvenile production estimate. All the way through, we're trying to propagate uncertainty through the sampling to be able to apply that to our final juvenile production estimate. And this is just a quick, this is a quick and dirty cut at a probabilistic length of date model. This is for Battle Creek. And if you, for those who can distinguish, the pinky red dots were the genetic spring run, and then the green ones are the genetic fall run. On the left side, is a probabilistic model for spring run, showing the mean size of the genetic spring run with a confidence interval. And don't ask me any questions about heterozygotes for. That's Jeff's job. And then the dotted lines are the 90% confidence interval around, I believe, around the distribution of fork lengths for the genetic spring run, similarly for the fall run. And then we've also just wrapped up the field work on a health welfare study so that we can understand how the various kinds of genetic tissue sampling that we're doing affects the health of the fish, because we want to minimize that to the extent possible. So this is something that I'm really excited about, and it's the data management system. So I'm sure, is everyone here really excited about data management? Because I've become very excited about it. And so I'm just going to kind of walk through what we're doing right now, and most of the stuff is really ready, almost ready to launch. Most of it's occurring in a pilot sort of a framework right now. But most of the programs that collect spring run data in the valley, even if they're on the same sort of data management platform, like such as CAMP, don't necessarily match up. They're not necessarily compatible, and that's part of the compatibilizing part of the title of the talk. And that's been a real problem. It took us about a year to actually go through a lot of discussions with the people collecting data to create a compatible database so we could actually go and start working on the stock recruitment models. And so what we're doing now is working with all of these different programs to develop data management plans. And what those essentially do is ensure that the data from the point where you actually enter it into a computer is compatible with every other program collecting the same kind of data across the Sacramento Valley. So as soon as that stuff gets uploaded into our cloud database, it's already compatible. And you can crunch that data without any concerns, or large concerns that the data is just not really fitted together well. And to take it a step further, we've actually developed an app using open source software. So it's going to be available to everyone and for anyone to mess with, because it'll be the code will be posted on GitHub. But it's for tablet-based data entry in the field, so that from the point of tapping the data into that tablet, it'll get uploaded to the cloud, and it'll be compatible with all the data that's being collected across the valley. And we're hoping right now we're doing this in Deer Creek, Mill Creek, and the Yuba River, and as a pilot application, and we're hoping that it is adopted by many other programs, not only in Sacramento, but potentially down in the San Joaquin, as I've been told. But that's out of my scope right now. We're also doing a similar thing for the genetic data. So the genetic data, and we're developing we're continuing to do the sort of historical sort of genetic tests, but we're also working on rapid genetic tests so that we can get that genetic data rapidly into the cloud, and then it'll get crunched through the plaid models to give us the run designations for the fish that are being collected, and then ultimately that information will feed back and allow us to target genetic sampling for the fork lengths that have the greatest degree of uncertainty. That's the dream anyway. And then we'll also be able to attach these things to, we're right now beginning the process of automating some of the models that we build, so that the data will build and auto update the JPE models, potentially even through the season. So you might not have your one JPE. There may be a defined JPE for some sort of regulatory measure, but through the season, that JPE is going to be auto updated based on the data that's coming out of the various tributaries. So we know what's going on. I don't know if people are familiar with the winter run JPE, but sometimes the fish that actually make it to the Delta probably aren't quite what we predicted. So we're hoping to provide more information. And then about every week or two, that cloud repository, the cloud database, which is somewhat private to the people that are collecting data, we'll dump out into a repository in EDI, into provisional databases that are annually then moved over by each sampling program into a more approved historical database. And then that becomes a place where external users can get that data, but we're also going to have automated pipelines of data that go to regular users, like the salmon monitoring team or SAC-PASS or the CDFW databases where they also collect this information. So just to sort of walk quickly through the modeling thing, I already mentioned that sort of stock recruitment approach that we're taking, that we're then flip around and use to create a forecast. And we pretty much did our first cut of the whole estimating juvenile abundance at all of these screw traps. We were about to publish that report, and we were working on a manuscript, but we held back because we got a lot of feedback. And now we're updating our models, and you can expect something mid to late summer that'll be made more public on that. And by the way, the EDI database is also just about ready, the historical data, to be daylighted. We're just waiting on final approvals from various agencies. And now right now we're working on this database, on working up the adult data. And this is the only thing I'm going to show for modeling, but this is just an example of what we've done. We've used a hierarchical Bayesian approach to estimating abundance so that we can use the catch across the valley for where there's some kind of correlation going on between the valley to particularly in trap efficiencies, but also for trap abundances. And so on the bottom of the figure are the catch probabilities or trap efficiencies. And all of the red boxes are essentially where there was no trap efficiency releases. That's where they take fish and they mark them and they release them upstream of the traps and figure out how well they're able to sample those fish. But using this approach and being able to borrow data from across the system, we're still able to come up with abundance estimates. And that's what's shown in the top figure here. And this is for the Feather River eye riffle. And just for anyone who's curious, those little blue circles are the Peterson point estimates that you would get if you just simply applied your catch efficiency to that data for that week without looking across the weeks or across the region at data. And this is the last slide. This is our timeline, and I'm barely over time. And I don't even know why I put this in because it's really boring. It's a table. But people always want to see a timeline of what's coming up, what's coming up. So the first row there in the sort of the warm colors is our expected dates for the data management plan. We hope to get the data into EDI, that says by the end of March. So maybe mid-April. We'll be working on code automation through the spring. And then we're going to be working from July onward on these data pipelines and the documentation for going beyond the screw trap data into the adults and getting all that into EDI. The next line is the modeling. So we have draft models pretty much that we need to have done by the end of June. We have a structured decision-making date. And that's in the bottom rows, the structured decision-making, where we need to get some serious model selection by the end of the year. By end of March of next year, we have to turn all this stuff over with a recommended approach to an independent review panel. And then they get, I think, way too much time to review, and give us feedback. And then we finally submit that to DWR, who then determines whether they want to submit it to CDFW or not. And that's where we're going. Thank you.
So we have time for questions. Is there any question on Zoom?
It's usually better to use all your question time up.
Yeah, I actually have a question, and I have great talk, and I'd be interested to learn more about the data stewardship side of things. But my question is a little bit from left field in that I'm not a fish biologist, but I'm wondering if any of your covariates that you're considering ever look at like physiology, behavioral things with elevated temperature in terms of predicting behavior.
Well, the difficulty is I don't think we're really coming up with a mechanistic sort of behavioral model. We have to have that data for every year and across the entire span of the program in order to model, juvenile production. So although I think that the behavior informs the model and it informs how we, like, for instance, determine a young of the year versus a yearling.
So
ultimately,
it's only based on past behavior.
Yes, okay. It is.
Yeah, that remains a question.
Yeah.
Any other question? Nothing on data management?
So hold the genetics questions. The next.
Thank you, Brett. So our next speaker is going to be Jeff Rodson. Sorry if I butchered. who is going to talk to us about genetic.
Let's do one quick thing here first. Yeah, so good afternoon. Yeah, I'm Jeff Rosen from California Department of Fish and Wildlife. The co-authors on this are Joy Gaines from our laboratory at CDFW, as well as Melinda Bearwald from California DWR and Sean Canfield from UC Davis. So this talk here that I'm going to give you is really kind of builds upon much of the information that Brett just presented to you as far as sort of the background of what the Central Valley Spring Chinook Juvenile Production Estimate Program is. It's about a mouthful. So anyway, so what I basically want to present to you is kind of what we've been doing on the, more specifically on the genetic side in relation to the Spring Chinook JPE, and really kind of use this as sort of a case study to really kind of illustrate how, in some senses, even like the genetic marker of choice that you're using for Chinook salmon monitoring can paint you actually quite different pictures depending what approach that you're taking. So just a little bit of background. I know this has already been covered by a couple of the previous presenters. You know, of course, Central Valley Chinook salmon are very, very heavily managed, recreationally and commercially important. You know, we have several different runs or races or whatever you want to refer to them as of spring Chinook salmon in the Central Valley, anywhere from the fall run, which are not listed at all, which you can commercially and recreationally harvest, to fully protected winter run Chinook. So really what I want to present is sort of a case study, you know, about how we're using genetics, you know, in our laboratory in collaboration with DWR to use genetics to help identify the particular, you know, genetic types of juvenile salmon that are being captured, you know, in that rotary screw traps to help try to inform the probabilistic length to date model that Brett was just referring to in his previous presentation. You know, as we know, there's a complex regulatory environment in place, you know, in relation to just the overall management, regulation, take of Chinook salmon between fall run, spring run, winter run, you know, in the Central Valley, and just how that affects a wide variety of different operations. You know, as Brett mentioned or alluded to, many of you are familiar with the traditional LAD model or length of date model that's been used for quite a few number of years to try to identify a specific run of a salmon, of an out-migrating juvenile salmon, depending on its fork length. You know, maybe, you know, as many of you know, it can be accurate or fairly inaccurate, really depending where in the system you're actually taking the data from. So that's kind of helping to feed into the probabilistic new length of date model that Brett alluded to, that Noble Hendricks is actually working on developing for folks in the Central Valley. You know, as Brett alluded to as well, there's a large sampling event currently underway to characterize patterns of genetic diversity and when and where the protected juveniles, mainly spring Chinook and winter Chinook, are occurring in the Central Valley as they out-migrate. So the data I'm going to present is essentially the results of our genetic testing that we've done on, I think, roughly 1,500 juvenile samples that were collected in several valley locations earlier in 2022 between January and May. We're currently now in our second year of collecting, but we don't have any data on those quite yet. Oops. Yeah. So as we know, there's some different life history ecotypes of salmon in the Central Valley. Largely, these have been categorized quite a bit, you know, on the time that the adults are returning to fresh water. So I'm going to introduce a couple of terms that I'll be referring to. We call essentially like the late or the early life history ecotype. You know, these are fish that tend to arrive in, you know, winter and spring and then hold during the summer in cool water and then spawn in the fall. That's traditionally what we think of as our winter and spring run Chinook. You know, as opposed to what we call this late life history ecotype. Those are the adult salmon that return to fresh water, you know, in late summer, you know, and in the fall and winter, or late summer and fall. What we traditionally think of as our fall run. So really, the early fish are the ones they arrive in winter, early spring as adults. They hold it, so when they arrive in fresh water, they're not ready to spawn. So they have to spend a few months in fresh water, becoming fully ripe and then ready to spawn, you know, in the early fall. You know, fall run fish, you know, they arrive in, you know, late summer, early fall. They arrive in fresh water. They go straight to the hatcheries and they're pretty much ready to spawn. But what we're really interested in this study is the out-migration of juvenile fish. So what we've been finding generally is we think these fish that are early, kind of your winter and spring run juveniles, you know, tend to out-migrate first from the Central Valley, and then the fall run fish, we believe, you know, they're juveniles tend to outmigrate, through the system, out through the delta, later on in the system, later on in the season. That's been kind of shown, through the LAD models and quite a bit of other work that's been going on. But, we wanted to dive a little deeper into the genetics since we have a great opportunity to do this. So what we've been working on, again, in a partnership between our laboratory and DWR, is to try to dig a little deeper into the genetics of these outmigrating juveniles. Really, how do the genetic patterns that we see in these different groups of juveniles, how does it vary over time and space? So there's a couple different approaches that are used on the genetics. And so I want to spend a couple of minutes to talk about these because they paint quite different pictures of the genetic composition of a stock of fish that you're looking at or a set of samples. So we know in the salmon world, and maybe I've heard of this, is what we call these early or late fish. Also linked to this small section on salmon chromosome 28 that we know has a very tight association with what we call this early or late ecotypes. Fish that tend to return as winter/spring Chinook, or fish that tend to return as fall run. And so that's also referred to, as kind of this ROSA region, this section of chromosome 28. You may hear people refer to it as the Greb1L or Greb1L/rock region. As opposed to more traditional genetic testing, which you kind of use a set of genetic markers, maybe 90, 100, couple hundred markers that are spread all the way across the genome to do kind of more traditional genetic stock assignment, to where you may have heard them classified as like, this is a fall run, a spring, a winter, or late fall fish that are based on genetic markers that are spread all the way across the genome. I was going to show you, in the Central Valley, depending which approach you take, you can get very different pictures of what the actual genetic composition is of the set of fish that you have in hand. So kind of what did we find? So this is essentially a summary graph. I'm going to spend a few minutes on this. So this is a summary graph of the results of our genetic typing that we've done on, again, about 1,200 to about 1,500 or so juvenile fish that were sampled between May and June of last year. And so what we see, again, remember, these are outmigrating smolts. And so one of the things I want to point out, so essentially here on the left is January, and then we see kind of a temporal trend over all the way to the right, which is May. And this is kind of combined valley-wide, where at our early sampling dates in January, most of the juveniles proportionally going out are, we call this early haplotype, meaning those are fish that are going to come back as essentially spring run or winter run Chinook, as opposed to the orange down here at the bottom, which are we call late fish. Those will return as adults as fall run adults without fall run adult migration timing. The green fish in the middle are fish that actually, individual fish that actually have one genetic copy of this early form and one genetic copy of a late form. We know that those fish tend to return as adults in the Central Valley with kind of a more intermediate return timing into fresh water. They kind of, in the Central Valley, they tend to come in more closer to when the fall run fish come out, but they're kind of very important in evolutionary context because those fish actually are a genetic reservoir of both spring and fall genetic variation. So really just big picture, as we see kind of a temporal trend over time, is that we tend to see early in the year, kind of January, majority of the juveniles we see out migrating are ones that are going to come back as spring run or winter run adults. And then as we go across the season towards May, then we see almost all predominantly out migrating fish being the ones that are going to return as fall run adults, in 2 to 3 years. Now, interestingly, what we see is that this pattern of out-migrating juveniles can actually vary quite a bit between your sampling location within the Central Valley. For instance, here's Butte Creek on the top left. This is Battle Creek, I'm sorry. And so what we see is sampling event 1, which is January, we still see a lot of out-migrating fish that are essentially going to come back as spring run or winter run adults. So we see a lot of blue going across almost till May when we see a handful of fish that are fall run, as opposed to, for instance, Feather River here, which is Feather River Mile 17, to where even, we didn't have fish from the first January event, but maybe late January, early February, what we see here are predominantly fish that are going to come back as fall run, and very, very few actually spring run fish scattered throughout that plot. And so kind of long story short, what it really shows is that the genetics of out-migrating juvenile salmon at any time point really is pretty highly variable depending where exactly in the Central Valley you're looking. You know, where are you looking and what are you collecting? So if you've heard of so if you think traditionally of like the 3 ESUs of Central Valley Chinook salmon, you would generally think of like fall run, spring run, and winter run. Now, if you look like across their whole genome, as opposed to the previous slide, we'd only looked at their run timing genes. We generally see, fall run fish, their spring run fish, and then we know winter run are a lot different genetically, primarily because they're pretty highly bottlenecked. I'm going to shift gears here a little bit and just show you now how the genetic patterns look when we actually look all the way across their genome, not just looking at a very particular section that has to do when those fish return to fresh water. So interestingly, what we see up here is that we see a cluster of individuals out here by themselves, which is our Battle Creek fish, which is kind of our more traditional kind of genetically pure spring run, if you want to think of them that way, and ones down here that are more of our kind of genetically pure, for lack of a better phrase, spring one in Butte Creek. And then over here on the right is kind of everybody else. And so interestingly, like if you look at across the whole genome, is that you don't see a whole lot of fish that really, in the Central Valley, there's not a ton of fish that really pop out as being a, kind of both a early fish and a genetically spring run stock when you look all the way across their genome. Because we know in the Central Valley, there's a lot of integration that's gone on between what was traditionally a spring Chinook and a fall run Chinook. So basically what we look down here is across all of our other sites, there's Night Landing, Tisdale, Feather River sample sites, is that they're pretty much inseparable from one each other when we look at a couple hundred markers spread all the way across their genome, that they're all pretty closely related to one another. Again, with the exception of Butte and Battle, that tend to still sort of pop out as kind of their own unique genetic populations. So now let's look at when we look across markers across the genome, as opposed to one particular section that only looks at run timing, what do the genetics look like now? So interestingly, I know the numbers are small and kind of hard to read, and there's a lot going on this slide. So what we find here is that the very early sampling dates like 1 and 2, remember on the couple slides back, this is kind of basically 1 and 2 sampling dates, essentially kind of late January, perhaps very early February. What we see are these guys, again, across the genome, looking quite a bit different, than the other fish that we're finding. So in here is a handful of out-migrating winter run smolts or par that we found, as well as, again, probably the early spring run that are starting to come out of like Butte and Battle Creek. Interestingly, as we shift down here, I don't expect you to be able to see the individual numbers on here. So kind of big picture is that we see kind of on the left side of this distribution is our fish that are, again, kind of coming out maybe around sampling date 3. So again, these fish down here are ones that are coming out, again, probably late January, probably early February. And then as we go through the sampling season going this way, as we get towards, mid-May, is that we see essentially kind of a trend really in our genetic distributions. That's almost has a very tight correlation with actual sampling date. So we kind of go like 3, 4, here's sampling dates 5 and 6, 7, 8, 9, and 10 finally over here. And so really kind of what we see is sort of a trend valley-wide is really kind of a trend in sort of the shift of our genetic composition of our stock going, from the early in the season to late in the season. Again, not entirely unexpected, but it really kind of goes to show that as even from May, January to May, is that we see pretty interesting and kind of almost like a pretty interesting, temporal trend in how the change of the genetic stock of our out-migrating juveniles really changes and evolves, from getting fish you collect in the RSTs in January to when you get to the end of that field season in May. So what does this mean? Well, there's a lot more details in here I don't really have the time to go into, but just kind of takeaways generally is that genetic patterns that we see in Central Valley Chinook salmon really vary both by time and space, but also with the genetic marker of choice. So if you're involved in a genetic monitoring project, and perhaps as a manager or end user, is really be cognizant of what specific genetic markers the folks that you're working with are using, because they may paint quite different pictures of what the actual genetic makeup is of the fish that you're handing them to analyze. So interestingly, like I said, the genetic identification of Central Valley salmon can be quite nuanced. The juvenile outmigration timing really kind of more closely associated with a small segment of salmon chromosome 28. So like if we look back, we have a slide a few slides ago where we saw like these early fish, the early late fish, and then the late fish. We saw kind of a very, measurable or detectable trend going from sampling date 1 in January all the way through the end of May. We really kind of saw kind of a systematic shift, on 2-week periods, going from predominantly juveniles that are to come back as spring or winter run to almost a stock composition that was almost entirely composed of juvenile fish that are going to come back as fall run adults. So the traditional genetic stock ID, where I talked about like this old school kind of classification, where we had like using genetics to assign this fish as like, winter run or winter fish, or spring fish, or fall or late fall. In some systems, that's actually pretty closely aligned with what we get from results from our chromosome 28 typing. Like Butte and Battle, for example, was almost entirely fish that were going to be early, having that spring, kind of coming back as spring adults, as opposed to ones like in the feather and Yuba, to where they're across their entire genome, their genetic background is essentially very similar to that of what we call fall run Chinook, with the exception of this one particular very small part of their genome that has some of that stock returning as a spring Chinook. So when you think about like the spring run Chinook in the, in the feather, for example, is you have fish that are sort of like this fall run genetic background, but those individuals actually migrate up in the spring. So they have that same ecotype, that same return to fresh water as a true spring run Chinook do. And so kind of long story short is, like I said, it gets complicated. It is nuanced. So just something to keep in mind, like if you're a manager, biologist, environmental scientist, and end user of genetic data, is you really do need to pay attention to how exactly they're collecting the data and really kind of what markers they're using and what inferences they're trying to draw from those, because they can paint quite different pictures for you. So what are our next steps? Well, there's 2 more years of juvenile sampling and genetic data collection. So we finished year 1. That's what we presented today. We're right in the middle of year 2 sampling. And then there'll be a 3rd year of sampling and genetic typing next year as well, under our current contract that our lab has with DWR. So it'll really be interesting to see kind of how these patterns hold up over years as well, because of course last spring, wasn't that great of a water year. And then this year there's been tons of storm events with high flow, which have actually been sort of problematic in some senses, because there's been a lot of times when they haven't been able to safely fish the traps, because of high flow events. And then, we're in California, so who knows what next winter is going to be. Maybe it won't rain at all, or maybe it'll snow in the Central Valley. Who knows? It seems to be the way we're going these days. So anyway, this kind of provides really kind of an opportunity to really do kind of a deeper dive into patterns of genetic diversity in our outmigrating juvenile Chinook. We've spent a lot of time, looking at the genetics of returning adults, but there really hasn't been a whole lot of work done really looking at the genetic stock composition of the outmigrating juvenile. So this really could probably make a pretty neat paper, in a couple years, along with all the other things I provided to write that I never have. But okay, so and then of course, being able to incorporate new information into salmon management. So again, not just monitoring a take, to help inform the new probabilistic length of date model that Brett was referring to, but also just kind of answer some broader questions of just population genetics and population resiliency, to be able to look at perhaps questions of like relative spawner success in some of these different tributaries, some of these different ones. Like if you're fishing a certain date and have a group of fish that you've collected, some of the other questions we have that a little bit, probably wait till the end of the study to see like, how many spawners actually contributed to that group of fish that we collected. Is they the result of only a couple successful spawns? Or are they quite a large number of families of fish that we're collecting? And all that stuff kind of feeds into this broader picture of how do we really manage, Central Valley salmon to be, to help maintain their resilience. Are we doing things that are, perhaps promoting only, a handful of families to be produced each year and spawn successfully? There's lots of different rabbit holes and questions we can go down. So just some acknowledgements is definitely all the hard work from our state and federal stream teams have been out there diligently collecting and fishing these traps, all last winter, this winter, and they'll be at it again next year. California Department of Water Resources, of course, they provided the bulk of the funding for the work that we just presented. The genetic markers we're using, our panel was provided to us essentially as a courtesy from the Garza Lab, at the NOAA Southwest Fisheries Science Center. I didn't really go into this, the Sherlock assays that Brett was referring to, the real-time genetic testing methods, which are kind of still under development. Those are being done, in Andrea Schreier's lab at UC Davis, the genomic variation laboratory where Sean Canfeld is working on this. And then nothing last but not least, our CDFW, Central Valley Tissue Archive, Leah Korber's shop in CDFW. And then, of course, Laura for organizing our session and keeping us all on task. So with that, I don't know how I did on time, but if we have time for a question, probably take one or two.
Yeah, we have time for some questions. Oh, I see one here. Brad. That's nice. Thanks.
Oh, thank you.
So with regard to the adult immigration timing, an early fish that isn't a winter run is definitely a spring run. Has a decision been made about what a late fish is? Is it a spring run or is it a fall run? Because it seems like that question needs to be answered in order to produce a juvenile spring run production estimate, right?
Yeah, no, that's perhaps the $1 million or maybe multi-million-dollar question. I mean, what exactly these, I don't call them hybrids or heterozygotes, what exactly those fish are? I mean, people tend to do this dichotomous classification of a winter spring versus a fall fish. But there's a lot of these fish out there that are heterozygous that carry both genetic variants. And ecologically, they perform a very important role because they're a reservoir of genetic diversity of both life history types. But in a management context, where do they fall out? I mean, that's a very open-ended and currently unanswered question. So a lot of us have been thinking about that. Like, how do we want to manage them in the Central Valley? Because it's kind of been shown that if you take two fish that are late heterozygotes and you breed them, you basically will have families that are a fish where some of them, they have some offspring that are early. They're going to come back as spring run. They're going to make offspring that come back as fall run fish. And then they're going to make some heterozygotes just by pure Mendelian segregation. And so, yeah, it's a really good question. A lot of folks are thinking about that. It's truly kind of unresolved at this point.
Is it leading anybody to question the approach that's being taken? I mean, because if you can't, you need to make a classification. And if you can't make a classification, the goal of what we're trying to do isn't going to work, right? So somebody needs to make a decision, or you need to say, we need some new tool for develop or distinguishing the runs that isn't available yet. So let's not do this right now. You know what I mean? Like, it has immediate implications, I guess is what I'm saying.
Yeah. I mean, I know we traditionally think of them as like coming back as like a winter spring entry into freshwater versus a fall type entry into freshwater. But there is this other group of fish out there.
Right. But what are they?
And conservation. And so really in a conservation context, you almost have like three groups of fish. You have your spring winters coming in. You have these heterozygotes that kind of come in between. And then you have your fall run fish. So like, I don't know that we can just necessarily just ignore them because they're out there and they have some conservation value. But again, it's a really good question. It's definitely problematic in the long term. But I'm hoping eventually, through enough discussion and stuff, we can come up with some sort of a coherent, path forward. Right. Yeah. Yeah.
Any other questions?
Have any online, Stephanie?
Okay.
Okay.
Well, thank you, Jeff.
Oh, thank you, Laura.
And last but not least, Russ Perry is gonna thank you to everybody who attended this session and didn't fall asleep because of food coma. We have now a 20-minute break, so it's 3:23. We are reconvening at 3:40.
All right, welcome everybody to our final session of this IEP workshop. Thank you to everybody at IEP, especially Christine Joab, who did so much work on this. Thanks to all of you. Oh yes, round of applause for her. And thanks to all of you for hanging out for this final session. I think we have some great speakers and presentations lined up. My name is Jeff Steinhardt. I'm with the U.S. Fish and Wildlife Service, and I'm one of the co-chairs of the IEP Predation Project Work Team. My other co-chair is Tyler Pilger, who will always also be our first speaker. And Tyler started studying predation as an undergraduate, actually, when he attended Fort Hays State University in Kansas. He stayed in Kansas for his master's, went to Kansas State University, and then switched more towards population genetics for his PhD in New Mexico before he joined Fish Bio in California, where he works on multiple large river and kind of basin-wide predation studies and other work. So Tyler will kick off with our first presentation.
Excellent. Thank you, Jeff. And I want to thank you, Jeff, for spearheading this session and getting the speakers lined up. So really appreciate that. And thank you, everybody, for being here on the last session of the day. Today, I'm going to be talking about a multi-year predator research study that we've been performing on the Stanislaus River. This particular study came about in response to the Water Infrastructure and Improvements for the Nation Act, which was signed in 2016. It's funded by the Oakdale and South San Joaquin irrigation districts, and the study design and permitting came through partnerships with NOAA Marine Fisheries Service, as well as California Department of Fish and Wildlife. And as you can see, all the names on this slide, many people have contributed to this program or to this study over the years. So the Stanislaus River, we had some steelhead talks earlier today. I'm going to run quickly through this. The Stanislaus is an important tributary, is an important salmon-bearing tributary to the San Joaquin River. And not just for fall run Chinook salmon, but steelhead, rainbow trout, and Pacific lamprey, as well as a suite of other native fish species that are resident species. So the upstream limit barrier to anatomy is Goodwin Dam, which is pictured here. So all spawning and rearing has to occur in below that dam. And so the beginning of the migratory corridor is roughly around Oakdale, California, which is the top of our study area. And so all of those tiny diamonds on this map here in the lower portion of the river are one of our sampling units. And there's 39 of them that we randomly selected in 2019. And we've repeatedly sampled them for multiple over a year, each year on back-to-back days, starting in February and going into June. Okay, so today I'm going to focus on 3 research objectives that link the vulnerability of salmon to predation to the diets of these piscivores, their distribution, and their demography. So in addition to characterizing diets, we used occupancy models to estimate occupancy of striped bass and mark recapture methods to estimate abundance and survival of black bass. And for the purposes of this study, we lump largemouth, smallmouth, spotted bass into a single black bass functional group. Here's a quick rundown of the methods that we use. We capture fish using boat electrofishing. In each of these units, we sample one bank out to the midline of the channel. The fish that we capture, we measure. We also take scales for aging. We perform non-lethal gastric lavage to collect stomach contents. Those are preserved and then taken back into the lab. In the lab, we use visual identification to kind of separate out invertebrates and stuff from fish. And then we use genetic identification, or we do use genetic methods to identify fish down to species. And then we also release the fish with pit tags for mark recapture. And the thing with this study design, sampling back-to-back days, allows us to use models that account for imperfect detection. Now I'd like to give you guys you all an idea of what the sampling conditions were like in each year. So each panel on this slide represents one of our sampling season with date across the x-axis. And those gray bars there indicate each of our sampling events in each season. So we missed a season, we missed an event, February event in 2019. In 2020, we missed our event in April. I think you maybe know why. And then in 2021 and 2022, our events later in June got canceled due to critically high water temperatures. So the left y-axis is discharge and the right y-axis is temperature and turbidity. And I want to focus just on the blue line for a bit. So in our sampling events in 2019, which was classified as a wet year, we had higher discharge during our sampling events than discharge during 2020, which was a dry year, or 21 or 22, which were critically dry years. If we focus on the red line, that's water temperature. No surprise there, water temperatures were cooler longer into the season because it was a wet year, there was more water than our dry or critically dry years. And then the last thing on this slide is turbidity. So the circles represent the mean turbidity for the sampling event with the lines representing the minimum and max. So the max turbidity that we saw across all years was in 2019, but it was still under 20 NTU, which means the water was still quite clear, even in the higher flow events. All right, we're gonna jump right into diets. This is the only diet slide I have for this entire predation talk. So the top panel is the diets for striped bass, and the bottom panel is for black bass. And the height of those bars, each group of bars represents a sample year. I follow wet, dry, critical, that's the same following along. And then the height of those bars represents the proportion of predators that we saw that consumed a particular prey item. So the prey items are Chinook salmon, lamprey, and then we grouped the other native species together as to one. And then the orange is the any non-native fishes that were found in the diets. So as we can see across all these years, both striped bass and black bass consumed more native fishes than non-native fishes, and particularly Chinook salmon and Pacific lamprey. And then black bass were consuming a lot of sucker fry and sculpin. Okay, so as this study progressed, we started seeing some interesting patterns in our data, particularly with striped bass. So we don't catch a ton of striped bass, but they are there. And as we sample throughout each season, our early sample events, we pick up large striped bass. And then as the season progresses, we get more and more smaller size striped bass. So we used some length frequency stuff and found out that 300 millimeters was a pretty good size cutoff. So the darker color here is fish that are greater than 300 millimeters, and the lighter color is fish that is smaller than 300 millimeters. So we decided to look at this pattern across all years through occupancy modeling framework. So here, the squares represent the circles represent fish, striped bass that are larger than 300 millimeters, and the squares represent the fish that are smaller than 300 millimeters. And the x-axis here is date. And so each of these points lines up with one of our sampling events. And the y-axis is the estimated proportion of sites, of those 39 sites throughout our reach, that were occupied by striped bass. And so essentially what we saw was that at the beginning of our sampling events in each season, we saw a higher proportion of occupied sites occupied by striped bass than large striped bass than small striped bass. But then as the season progressed, both size classes occupied greater proportions of the river. All right, we're gonna jump now to what we're seeing with the black bass. So black bass, we actually captured quite a few of these and enough that we can actually run some marker capture models. So we use this robust design marker capture model in program Marc. And what it does is it allows us to estimate abundance within each season by assuming that the population is closed to births, deaths, immigration, or emigration. And then it allows us to estimate survival from year to year, assuming that the population is open. So if we focus on abundance first, so our first year of abundance had really wide confidence intervals. We didn't have as many pit tags out there in the system yet. We didn't have as many recaptures, so a lot of uncertainty in that estimate. But as the years progressed, we started getting lots of recaptures and our confidence intervals tightened. And we saw a jump in abundance, black bass abundance in 2021. And I'm gonna get to that on the next slide here. So if I jump over to survival, so we ran these models, a series of models looking at different covariates for survival, looking at some different flow conditions at different times of the year. We allowed survival to vary from year to year, but the only model that came out as the most strongly supported for survival was the length at initial tagging. And so what we see that across all these years, the 4 years that we have so far, black bass survival is associated with the fork length at tagging, and it's quite high. So even across the range, we're seeing survival rates around 80% or higher. Okay, marker capture data wasn't all that we have. We were measuring these fish, so we have some really interesting length data on these fish. So each of these, so these panels here are what we're calling population pyramid plots. So some of you may have seen these kinds of plots for like human populations only by age. So each bar here represents, we're using it for size. So each bar represents a size bin, a 10mm size bin. And the width of that bar represents the proportion of the catch in that size bin. So for example, in 2019, we saw a good chunk of fish that were under 150. And we also saw a good chunk of fish that were between 180 and 300 millimeters. Okay, that's where those population pyramids get wide. Okay, so the fish that were under 150 survived to a pretty good clip into 2020, as well as those larger fish. They also survived to a pretty good clip into 2020 as well. But then in 21, we saw a huge influx of fish under 150 millimeters. So this meant that there was a pretty substantial recruitment event. So we think that conditions during summer and fall of 2020 allowed high spawning success for black bass, for those fish to recruit and for us to capture them in 2021. And again, that slug of smaller striped bass, age 1 plus striped bass, survived to a pretty good clip into 2022. And we can see that signal on that last plot there. Now, another important thing with the diet data is everything over 90 millimeters, all these black bass over 90 millimeters are capable of consuming Chinook salmon fry. And we've seen that. Every time we go out and we capture black bass, whether it's early or late, they're consuming Chinook salmon. All right, so I'm gonna do a quick rundown of these, our objectives. So both black bass, striped bass and black bass consume more native fishes than non-native fishes, and this occurred in both high and low flow years. And actually the proportions of fish that they were eating remained pretty consistent across those different flow years. All sizes of striped bass that enter that are in the Stanislaus River are capable of consuming native fish. Black bass, we see that they're shifting to piscivory within their first year. Striped bass, we don't see any evidence of actual recruitment of striped bass in the river. They're coming in to feed, not to breed. Throughout the season, what we're seeing is by May, the lower migratory corridor is estimated to be occupied 50% or more by striped bass, and that occurs in both low and high flow years. And they are present year round in some low level because we have monitoring programs outside of this sampling that has documented or that has observed striped bass throughout the river. So they're in there at low levels all year round. Black bass are quite abundant in this migratory corridor, and they're very widely distributed. There was no point in us running occupancy models on them when they occur at 99 to 100% of the sites that we sampled. High survival for fish that are greater than 80 millimeters. I should have mentioned earlier that the survival rates only are for fish that we can pit tag. And that smallest size is 80 millimeters. So very high survival for those fish. Once they get to that age, they're going to be around for a couple of years. And then recruitment, our current hypothesis is that it may be linked to flow conditions in the summer or fall, but we're still trying to flesh that out. So what does this mean for juvenile anadromous fish? Well, this migratory corridor is a gauntlet of both roving and sedentary predators. So the vulnerability of Chinook salmon and other anadromous fish to striped bass, what we think their vulnerability might increase throughout the season as more and more striped bass enter the river. But for black bass, the vulnerability of predation might be more persistent in both space because they occur throughout the entire stretch and in time. So they're there early in the season and they're there late in the season and they're there year after year after year. And what we are, again, kind of hypothesizing right now is that these native fish are vulnerable to predation regardless of the discharge, how high the flows are. And we're basing that off of the data that we have from 2019 to 2022. Now, we all know that 2023, we're having very different conditions. We have both high flow and high turbidity. This is our last year of sampling for this study. So this is going to be an important data point to see how both high flow and high turbidity influence the vulnerability to predation. And so with that, if there's time for questions, but if you want, you can hit up myself or Matt Peterson with any questions about this study. Thanks.
Do we have any questions?
I have a lot of questions, so you and I should catch up afterwards or later on. So cool talk, Tyler. That was really cool data. One of the biggest things I was wondering is, do you have plans to link all of your data into a bioenergetics model and actually try to predict the quantity of Chinook salmon that are being consumed?
That is a step that is in the plans. Whether we end up actually having the funding to pursue those efforts is another question. But ideally, that's where we want to go, is understanding how many Chinook, how many fish that striped bass can eat, what temperature influences are for black bass, because we know that black bass, there is some evidence that they are somewhat temperature dependent, but it doesn't appear to be that way in this river. But a bioenergetics approach is the way to go. And this data is on its way to making it that way, in that direction.
All right. Well, thank you, Tyler. Next up, we have a team presentation. We have Paul Bergman, who is the NorCal Fisheries and Aquatic Science Practice Leader for Environmental Science Associates, when Paul is not at work managing a wide range of projects focused on Central Valley salmonids. He is probably doing something basketball related, either managing his office's March Madness pool or cheering the Kings to victory in the Golden One Center. Joining him is Andrew Kalmbach, who is a senior aquatic biologist with ICF, where he works mostly on trophic relationships in the Bay and Delta, including predation and lower trophic studies.
Thanks, Jeff. I get the fun anecdote, I guess. So I'm special. So yeah, light the beam, everybody. Light the beam. All praise the beam. So I'm here today to talk about a predation study that we wrapped up at the end of last year, looking at predation at an emergency drought barrier in the South Delta. So we have a large team of authors you see on the bottom right. Andrew's going to present on the results, and I'll be walking you through the methods and then come back for the conclusions at the end. And then I want to acknowledge the large team effort here. We had a huge field crew out there, 66 nights of sampling over the last couple of years. So a lot of late nights to bring you this cool data you'll see today. So just to orient everybody to this location, we're at the West Falls River in the South Delta. On the west is the San Joaquin River. On the east is Frank's Tract. And basically it's called the Falls River because there's a huge tidal flux in this location. There's a lot of flow going east and west. So as you can imagine, that's why DWR needed to put a barrier in. That yellow line that's located in West Falls River was there basically to limit salinity intrusion. So limit salt water from moving towards the pumping facilities in the south. And so this channel is 700 feet wide. There was 90,000 cubic yards of rock put in this location. So large structure. And we were focused in on this area that's 500 meters downstream and upstream of this location. So the red and that blue line. And we were looking at predation. So kind of like the incidental impact of putting in a large artificial structure in there, as you can imagine, could influence predation risk to listed species in this area. So we had a couple of objectives. This is the primary one, was to determine the effect of the construction, the presence, the notching, I'll talk about what that was, of the barrier on juvenile salmon predation rate related to other environmental variables as well. And then secondarily, as this barrier was in place through time, it was in place for over a year and a half, did predation rate change? Did we see a change in predation risk at this location? And our gear of choice here was predation event recorders, or PERS. We constructed 40 of these units using National Marine Fisheries Service design. I know Cyril's here talking later, and we consulted with him a lot. We wanted to make sure that our units were really in line with these other studies so our results could be comparable. So on the right there is a schematic of these devices. Basically, they're buoys, and on board you have a GPS transponder, a magnetic timer, and then tethered below them you have a fish. And in our case, we used a golden shiner as surrogates for Chinook salmon. So if the shiner was preyed upon, eaten, or was pulled upon, the timer would be triggered, so you'd know when that predation event occurred. And then the GPS transponder would show you where. So you could then link up exactly where and when those predation events were with those environmental conditions. So you can have a sense of what's driving predation risk at any given location. So we released a lot of these units over many nights. Here's a sense of the effort that we had here. So 66 sampling events, sampling nights. During that first objective I mentioned was really focused on those three first periods. So before construction, during and after construction. So we got out there 6 days before construction, 11 during, and then 10 after construction. And then a few months went by, and we decided to go back out there again to get a sense of how this was changing as this barrier has been in place. So we got 10 days in before it was notched. And then on January 18th, DWR notched this barrier. So they put in a 400-foot long notch, 12-foot deep into the barrier to basically, during this high flow period, to allow for boats and to allow for juvenile fish to have connectivity through this region, to allow passage basically. And then it was filled back in end of March, when we got out there for 3 more months of sampling, so 14 more days. And so sampling design, we focused on this sunset period. So when we believe, predation risk to be the highest, so we're out there 3 hours before to 3 hours after sunset. On the left there, you see the image of what the notch condition looks like. So during these open periods before and during construction and during this notch period, PERS were deployed 500 meters up current to 500 meters down current. Or if there was insufficient current, which happened, it's a tidal location, we would let them sit out there for an hour and then we would pick them up and redeploy. And then during these closed periods, so that image there shows you this closed condition, the barrier in place. We had 2 crews on each side of the barrier, and we released these units simultaneously, 2 different crews. So we either released them at the barrier to 500 meters away or 500 meters away towards the barrier, depending on tidal conditions. And just like the open period, we would let them sit out there for an hour if there wasn't enough current. And I'll hand it over to Andrew for the model.
So Cox proportional hazard model predicts the risk of some event, in our case predation, happening over the course of a time period. This is these 1 hour long deployments of an individual per. And so we are predicting this risk of predation with a variety of spatially and temporally varying covariates. The most important covariate is barrier period. Coming into it, we expected lowest predation before construction began, highest predation when the barrier was actually in place and hydraulically closed, and intermediate predation risk when it was notched and when it was under construction. We expected predation risk to decline as you moved away from the barrier, in the same way predation risk declines when you move away from shore. And we expected faster moving PERS to have lower predation risk as they rapidly move past any predators. And at least in shallow areas, we expected the bottom roughness to provide good habitat for predators. We expected risk to peak right around the beginning of nautical night when it got dark. And as far as water quality, we expected predation risk to increase with salinity, although this was expected more to be a seasonal effect than a direct effect of salinity. We expected predation risk to increase with temperature and to decline as turbidity increased. So what did we see? Before getting into the results directly, these plots just show this predation hazard ratio. The way to read it, if you double the predation hazard ratio, you double the risk. If you have the ratio, you have the risk. And the y-axis is on a log scale. So a doubling or a tripling is a consistent distance. So during those first 3 periods, the construction period and the post-construction period, predation looked pretty similar to the pre-construction period. They were getting preyed upon at about the same rates. But then about 3 months after post-construction in the pre-notch period, predation rates were much higher. And that was consistent through the notch period and the post-notch period until they removed the barrier. As far as these other parameters, on its own, distance to barrier had no effect. But I did look at the interaction effect between that and study period, and we'll get into that in a moment. Predation rate did drop as you moved farther away from shore, and it peaked right at night, right when it got dark. And predation increased with salinity and with temperature, and was not really affected by turbidity, per speed, or the bottom roughness. So the distance to barrier probably didn't matter before the barrier existed. So we looked at the interaction effect in between the two. And for these plots, the gray shadow and the dotted line show the relationship between distance from barrier and predation risk during the pre-construction period. The colored solid lines and shadow show the risk during that panels period. And so in the construction period, it looked very much like the pre-construction period, no risk in relation to distance from the barrier. And in the post-construction period, if anything, risk declined overall, and there was a declining effect of distance from the barrier. The farther you got away, the less likely predation was. However, in the pre-notch, notch, and post-notch period, we continued to see that trend with distance from the barrier, risk declining, moving away from the barrier. And there also seems to be this area, maybe about 150 meters away from the barrier, where the risk is higher than the pre-construction period of predation. And just as a reminder, this post-construction period and the pre-notch as well as the post-notch period, the barrier was in place and complete. So it looks like in the 3 months between the post-construction period and the pre-notch period, there may have been some colonization of predators. Finally, we noticed a discrepancy between our model and the observed predation. Our model predicted pretty high predation risk during the notch period, whereas we observed the lowest predation of all the periods on the rightmost plot. So to look at this, I removed parameters sequentially from the plot, or from the model. And first, removing temperature had the greatest effect on the notch predation risk, followed by distance from barrier. And if I remove temperature, distance from barrier, and salinity, the predicted model looks just like our observed data. So what's important about those three parameters? Well, the notch phase was January through March. So the notch phase had the lowest salinity and the lowest temperatures, both of which predicted low predation. And the PERS spent the least amount of time near the barrier. Most of these passed through the barrier relatively quickly and ended up on the other side unpredated. And so the model attributed all the predation that did exist just to the influence of the barrier. And this kind of holds true because most of the predation events we did see during this time period were in the eddies on either side of this 100-meter cut, where the PERS may have been trapped in higher predation areas. So to put this all together, I'll hand it back to Paul.
All right, thanks, Andrew. Yeah, I have some key findings and then some implications for kind of future studies. Basically, I think the biggest take home here, regardless of the model results, which are interesting to explore and we'll talk about, is that predation rate looked a lot like other studies out there. We got an overall predation rate across the whole time of about 10%. That was either lower or within the range of some recent efforts, Demetris and Michelle and Nelson. So seems like, didn't appear like this barrier was an extreme outlier, like it was a hot spot, basically. Looked a lot like the rest of the South Delta, which I guess is not surprising. It does look a lot like the rest of the South Delta. I mean, it's a big rock barrier. Look right to the left or the right, and that's what these levied habitats look like. But you could still explore within this range of predation risks that we're seeing. You can still see what's driving variability. And as Andrew said, once the barrier was in place for 5 months or so, you're seeing an increase in predation risk. And we think that this could be attributable for time for resident fish like largemouth bass to set up territories, or like striped bass even to hone in this location, to come back to it time and time again. Just a caveat, this is 1 year of study, of course. We don't have a proper control. So really attributing anything to this increase is a little problematic, but just want to make that clear. And we are seeing, though, this zone of influence of the barrier, as Andrew showed, it's kind of within 150 meters, we're seeing this increase in predation risk once this barrier was in place for a while. We also don't know if that's, you might see that at a shoreline nearby as well. We couldn't get far away enough from the shoreline, though, to really test that. 150 meters is, past the midway point of this channel, basically, but that's something that could be explored in the future. And just to make clear, we're only really looking at the near field impact. So this is really the impact of predation for fish that are already exposed to this location. Fish that are already there, this is what their risk is, right? But what is the far field impact of this barrier? I think that's something we can explore in the future, and I'll talk about that in a second. So we had the lowest predation rate observed during this notching period, and we think that that's really attributed to this high flow condition when the notch was put in place. There was really high flows, low temperatures, high turbidities, conditions that aren't really conducive to a lot of predation. So we think that's what was driving those low predation rates, but the model still predicted pretty high predation risk during that time, despite these good conditions. We think that could be related to the complex dynamics out there. I mean, you had a notch, the flow was moving through a much smaller part of that channel, creating a lot of eddies. There was a lot of scour happening out there as well. So a lot of complexity. There was a 2D velocity study that was conducted, but it wasn't really paired with this study directly. So we had limited sample size to really make much inference there. That is one of our future kind of implications. Would be interesting to have a 2D velocity study paired directly with the predation study, so you could actually see exactly where these predation events occurred in that velocity field. Are they occurring along these eddies? Is there anything interesting about these specific places where they're getting preyed upon? And then, as I said, I think a big one is looking at this far field impact of the barrier and movement in this region in general. There's not a lot of great acoustic receiver coverage in this region. So like all around Frank's Tract, which is kind of a known hotspot for predation. We don't really know how fish are moving into the Frank's Tract, moving out, getting into the West Falls River, going back into San Joaquin. So having a baseline of how fish are moving in this area would be great. And then we could then overlay, well, how does the barrier then influence that? Because you're putting a big barrier there, you're really muting the tidal dynamics. You could actually see where a barrier could be beneficial, right? It could prevent fish from moving into that Frank's Tract region. So a lot of things to explore, I think, on that overall kind of far field sort of impact of a barrier like this. And with that, I think if we have any time, take questions.
We have plenty of time. Kyle. We have a microphone.
Hey, great presentation. I noticed you had GoPros on the schematic, so I was just wondering if you were able to get IDs on any of the predators.
Yeah, I should have deleted that. We did do some like tests, I think, early on, but you know, we're at that sunset time period, and it's just the visibility to really make a call on species, I think. So we kind of scrapped it. Plus, we had to get this up. So one thing we didn't say is like we had to get this up and running in like 10 days. So we had to mobilize, build these units, get everybody together. So yeah, we never really implemented a real testing program either. So that would be great though, because you know, a lot of our inferences, we're making kind of guesses on species assemblage here, like, so yeah.
Any other questions? So is the plan to kind of continue running some of these experiments in the future?
At this location, I mean, I think, well, it depends on if there's a drought.
Okay,
yeah. Right. It depends on a lot of things, I guess, but you know, we intend to do some similar work at other infrastructure in the South Delta. Yes. Yeah. But.
Yeah. So is there, you know, one thing maybe I've seen some folks, you know, Fred Fryer showed the Eris footage from earlier, and I wonder if there was any, you know, maybe there's an like looking at these structures to see if predators congregate around them, or if it's more just a function of the predators are more dispersed, but those purrs kind of get sucked into eddies.
Good question. I mean, you know, you saw you saw like the increase in predation risk over time. It seems like these fish are honing into structure regardless, right? It might take them a little time, but then they find it, you know. And, you know, as I was saying, it is similar to the levees beside it, but there was tidal flow through this barrier too. It wasn't like it was a complete barrier to flow, right? So that's interesting to us. It's, I think, partly why it's good to explore, because what does that mean? Like fish are maybe moving in, hitting this barrier. So yeah, I guess I don't have a great answer to your question, but yeah, I think these structures are congregating predators. Now, is it how many fish are exposed, you know, to it, and like what does it mean in like a population context? I guess the bigger question.
Anecdotally, some anglers were having pretty good success getting right up to the barrier.
Just some random anglers, yeah.
That was going to be my question too. I was wondering if there was any, obviously there was some incidental fish sampling, but maybe in the future trying to pair this with actual sampling before and after.
Well, it'd be great to get a sense of the actual predator assemblage. Yeah, while you're doing this, that's, yeah, obvious pairing.
I had one other question. I know the error bars were kind of large, but it did look like during construction, there was actually lower predation risk, which seems like there's a pretty logical explanation. I mean, what are your thoughts on whether that was real or just variance?
We think it was real. So this project got funded in kind of 2 stages. And so we didn't know we were going to be funded for that second half of the work. And so on the initial model runs, that actually came out pretty strongly. And it seems like there was just a pretty big disturbance to the area with all this construction work occurring at the time. And by the time the barrier was put in, it took a little while for the predators to get back.
Yeah, good point. Yeah, and we were out there. I mean, I was waiting for the question, why were you out there in June, July, August, right? Because salmon aren't typically out there at that time, but that was part of it too. We were out there at a kind of an odd time, just we had to get out there and mobilize, and that's when it was constructed. So or hope we were lucky we were able to get back out there during the really, peak immigration time. And I think that means probably a little bit more to like what's really going on as far as the impact of salmon anyways, but yeah.
All right, well, thank you, Paul and Andrew.
Thank you.
Next up, we have David Ayers. David received his undergraduate degree from the University of Idaho. Before moving is Cyril Michelle. Cyril received his undergraduate and master's degrees from the University of California, Santa Cruz. He currently works for NOAA at their Southwest Fishery Science Center, also located in Santa Cruz, and he has led the Salmon Acoustic Telemetry Program and Salmon Predation Program. His personal research interests include how survival and migration of juvenile salmon are influenced by environmental conditions, habitat, and predation. In his personal time, he fancies himself a predator of the same salmon he works to restore. At least that's what I've heard, according to Google.
All right, thank you. And thanks everyone for showing up. I know I'm the last talk at the conference, which is not my first time to do that. But I can promise you that if you can stick around, I've got some really cool videos of salmon getting eaten by big predators to share. So yeah, I'm going to talk about, actually it's relevant to the online question, the impact of submerged aquatic vegetation on fish predation. So first, a little background, as some talks have shown, and many of you likely already know, there's really low outmigration survival of juvenile Chinook salmon in the Central Valley. Both from the Sacramento watershed. So these are acoustic telemetry studies done over multiple decades now of fish in the Sacramento Valley, as well as the San Joaquin Valley. This is a figure from Rebecca Buchanan. And to put it in context, these survival rates are extremely low compared to other major salmon-bearing watersheds on the Pacific coast. So there's something else going on in California that leads to really low outmigration survival. And one of the potential reasons and a likely contributor to that is the large populations of predators we have here in the Central Valley, in particular, non-native predators. And so a question often comes up of how can we potentially alter habitat features to minimize the effects of these predators? And if so, what features should we focus on and what would be the predicted impact of removing or modifying those features? So that particular question was identified by the Central Valley Project Improvement Act science integration team as a critical knowledge gap in their structured decision-making model. And so we, UC Santa Cruz, were funded to kind of investigate that further and try to investigate what we call predator contact points. And so we define a contact point as habitat features that are a result of human alterations to the riverscape and that may locally increase the probability of juvenile salmon being predated upon. And so when we first started this project, we kind of wanted to start from ground zero. We did a literature review on the existing contact point literature with regards to any fish. It wasn't salmon specific and anywhere in the world. So we did an extensive review of the literature and we focused on particular habitat features that are common here in the Central Valley. And of those, the habitat feature that had the most support in the literature for affecting predator-prey dynamics was submerged aquatic vegetation. And so I'm going to jump a little bit into the specific literature regarding SAV, submerged aquatic vegetation, in the Central Valley as a little bit of a background. First off, a lot of the SAV we have now in the Delta is Egeria densa, which is known as Brazilian waterweed. It's an invasive plant, and it kind of really started to take off in the late 1990s. And it quickly led to a boom in certain non-native fish populations, in particular centrarchids. So, you know, we saw some talks earlier, bluegill, red ear sunfish, largemouth bass are kind of the three big ones we think of. And those populations seem to really take off and kind of hit a plateau around mid-2000s. And at the same time, the SAV has also decreased turbidity throughout the Delta. And this schematic here this recently published schematic kind of shows what's going on here. The SAV kind of slows down water, lets incoming sediment kind of fall out, and the remaining water is lower in turbidity. And then on top of that, the SAV itself creates habitat for those centrarchids. And so you have more predators, clearer water, which results in higher predation rates. And there's been several studies that have looked into this, the effect of centrarchids of higher population centrarchids on native fish and also of lower turbidity. Some of these have been very localized, some of them have been laboratory studies, but we haven't yet really dug into the effect of salve removal essentially on predation on native fish. And one thing that's fun to note is, you know, this whole kind of schematic, this whole idea kind of seemed to take off around the mid-2000s. That does happen to coincide with when outmigration survival in at least the San Joaquin Valley started to really tank. I'm sure a lot of other things were also happening at that time, but it is something to think about. So, you know, this led us to try to figure out the impact of salve by performing a removal study. So we collaborated with Metropolitan Water District, who owns Bacon Island, and we were able to have full access to the island and its shoreline, and we picked the ideal location to do a salve removal study. And so for this, we really wanted to do a Baki or before-after control impact design with blocks. And that would really allow us to try to control for other changing factors in the river and accurately measure the impact of salve removal. So we delineated a 300-meter piece of shoreline that was about 7 meters. The part we focused on was 7 meters wide away from bank, and that's because that was approximately where the salve went out into the channel. We broke it up into 6 50-meter blocks, and then we overlaid essentially a virtual grid. And the grid cells were 2 meters wide, so 2 meters along bank and 1 meter out. And we didn't sample in the first meter because that first meter was often dry. So essentially we had 6 meters going out, 150 cells along shore for a total of 900 cells. And then we approached those cells with pole pers. So you've heard about floating pers. We had to adapt it to be able to work in kind of shallow water environments. So we essentially attached the whole mechanism of pers, but to the end of an extendable pole, and a pole that can be extended 7 meters out. So every half hour, we would change the length of the pole to fish the appropriate grid cell. On top of that, we also perform boat electrofishing before and after the salve removal to kind of get an idea of fish distribution and fish community in the sampling area. So we sampled before the salve removal for 2 weeks, and then we performed a salve removal at 3 of the 6 blocks. And these are the actual blocks here that the salve removals were performed in. And then we did 3 more weeks of sampling after the salve removal. This is kind of what it looks like. Here in the picture, we have, I think, about 9 poles that you can probably see. There's some out here, there's 3 more here, there's 3 more here. We would sample cells randomly without replacement within each block, and then we'd start over once all 900 cells had been sampled. In total, we used 18 pole pers, and they were deployed for a minimum of 30 minutes each, and 3 per block or section. Daily sampling occurred 3 hours before sunset until 1 hour after sunset, again, to maximize predation, which is our response variable, like in the other studies we've seen. And in total, we were able to get out about 75 to 100 pole pers, so fish 75 to 100 cells per night. And it occurred from April 11th to May 15th, 2022, with the salve removals occurring during a 3-day effort from April 26th to April 28th. A little bit on salve removals. It took a lot of people. What we found out was having scuba divers was kind of a critical component to the project. So we were able to get some NOAA divers to help us and a lot of extra crew to kind of gather the floating strands after they'd been removed from the sediment and put them away. We got approval from the local landowner to let the salve kind of be distributed across their fields and have it just desiccate away. In total, we were able to remove 16.2 cubic yards of vegetation. This was its size, essentially pre-desiccated in air. So this is kind of what it looks like immediately after. This is our site. I've kind of broken the site, the 300-meter long site, into 200-meter chunks just to make it easier to see on the screen. The removal areas here, as you can see, from this fixed wing drone imagery is much clearer. The salve was completely removed. The scuba divers went through a couple extra times and confirmed that pretty much every strand was gone out of the removal sites. And then here's kind of another view, and also for scale, you can see a boat. This is kind of the salve removal site here, and then here's another salve removal site here. So before I jump into results, it might be fun, given the background I've given you, a show of hands. So if you remove salve, let's see how many hands would predict that predation would decrease, AKA survival would increase. Okay, how many people think that nothing would happen, no significant change? Okay, and how many people think that removing salve would decrease predation, increase survival? Okay, I think you guys, seems like most people chose the third option, which is what we thought would have happened. But let's go through it. So the number of pulpers that were predated upon was approximately 25% for the before control treatment, about 30% for the before impact treatment. It was about, I think, 38% for the after control treatment. And then it was almost 90% for the after impact treatment. So essentially, once you remove salve, predation risk goes up by at least an order of twofold increase. And not only that, but the predation happened really quickly. So this is the median time to predation. So kind of the before period, it took about 270 seconds median time to predation. After control was 66 seconds, which is definitely quite a bit faster. And I think that partly had to do with increasing temperatures as the study went on. But then in the after impact, it took a median time of 11 seconds for predation to occur. And usually it happened within 1 to 2 seconds. So here's that video I promised. You're going to see videos of both largemouth bass eating smolts, striped bass eating smolts, and one video of the two competing for a smolt. Let's see here. So the pulper is being deployed here. That's the live smolt. In the water. Being lowered down, and it's gone. So you'll probably be able to see it, but this one's a largemouth bass. This one's going to be the video where you're going to see a striper and a largemouth bass competing for the smolt, and you'll see who wins. Despite all odds, the largemouth won. That's not usually the case. So that's the magnet you see hanging down. Once the smolt gets eaten, it pulls a magnet off of a timer and starts the timer. This is the same video again. There's not much to be learned from this. It's just fun to watch. Okay, it's in. It's gone. This is pretty typical for stripers. When striper predation events occur, it's instantaneous. Largemouth tend to take their time and kind of investigate a little bit. And it's gone. Okay. All right, that's probably enough. So the results. So we deployed 1,700 P per deployments, of which 48.5 were predated upon. So this is still preliminary, but so far we've used a mixed effects logistic regression model to look at the impact of salve removal, which, you know, is pretty stark. So we don't necessarily need the model to tell us what we already know, but nonetheless, these are the different factors we use. Before, after was a factor, control impact was a factor, distance from shore with 2 meters being the intercept. And then finally, the before after control impact interaction, which is really what we're testing here. The ones in bold are the ones that are significant. And what's important here is to see that the before after control impact interaction was a significant factor. These are the model predictions for the different treatments as well as distance from shore. What we do see, which is interesting, is in the before control, before impact, and after control, the ones where the salve's still present, the highest predation rates are at the furthest outside deployment, which is typically right beyond the salve. So kind of that open water habitat right beyond the salve. And I think that's telling of what's going on in the system. In the impact after with the salve removed, it was near 100% predation for essentially the deployments 4 meters out to 7 meters out. We also wanted to quantitatively measure kind of the vegetation. So we used normalized difference chlorophyll index from a multispectral imagery from the fixed wing drone. And this is kind of what the site looked like before salve removal. So hotter colors represent more vegetation. This was 1 week pre-removal, 2 weeks post-removal. This is what it looked like. You can see a pretty much total absence of vegetation in those removal sites. Sorry. And then this is what it looked like 6 months post-removal. So we're trying to also find out how quickly the treatment sites come back to normal. We still see a significant effect. So this is, these are box plots of the NDCI values. So for the 6 months post-removal, we still see a significant effect of the removal sites on the control sites. And we're hoping to do one more 1 year after. And so we were able to deploy GoPro cameras in the after period. We didn't put any out in the before period. But what we did see was the majority of the predation events were from striped bass. So 149 from striped bass, 24 from largemouth bass. And all of those kind of outer predation events that I was talking about right beyond the salve tended to be striped bass predation events. The largemouth bass were kind of more distributed across the entire 7 meter width. So this is largemouth bass predation events in the control areas, in the removal areas. This is striped bass in the control areas, striped bass in the removal areas. Essentially, striped bass, once you remove the salve, the predation events are occurring almost in the entire cross section, with the exception of the very closest grid cells. As for the electrofishing, this was done with the help of Fish Bio. We did 3-pass electrofishing in each block in the before and in the after period. In addition, we pit tagged all of the kind of predator-sized fish that we captured. So what we did see was in the before period, we kind of had similar, these are just counts, similar numbers of largemouth bass in kind of the first 2 blocks, and then similar numbers in the remaining 4 blocks. And then the after period, so the light blue cells are the no salve blocks. We saw an increase in the number of largemouth bass. Shoot, I don't know if I got these colors wrong. Yeah, and there should have been. There was an increase in the number of largemouth bass in. Oh, this is embarrassing. Control, yeah, control was A, C, and E. Yeah, I got the colors all wrong. All right, so these are the areas that had salve, and B, D, and F are the areas that did not have salve. So we saw an increase in the number of largemouth in the salve areas, and essentially, comparatively speaking, a decrease in the number of largemouth bass in the no salve areas. And the result of the pit tagging was we did get 13 recaps, which were all largemouth bass. We saw 6 of the bass move from control to control, essentially stayed put. We saw no bass went from control to impact. We saw 4 bass went from impact to control, and 3 bass stayed put in impact. So there seems to be some evidence that perhaps some largemouth bass, once the salve was removed, moved over to areas that still had salve. And here is kind of the spatial distribution of those predators. So the blue fish are striped bass, the red fish are largemouth bass. The removal sites are the 3 bottom ones, the control sites are the 2 top ones. As you can see, this is from the after period. There's a lot more largemouth bass in the control sites and less so in the removal sites. And also the striped bass are primarily in the removal sites. And we did see a significant difference in largemouth bass abundance estimates in the after control compared to the after impact. So in summary, we found that removal of SAF led to a decrease in LMB abundance. However, we also saw that a removal of SAF increased largemouth bass and striped bass events. Ultimately, predation risk was 2 times higher in the SAV removal areas. Predator efficiency appears to just be higher in the open water habitat, similar to predation above SAV in the control sites at kind of that 7-meter grid cell. Hand removal of SAV did require significant manpower, but it does result in complete and near instantaneous removal, which is useful in this sort of study. We did develop a new standardized and randomizable methodology for measuring predation in the nearshore environment, which could be useful for other studies. And I think one of the take homes too is there's a question of scale here. So obviously, if you remove SAV from large swaths of the river for extended periods of time, eventually you might imagine centrarchid populations would decrease, and that would actually ultimately lead to potentially lower predation rates. Nonetheless, the striped bass were the major predator here. So that is not, still to be determined. And with that, thank you very much to our funders, the field assistants. This was a CVPA project with funds distributed by U.S. Bureau of Reclamation, and land access for Metropolitan was also critical. Thank you.
We are pretty much out of time, but we do have a couple online questions, so I'd like to give those people a chance. The first is, and you kind of addressed this, is the increase in predation from SAV removal an issue of spatial scale? Did you simply increase the amount of SAV edge habitat where bass like to forage?
Yeah, and we're going to include that more formally in the model. I think the SAV edge habitat is a critical piece of the story. So I think we should be able to include in our model some kind of distance from near a SAV edge, and that might be an important predictor in predation risk.
All right, and we have another one. Do you think that the technique interacted with treatment? It would be easier for a visually oriented predator to detect an impaired prey item in open water relative to SAV. Prey would not have that same behavior if their movement was volitional.
Right, yeah, I mean, I think there is some element where prey could potentially do a better job hiding in the SAV if it wasn't tethered to a pole. And that's certainly another kind of line of evidence that needs to be investigated. But I will say, as is often the case with striped bass predation events, I don't think there's really a lot of time to evade a striped bass. These predation events are nearly instantaneous, and the striped bass are swimming really fast. And so maybe with a largemouth bass, there's potential to escape, but I'm not so sure about the striped bass.
All right, and we have one more. I think this will be short. Did you look at the effect of time since SAV removal on predation risk? Or I would add to that, is that something that we should look at, like a longer time frame, like you suggested, kind of?
Well, within the data we already have, we can certainly add that as a covariate in the model. But anecdotally, the minute the SAV was removed, predation risk went up to almost 100% in the removal sites. But there is value in the longer period to potentially go back out to Bacon Island and do this study as it stands with whatever level of impact still exists. Although it probably won't happen, but maybe a future study could look at a longer time frame.
All right, well, I want to thank all our speakers. Go Predation Project Work Team.
We're still looking for speakers for our May meeting, so if anybody wants to come to the predation meeting.
All right, just one additional item I think we have on our agenda for today. So as Ted makes his way to the front, on behalf of the Planning Committee and everybody involved with IEP, thank you for your attendance this year. I think we've demonstrated the value of doing this in person. Our plans will be to do this in person again next year. So with that, Ted.
All right. Well, I am here for the very exciting job of announcing the winners of the Early Career Presentation Awards. So this was an opt-in award in that we used a definition of early career as someone who was within, 5 years basically of starting work in the Delta. So it kind of spans the gamut from students to SciAids to new ESs and postdocs. So there was a whole bunch of really, really great talks, and we had a lot of really tough choices to make. I really appreciate the effort of all the judges who took the time to take detailed notes on all the talks. Thank you all very much. And so without further ado, I will start off with the Best Poster Awards. So for the third place award for Best Poster goes to Andrea Jaggi from USGS on her poster for identifying microcystin sources and producers in San Francisco Bay. So give her a round of applause. And the second place poster award goes to Diana Munoz from UC Davis and USGS on her poster on long-term data set reveals black crown night herons nest survival is primarily associated with bottom-up processes on Alcatraz Island in San Francisco. And the winner is Alyssa Dahl from CDFW on her poster, confirmed presence of green sturgeon in the Stanislaus River. Right, so for the Best Talk Awards, we actually had a tie for second place, so I will announce these both at the same time. Our second place talks were Letitia Cabol from UC Davis on fitness trade-offs. Pardon? Okay. So if I mess up your name, I'm really, really sorry. Fitness trade-offs for migratory and resident Delta smelt in the San Francisco Estuary, and Emily Richardson from USGS, a multi-parameter approach to modeling light attenuation in the Delta. And our winner for first place winner for Best Talk is Madison Thomas from DWR on controlling invasive Phragmites australis at a restoration site in Sassoon Marsh. Okay, so all of these winners will have their presentations highlighted on the IEP website. And so let's just finish it off with one more round of applause for all the winners and all the contestants.
All right, we're adjourned. If you left the poster upstairs,